contamination. These are called migrants leaching from packaging materials. Propagation and concentration of pollutants through the food chain is another aspect. These pollutants and carcinogens belongs to many crops. They may include metals and metalloids, polycyclic aromatic compounds, hydrocarbons, persistent organic pollutants, perfluorinated compounds, pharmaceuticals and personal care products, radioactive elements, electronic waste, plastics, and nanoparticles. Some of these pollutants occurs naturally in the environment, whilst others are produced from anthropogenic sources. They may contaminate our food crops, livestock, and seafood. Let's go to the metals and metalloids. Metals and metalloids in the environment have various sources. One source is industrial regions. They often have extensive environmental contaminations by metals. Arsenic, mercury, selenium, lead, cadmium, copper, and zinc are the prominent to contaminate soil, and hence vegetables and other crops grown near metal plants exceeding standards for the maximum permissible levels and pose a potential health risk to people from consumption. In most of the cases, ingestion of the food is a major exposure pathway. In the global we see methyl mercury has been detected in fish and other seafoods around the world. Fish tissues are contaminated with copper, iron, zinc, and manganese, exceeding regulation limits. This methyl mercury bioaccumulates in the fishes, and then biomagnification goes across the food chain. Major sources of these heavy metals release includes natural sources like rocks, volcanic eruptions, dust particles, aerosols, sea sprays, agricultural sources like inorganic fertilizers, pesticides, waste phosphorus, fungicides, sewage sludge, fly ash. Similarly, industrial, domestic, and miscellaneous regions are there. They discharge heavy metals in the gaseous or liquid or solid form, and from there these pollutants enter into the soil, water, and air from there where they leach down into the food chain. That's the process how this flow. These metals from soil, air, and water, they are uptaken by the students, or they are deposited by the plants, or their decomposition into the soil. From there, they bioaccumulate into the food, and from there, biomagnification occurs, and there are health risks for the human beings, and they can be corrected by bioremediation processes, which we may discuss during the round proceedings. The possible health hazards due to exposure of these heavy metals are, I have listed some of them here from the literature. For example, lead that leads to complications in the nervous system and red blood cells. It also has reduction in cognitive development and intellectual performance, death among the children. Similarly, cadmium, it causes renal tubular dysfunction associated with high risk of lungs and breast cancer. Arsenic is associated with dermal, respiratory, nervous, mutagenic, and carcinogenic effects, so on. Another class of pollutants includes polycyclic aromatic hydrocarbons. These polycyclic hydrocarbons primarily occur after organic matter undergoes incomplete combustion or pyrolysis or from industrial processes. Food contamination comes from the environment, industry, or home cooking. These compounds appear to be genotoxic and carcinogenic. Oil spills from the transporters, ships in the ocean are all too common and will result in contamination of seafood. Besides the petroleum-related polycyclic aromatic hydrocarbon compounds, chemical dispersants are often used to mitigate effects of oil emission, which ultimately pass on to the fishes and all seafood and ultimately affects the human beings. The possible hazards of these polycyclic hydrocarbons includes mutagenicity and carcinogenicity, DNA damage and oxidative stress, impaired breast fertility, respiratory diseases, and conjunctive dysfunction among children. Similarly, Another category of pollutants and carcinogens, they include industrial chemicals. Persistent organic pollutants, particularly are synthetic organic chemicals. Some are used in the industry, some are pesticides, and some are byproducts from industry or combustion. They include pesticides like herbane, chlorine, and DDT, industrial chemicals like polychlorinated biphenyls and hexachlorobenzene, and unintended byproducts like dibenzodioxins and dibenzofurans. They persist in the environment, are distributed globally in air and ocean currents, and accumulate in animals, the food chain. Their side effects depend on the chemical and contaminated species. For example, they may have effects on reproductive or immune system or increase cancer risks in human beings. These persistent organic pollutants, they are extremely resistant to natural breakdown processes in soil by bacteria, mold, and in animal species. They persist for years in the soil, even for decades. They remain in the environment. Many are also highly toxic and build up or bioaccumulate in the fatty tissues of animals and humans, and they are also harmful to the body and tissues. Some of the chemicals like chlorpyrifos, organochlorine pesticides, pyrethroids, and PCBs, they have been found to be destructive to the human health. The possible risks of these chemicals, they include neurological symptoms, endocrine disruption, infertility, and fetal malformation. Similarly, dioxin and PCBs, they have language delay and neurological disorders. Then pharmaceutical and personal care products. The term pharmaceuticals and personal care products include a wide range of substances that may enter the environment and hence food and water sources. Antimicrobials and other drugs may originate from use in both humans and animals. For example, swine waste containing animal microbials may contaminate both water and food. So we have already discussed these antimicrobials in our earlier uh, workshop organized under NAGP, and there were very good recommendations, and we have already come up with a booklet on that. Besides that, from very real threat of increased antimicrobial resistance to exposure to exogenous sources of these chemicals, it has also been shown that many drugs have their other side effects, including endocrine disruption. In some of the circumstances, the medicinal products themselves may be contaminated. For example, in many of the herbal products, we find such type of contamination. These are some of the possible health hazards I have listed here from the literature. Then another is radioactive elements. Normally they are not present in nature, but after the tsunami damage affected the nuclear plant in Japan in 2011, they become important because they have affected the food chain seriously. Radionucleotides have also been detected in seafood in India, various foods in Balkans, and food and drinking water in Switzerland. So these are also important pollutants which need consideration. Then another electronic waste, modern society has become encumbered with many electrical devices and electronic waste, but which we call as e-waste, that has become a major problem nowadays. Inappropriate processing, for example, incomplete combustion of such products releases a variety of pollutants covered, including PBDs, dioxins, PHS, PCBs, and metals and metalloids. In addition, contamination from such devices can enter drinking water and food also. The major pollutant is plastic, which is of concern nowadays, because in recent times we rely more and more on packaging material, in particular plastics, to transport and help with the food. These materials are also inert and they themselves contaminate food, and drinks as multiple chemicals are released into the food and beverages from food contact materials. These are termed as migrants and include such chemicals as pellets, plasticizers, which have been detected in bottled water. Factors such as higher storage temperatures and prolonged contact time with the packaging were linked to higher levels of contamination. But a health risk assessment shows that the risk for consumers is low as now. But this is a concern for future. which we may discuss in the proceedings. Another recent development is that of nanoparticles. Nowadays, we are coming up with many nanoparticle-based products like nanofertilizers, nanopesticides, etc. These nanoparticles, they have one dimension less than 20 from -7 and millions, and genuine nanoparticles have been used in quite a range of in plant, paints, cosmetics, pesticides. Pathways and effects of these in biota are as yet unclear, but they have been shown to travel in chain. Nano-sized materials have been detected in foods such as wheat-based products, so they are also of concern and they need to be looked into. So, based on these uh, points available in the literature, we may conclude that it is important to perform the assessments of the associated potential risks, the protection of the environment and human health from the adverse effects of pollutants and carcinogens in food chain requires significant capacity in a variety of areas, which means monitoring and testing of resources, environmental and product contamination, as well as human exposure. Monitoring contamination levels, enactment of control measures, including remediation and consideration of socio-political implications, are vital to safer food. Supervision of chemical production, use and disposal, including production of waste, of operator health and safety, and control and monitoring of releases are important to control the pesticides in chain. Simultaneously, identification and application of alternatives and controls to these pollutants is again important. Awareness on awareness of pollutants and carcinogens in food chain should be intensified. Monitoring of illegal trade is important as we can see that many of the banned pesticides are in the market and being used. Legislation including control of manufacturing, registration, use and disposal of pollutants and carcinogens food chain, with particular emphasis on public awareness, cleaner production, legislation, increased capacity, and available of financial resources will assist in alleviating some of the problems that exist with pollutant carcinogens in food chain and search for alternatives of these pollutants. Research is also required to further assess the threat and to find out other methods to reduce emissions to the environment. So, with this background, this uh, brainstorming session and workshop was planned, and during the day of discussions, we have just 5 eminent speakers who will be deliberating on their impacts as well as remediation and regulations, etc. And I hope by the end of today, we will be back with some concrete recommendations to control these pollutants and carcinogens in food chain. With these words, thank you all for patience. Thank you.
Thank you, sir. Now I request Apurva to give a flower word to Dr. Anupam Dixit, speaker. Apurva. Sorry. And to Dr. Anushri Malik, who has come from IIT Delhi. So, I request Honorable Vice Chancellor and Chief Guest of this function for his inaugural address and blessings to the
Sir, please.
meeting
Very good morning to all my colleagues, deans and directors of this university, Professor Shamsher, Dr. Nasrana, Dr. Anil Siroi, of the country, dear students, under innovation grant of National Agricultural Higher Education Project, FICEA is organizing a brainstorming session and workshop on involvement and impact of pollutants and carcinogens food chains today. I'm very happy to know about this workshop because it has been organized on a very specific topic. I hope that at the end of the day, after the brainstorming session and of the workshop, we will have certain recommendations which are implementable in today's context. I remember that in the same hall, we had earlier workshop and brainstorming session which Dr. Sirohi has already mentioned on antimicrobials. That was a very successful workshop, and I have won proceedings and recommendations of that workshop. We are all aware that the environmental degradation caused by pollution, deforestation, climate change, biodiversity loss, declining soil fertility, ozone depletion has drastically impacted human, animal, and plant lives today. Rapid industrialization is the foremost factor for general pollution and increasingly prevalent toxins in the environment today. It is established that pollution of environment is caused by about 51% by factories or industries, about 25% by vehicles, about 11% by household activities, about 8% by agriculture, about remaining 4% by other activities. And we know that burning of fuels emits various chemical pollutants such as sulfur dioxide, nitrogen oxide, ozone, and other gases. The majority of pollutants are classified as carcinogens and are serious threat to human health and habitat. Mycotoxins, heavy metals, pesticides, herbicides, and other chemicals derived from industries are not only impacting animals and human health, but the ecosystem itself. I'm glad and very happy that Dr. Anil Siroi in the beginning has presented an overview this topic, and many of the things which I am talking are just the repetition of that. But to my knowledge, I'll get at least benefit from his presentation and of course, what I'm going to present myself. Then pollutants which are not biodegradable can remain part of the ecosystem for long periods of time. Many contaminants spread throughout the food chain due to bioaccumulation as they are known to accumulate in plant and animal tissues. Pollutants enter human chain by way of ingestion or inhalation. Plants uptake toxins directly from soil. When herbivores consume these contaminated plants, the toxins accumulate in their tissues from where they pass up to the carnivores. At each topic level, the toxins become even more concentrated. The process of biomagnification continues up the food chain, getting most harmful top predators, including humans who eat meat or fish. Polychlorinated biphenyls, the PCBs, polycyclic aromatic hydrocarbons, or the PAHs, heavy metals, some pesticides, and cyanides are all bioactivators. Polycyclic aromatic hydrocarbons can accumulate in sea animals and have been linked to cancer in humans that eat fish. Heavy metals include cadmium, chromium, cobalt, lead, mercury, nickel in environment are dangerous to animals and human alike. Persistent organic pollutants as PCBs are toxic chemicals resulting from industrial production that stay in the environment for a long time. Pesticides widely used in agriculture are persistent on crops even after harvest. We all are aware of the massive Exxon Valdez spill in Alaska in 1989 that badly affected marine ecosystem, which could not be fully restored despite the enormous efforts. In this, hundreds of thousands of species were killed. Deepwater Horizon oil spill in 2010 was also damaging to marine habitats, and oil from this accident entered the food chain in Gulf of Mexico. In addition to the heavy metals, pesticides, pathogen, particulate matter, emerging environmental contaminants as microplastics, personal care products, pharmaceuticals, electronic waste, nanomaterials, etc., pose serious health risks such as cancer. More research is required to know the effects of these novel pollutants on food safety and human health. Plastics, we all know, can exist in the environment for hundreds of years. They are going to stay the culture for about hundreds of years. Microscopic plastics debris called microplastics transport from land to river and finally to sea, and then from environment finally to human beings via food web. Attention should be focused on the environmental impact of microplastic particles found in the freshwater, ocean, and soil. In rapidly developing the field of nanotechnology, information about toxicological impacts of engineered nanomaterials and their potential health hazards is urgently needed for development of strategies to protect public health. While talking about the approaches to mitigate the input and impact of pollutants in the system, scientific advancements in fields of nanotechnology, smart materials, green chemistry, biotechnology have power to protect the planet's ecosystem. Promising sustainable remediation approaches are being developed to restore the ecosystems. Strategies to protect environment and ensure food safety involve use of biosensor for rapid detection of contaminants and use of biodegradable material for food packaging. Novel methods for monitoring contaminant level in crops, livestock, seafood, and drinking water are vital for global food safety. Also, national and international policies are regulating production and use of such substances. For example, the Codex Committee on Pesticide Residues has established maximum residue limits, is MRLs, for pesticide residues. There is much more to speak. I hope the speakers of the workshop will cover many of these aspects in detail, and the participants will be benefiting from these deliberations. I once again appreciate the efforts made by the College of Biotechnology by providing a platform for brainstorming session and workshop on involvement and impact of pollutants and carcinogens in food chains And I just believe that this is a very important topic and of great interest to all of us. The workshop is aimed to provide a platform to academicians, scientists, and students to discuss the scientific efforts and provide a glimpse of innovative trends and scientific research for global food safety and conservation of environment. I'm convinced that the workshop will be very useful. The researchers, faculty members, and students as well. I wish the workshop great success. Thank you very much.
Thank you, sir, for your blessings. Now, I request Director Experiment Station and Dean College of Biotechnology, mentor, Chief Guest of this.
लो ये देखो कौन आ गया
Rohit,
वो तो पीछे है मोटी मूछ वाला आदमी इसको बहुत गर्मी लग रही है तो मुझे एसी बढ़ाओ मुझे हार्ट स्पॉट तो मैं देखो ना चल तो रहा
It's a organizer.
देखो तो देख के आ गया फोटो खींच रहा
thank you,
होना
Dean
चाहिए
College of Biotechnology, Station for the Chief Guest. Now, I have been interested to the input of thanks. So, on behalf of organizing committee, I, Dr. Kumar,
लाल
I thank one and all present here. First of all, I would like to thank
ऑनलाइन ले लो हम नहीं जा रहे कहीं
guest of today's function and Honorable Vice Chancellor of this university who has taken pain to for this IG grant of NIHEP. In getting this grant has taken a lot of pain and allowing us to conduct this workshop and under this NIHEP project. I thank the Director Research and PI of this project. I also thank the Dean College of Biotechnology and the
ऐसी बढ़ाओ नहीं इन्होंने सारा
direction this college has been well and organizing this workshop. I am thankful to deans and directors of the university who have come here and addressed us directly or indirectly who have not come, and I thank them also. I thank all the HODs, faculty members, participants of this workshop, without whom we could was not possible. Registrations were there. So common, but around more than 10. I thank whatever had and yesterday, today also, and I thank one and all Dr. Mr. Manoj Sekhar, who was busy yesterday, but in seminar, but he came and arranged all the online arrangements yesterday, and today also he has come with his team. So if I forget anyone, I feel that you accept my thanks from the organizing committee. Now, I request Apurva and her team for national anthem, and I would request all to stand up, please.
बने अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंग। विंध्य हिमाचल यमुना गंगा उज्जल जलधि तरंग। तब शुभ नामे जागे तब शुभ आशीष मांगे। गाहे तब जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता। जय हे जय हे जय हे जय 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 हे।
So before I seek permission from our Vice Chancellor, in session, I would request all of you to time for mic. So I would seek permission from our Vice Chancellor. Thank you, sir. Thank you.
अब ठीक है। अब देखिए मूव कर रहा है क्या? मैंने वो रिक्वेस्ट किया है। ठीक है, तो मैं इसे ओपन करने दूँ। माइक ऑन रखो। माइक वहाँ कर दिया।
Welcome all to the first technical session of today's workshop on involvement and impact of pollutants and carcinogens in the food chain. I welcome co-chair Professor Anil Surohi, rapporteur Dr. Purushottam and Dr. Harshit Verma in this session. We are honored to have among us distinguished speakers, Dr. Anushri Malik, Dr. Manoj Tripathi, and Dr. Anupam Dixit in this session. And our first lecture will be from Dr. Anushri Malik on bioremediation of waste streams for agricultural applications. It is my privilege to give a brief introduction of Professor Anushri Malik. Ma'am has a distinguished career, she being gold medalist in MSc from Jabalpur University. And I will like to motivate our students, inspire my students who are appearing for next JRF, that ma'am was qualified CSIR JRF in year 1994, and rank was among first 10, and she was among the first 10 ranks in the CSIR JRF examination, which is a very prestigious examination. Then she completed her PhD from IIT Delhi. After PhD, she went for a postdoctoral by acquiring the prestigious JSPS fellowship from Japan. And after completing the postdoctorate, ma'am was appointed as professor in JNU in environmental sciences, where she worked, where she taught the students for about 1 year. Then she shifted to IIT Delhi in Center for Rural Development and Technology. Ma'am has completed many international projects in her distinguished career as Indo-Sri Lanka, Indo-Brazil, Indo-Dutch, and has guided about 20 PhD students. She has got many good publications in the journals of national and international repute and has more than 9,000 citations on Google Scholar. She has She has the recipient of the prestigious award DST Lockleed Martin Award, and also a BRSI National Fellow, Chair Professor and Head. And it is indeed a pleasure to have ma'am among us. Also, we have Professor H.K. Malik, who is also in IIT Delhi, and man's better half. And sir is a plasma physicist, and he is a JSPS fellow, ERD fellow, and also European Erasmus Mundus fellow. Sir has guided 30 PhDs and more than 1200 MTech, MSc, BTech students. So we welcome Ma'am Anushri Malik and HK Malik, and I now invite ma'am for the enlightening talk. I will first ask Dr. Anil Sirohi to chair this session and give some motivating words.
Welcome to this first technical session of this brainstorming session on environment and impact of pollutants and carcinogens. And as depicted by Honorable Vice Chancellor and myself in the inaugural session, another truth is that we cannot remove these chemicals from the system. The best way is to control them, and one method to control them is remediation, that is to keep away these metals from the these pollutants away from the environment, so that we can prevent them to enter the food chain. So first session is dealing with the bioremediation of these pollutants. As highlighted by Dr. Rekha Dixit, we have very eminent speakers in the area. Dr. Anushri Malik from Center for Rural Development and Technology, IIT Delhi. We have Dr. Anupam Dixit, who has pioneered in basmati rice research and is serving as a chief scientist in our nearby institute, BEDF. And another is Dr. Manoj Tripathi, who is principal scientist at Central Institute of Agriculture Engineering. So, to deliberate on the issue, how to remove these pollutants from environment so that our food chain becomes a little bit safe. First of all, I request our guest speaker, Honorable Dr. Anushri Malik, to speak on bioremediation of waste stream for agricultural applications. I'll request Dr. Purushottam and Dr. Harshit Verma to kindly note down the important points which may be included in our recommendations of the workshop. Madam, please.
So thank you, Dr. Dixit and Dr. Sirohi for a nice introduction about me and about the workshop. And it's a great pleasure to be in this brainstorming session. And I really look forward to a very good interaction and feedback, especially from the students, on this topic, because this is a great learning opportunity in my opinion. So my lecture, as you see, is titled Bioremediation of Waste Streams for Agricultural Applications. And this has been a challenging journey because all of us are in biotechnology, and we know that somewhere people have a perception that these things work in lab, but they may not be applicable in field. So if you want to develop something which can really help society, First of all, you have to convince the people on board who are decision makers that no, my work is going to be useful for the people. I want to take it to the field. That is the big challenge. So, but then the challenges teach us many things in the life. So, my lab at IIT Delhi is called Applied Microbiology Laboratory, and one of my PhD students designed this logo, which I take very pride in putting in my lectures. And the motto of the lab is resolution for green solution. And we work with what you see within this logo, bacteria and algae and fungi and plants. And we are trying to see how we can develop green technologies. So moving on to little bit introduction. आपकी डाउन की कहाँ पर है? Okay. So friends, I will link my work to the very topic of this brainstorming session, and that is about the water, right? Yeah. So I want to talk about this slide. Water use in India, and all of us know that as this pie chart is showing, that the water use, the maximum water use is in irrigation. That is for the agriculture. And if we look at the scenario in the coming years for 2025, then also there will be some changes, but still agriculture remains the largest sector, which will be basically drawing the water for its usage. And because the demand is so high and the availability of water is almost all fixed and it is decreasing day by day, especially when we say fresh water, so rainwater for irrigation, and we really cannot blame them for it. So my topic and my work is related to wastewater. And since we are people from different backgrounds, it's nice to know that wastewater is a kind of final product that can come from various sources like household, agriculture, hospital, and industries. But the word wastewater has a lot of, perception associated with it. Whenever you say this is irrigated by wastewater, our first reaction will be no, this is bad. But it may not be so, because some of the wastewaters, the grey water in rural areas, or the agricultural runoff may not be that wastewater from hospital and industries are going to be. So we need to actually look at this perception, because eventually we will have to go for wastewater reuse. Then we characterize the wastewater based on what kind of contaminants it has. So like the organic matter, which we monitor as biological oxygen demand, BOD, or chemical oxygen demand, COD. Then the nutrients, heavy metals, trace organic compounds, or the emerging compounds. There are various categories to define and measure. So we are still struggling to precisely, are they really hazardous? Are they going to cause cancer? And so on. And as the workshop from the morning you have listened, it has impact on health, environment, and economy. Now looking at what is the scenario of wastewater, treatment capacity. So if we look at this, then we understand that the level of development is correlated with the degree of wastewater treatment facility available. So the less developed countries, they have less capacity. And if you plan to use wastewater, you are sure that it may be untreated. So now we come to the Indian scenario. And in this particular chart, what we see is that if we go from metro cities to class 1 cities to class 2 cities, the availability of wastewater treatment facilities decreases very significantly. So we can see that around 68% of the wastewater is untreated. But in this data, we don't even find a bar for a village. So what is the situation in the village? I think we don't have properly developed treatment capacity for the villages, right? So assume what's going to happen in the village. So anyway, we were focusing on rural and peri-urban scenario because the market is in the urban areas and peri-urban areas more. And we know that the rain and river water in such areas is extensively used for irrigation of vegetables, which can be sold directly at the peri-urban centers. And there is a belief that these vegetables or whatever we harvest will have contamination of heavy metals and pesticides. So then what can our expertise biotechnology do in this scenario? Can it come out of the lab and contribute here? But since we have to work in the country, we have to look at the ground, we have to look at the ground reports, and we find it, and there are a lot of contaminants which are some more that what is the heavy metal concentration in the rivers or other water bodies, because all the eventually of the metals we were talking in the morning, chromium, copper, cadmium, lead, zinc, and nickel, and sometimes multiple metals are above the permissible limits. And this comes from across the India, Kali River, Maharashtra, Yamuna River. And accordingly, there are a number of studies in the literature which say that the vegetables, they are accumulating these heavy metals. And here we see some of the examples. So yes, the problem is real, and we need to find a solution for it. And besides, these heavy metals coming in food and impacting our health, they are also deteriorating the soil gradually. So the microbial activity, the enzyme activity, the pH and the cation exchange capacity is deteriorating because of the water which is contaminated. So we require some remediation process, but then it should be cost effective and it should be able to handle the bulk and it should be environmental friendly. So the challenge here is that huge volume of water is required for irrigation and the contaminants are diluted. So the chemical and physical methods for heavy metal removal do not work here because they can work better when you have highly concentrated effluent which like coming from the industries. And the difficulty in removing trace amounts of contaminants, treating large volumes is a very big practical problem. So the research questions that we designed were looking at this scenario where that which technologies have potential for creating a field-worthy treatment system. Then what kind of elements we should investigate to move in that direction. Do biological techniques have a scope? Because constantly we are facing this criticism. And then there are so many contaminants. So can we focus all? Probably not. So what are the major contaminants we should target and why? And how do we evaluate the direct and indirect impact of the treatment system? Because then only we can show that yes, it is worthy. And then since I had some friends in ICAR, so we also deliberated that whether it is the adverse impact of the wastewater irrigation. And then we thought why not employ a consortium approach to this complex problem? So the problem is complex. There are metals, there are pesticides, there are organics, and only one bacteria or fungus or algae cannot treat it. So we must use a consortium. Consortium is a group, right? So we were working to look at consortium, and learning from the nature, we saw that yes, consortium is going to be better because they can attack the pollutants at different sites. They have a very rich metabolic network, which is very useful for pesticide degradation or other organic degradation. They have a very beautiful division of labor, like you would have done it for arrangement of this workshop. And the consortium are more stable to the environmental fluctuations and can lead to effective bioremediation. So we had been working on several single organisms, and we could publish good papers that organism A is doing very good for chromium removal, B is doing very good for lead removal, and so on. But when we expose them to a cocktail or a mixture of contaminant, then what we see is that, in this graph, which is on the upper side, can I select enter? Which one? Laser pointer. So friends, sorry, the pointer is not available. So what we saw that y-axis in this graph, you see relative reduction in the biomass. So if any bacteria or organism has to work, the first indicator is it should grow in that environment. So in this graph, what say Aspergillus, Lentulus, Pteris, or Rhizopus oryzae, they may be doing well with one contaminant like chromium. The reduction is 30% of the absolute biomass. But as soon as we expose them to mixture of pollutants, then their growth is very significantly impacted. So which means that they are not going to be very effective when we expose them to the actual wastewater where we have the mixture. Then we looked at the consortium of all these organisms, and we found that they were doing much better. Their biomass and their growth was much fast, even in the mixture. And then we confirmed, so the lower graph shows the pollutant removal efficacy. And if you look at it, the violet colored bar is the activity of consortium for removal of any pollutant, whether it is a heavy metal or it's a mixture of heavy metals. And you will see that this violet bar is always the highest one. So it proves that the consortium can do much better than a single organism. So with this learning, we wanted to go forward and make a product where we can create a product and we can give it to the people that you can use this product in the field if you are having a mixture of contaminants. So this I have already told you. And of course, as a PhD research, we have to also investigate the mechanism. So this particular picture is just showing that the partners were doing their respective function very well. So the Lentulus was actually removing the maximum copper, and the Aspergillus terrace was removing maximum chromium. So with this mechanism, our next target was to make a product. And earlier we had produced a myco tablet, which is a tablet-like product, and which is patented now, which can be dosed into the wastewater, and it will help in the removal of metals and also other organic pollutants. So when we tried to make this consortium into a tablet, we failed. And we were thinking that what is the reason why this tablet is not so successful, why its shelf life is less, very small, it cannot be competitive. And then we realized that it is something that when they are together in the tablet, then actually they are, fighting with each other. So being together is a strategy in stress. They can work together when they are exposed to multiple metals, but in peace, when we want to formulate them, they are not working. So we declared that this is not suitable. And then we went for another product, which we call as micro capsule. And in the capsule, basically you can, partition different chambers. So we partitioned the chambers and we could now put 2 or 3 organisms within this capsule, and this becomes a customized package. So we could also use colored coded microcapsules for different applications, and this patent has been recently granted. So with this, we were happy that yes, now we have a product and now we can take it to the actual application. So this innovation was the one that Madam talked about. It got this India Innovation Growth Program Award from DST Lockheed Martin. Now, while we are working with the microbes and optimizing their consortium, we also felt that as a group, we should be multidisciplinary. So we should learn from the nature, and we were looking for people with complementary expertise so that we can divide our work and work with a common interest and goal so that we have a value addition. And it was at this time that the 3 partners came together. From IIT Delhi, my group, Professor Khare, who is now our Dean of Students, Professor Sri Krishnan, he is well known for bioreactor designs. And IIT Delhi worked on assessment of heavy metals at different sites across the Yamuna, estimation of metals and designing the suitable bioreactor, which can be used on site. The Delhi University was the lead here, and they worked on assessment of pesticide and getting the microbes efficient for pesticide removal, studying their mechanism, selecting the most efficient. And from IARI Water Technology Center, Dr. Neelam Patel, who took the responsibility of heavy metal assessment in soil and crops irrigated by the drain waters. So basically, we designed the reactor, we implemented it on the field, and we tested compared the impact on soil, crop, and food using treated and untreated water. It was funded from National Fund ICAR, and it was a 4-year wonderful activity that we did together. So basically, I dedicate this content to not only me, but my team, including our students and my partners from Delhi University and IARI. So just sharing with you the glimpses, it was a large work which led to many PhD, starting with the water assessment in the water. So this is the gist of the hard work that we did for more than 2 years. Sampling of Yamuna when it enters the Delhi NCR and when it leaves Delhi NCR, and also selecting the most polluted drain in Delhi, which is the Najafgarh drain. And for IIT Delhi, we were interested with monitoring 6 heavy metals: copper, cadmium, nickel, zinc, lead, and chromium. So the red colored ones that you see here are the heavy metals which are more than the FAO permissible limit for irrigation. So if you look at it, you will see that almost every site has contamination, right? And there are some sites which have multiple contamination, especially as soon as this Najafgarh drain joins the Yamuna river. After that, you see that the contamination has increased. And the Najafgarh drain is really polluted. Punjabi Bagh location here is the most contaminated site, where you have all the 5 or 6 metals present, more than permissible limits. And around these areas, there is a lot of vegetable cultivation, and these vegetables are sold in the market. So this is the summary of the previous experience and the highlights of the higher concentration. But since we wanted to also provide a solution and implement a bioreactor, so we could not do it on Yamuna or Najafgarh drain because people will take everything from there. So we selected another drain, which is called Lohamandi drain. And fortunately, this drain passes through the IRI Pusa campus. So we could install our reactor in that IRI campus. And this Lohamandi drain, as the name suggests, also is polluted. So then in the peri-urban NCR, the soil status that Dr. Neelam Patel's group worked out, and they looked at different depths. Fortunately, at that point of time, the contaminant level was below the permissible EU limits. We don't get the limits also at one place. Actually, in India, even we don't have the limits for the heavy metal content in irrigation water. So we are looking at the international limits. So these are some other examples. So nice to see that it was below permissible limits, and we were expecting a food which will be free of contamination. But looking at the crop, this was the list of crops selected, and the student worked jointly with the IIT Delhi and IARI for his PhD. And then he saw that this is again a very registered data that around 22 different vegetables had nickel and lead more than the permissible limits. So it was constantly present in all the vegetables more than permissible limits for consumption. And then there were some specific observation that zinc is higher in tomato, chromium is more than permissible in spinach, copper in bitter gourd, like that. So even if the soils were not so contaminated, the vegetables were accumulating. So this is something that Honorable Vice Chancellor was telling in the morning, that the contamination is there. And then the calculation of the hazard index shows that it is more than 1, and hence the vegetables are not suitable for consumption. So then we were looking for our solution stream, and as I told you, we selected this Loha Mandi drain, and near to Loha Mandi drain we had the IRL fields where we installed the irrigation mechanism using this rainwater. And this is the profile of the Loha Mandi drain water, and these are the 6 metals. So the red dotted line shows you the permissible limits. And on the x-axis, you have period from July 12 to February 14, around 2 years of monitoring. And from this, you can make out that copper, nickel, chromium exceeded at several places. Cadmium exceeded at each and every place because the permissible limit for cadmium is very low. Zinc and lead were fine because their permissible limits are quite high. So at least these 4 heavy metals were of concern. Looking at this background, our future research was targeted on testing the impact of irrigation with contaminated water on soil and crop, develop the suitable consortium for remediation, first test in the laboratory, then take it to the on-site reactors, scale up the suitable technology, compare the quality of inlet and outlet water, and study how the treated water is impacting the soil and produce. So this was a close-knit program between IARI, IIT Delhi, and Delhi University. So we, being the environmental microbiologist, we started selecting some organisms. And we began with Beauveria bassiana because all of us know that it is an entomopathogenic fungus. So it is also used for pest control. And we wanted if we can use metal remediation as well as for insect control. And our target was multi-metal removal. So yeah, I'll skip this, something that I told you. So the laboratory experiments were typical. We went ahead with finding out the minimum inhibitory concentrations, how the metal uptake is impacted by different factors, how it is performing when the metal contamination is becoming more and more complex, what is the mechanism of heavy metal removal, and how it performs in lab scale reactors. So this is the key result for the minimum inhibitory concentration. And if you look at the data, this ranges from 100 to 250 mg per liter. It is not a very impressive performance because you will get strains which can tolerate up to 500 mg per liter. But the contamination is so low in the drain, we don't need someone who work at very high concentration. So we were happy with this. And given that it can tolerate multiple metals, we were removal these strains can do. And you can see that the removals, they range from around 60 to 75%. The most interesting observation was when we had a mix of 6 metals, the metal removal was around 83%. So this was a very nice observation, and we went ahead. We did, long-term experiments, the kinetics experiment, how is metal removal related to the growth, and so on. Then for the multi-metals, a lot of experimentation was done, and we were satisfied with the performance. Then we also looked at why the removal of different metals is different, and we could relate it to the chemistry of the metals. It is related to the ionic radius and the water of hydration and so on. Then we looked at what happens as we increase the complexity of metal gradually. So because lead is present in most of the location, so we began with that and gradually added more heavy metals into it. And then we did extensive experimentation, and this figure shows the gist. And this here we see the residual heavy metal concentration when it is exposed to different combinations of heavy metals. And we found that introduction of zinc decreases the removal of other metal ions because there is a competition of the sites, and addition of the nickel increases the removal of chromium. So there were many complex observations which we later published in Journal of Hazardous Materials. But the good thing was that in the 6 multi-metal mixture, we were getting very good removal of all the metals, right? I think only zinc and nickel were problematic. Otherwise, you can see that the blue bars were almost not visible. So the performance was good in the lab scale. We calculated the specific metal uptake and metal bioaccumulation, and all those data was generated. Then, because it was a part of the work of a PhD student, we had to go for the mechanism and tell why it is happening like that. So we used several tools like SCM, AFM, EDX, TEM, HA, HAADF that allows to look at the metal within the cell with different color codes. So we found that this is the control, and in the control, the organism is very healthy, the hyphae are very long, the cell wall is intact, and the cell surface roughness is quite good. But as we expose them to the metal, so this is the example of cadmium, and here we found that when exposed to cadmium, the hyphae become short, tightly packed, and you can see the red color in this TEM, which shows that the cadmium is well distributed inside the cell. So that is how it is imparting the toxicity and reducing the growth. And the cells are accumulating the cadmium very well, and there is an increase in cell surface roughness. So based on similar observations with different metals, we could come out with the fact that the cell is very smart. Some of the metals it is distributing across the cell. Some of the metals are partitioned in the membrane of the cell. And some very toxic metals, where do they go? They go to the vacuole of the cell, so that they are not imparting the productivity. So in this way, we could understand how Beauveria bassiana is able to deal with multiple metal stress in the laboratory conditions. But then we had to take it to the ground, and we started working on certain lab scale bioreactors, and we saw how this is performing in the bioreactor. So the gist of the result was that it succeeded. We had to pump the air because fungus is an aerobic organism, but in presence of certain metals, the media turned reddish pink in color. And this was something a bit troublesome because color is not accepted in the treated water. So we were a little bit not very happy with this observation. But as a result of this extensive investigation, at least 2 things we could prepare, that based on the metal contamination in the environment, you can select specific strains or their combinations, right? So only if lead is present, all these 5 can work. If there is extensive contamination, these 2 are the ones which are going to be the best, available organisms. Then we went on to look at how does it work for the pesticide. So our Delhi University partner told us that the strain is quite good. For lindane removal, they selected lindane because they observed lindane buildup in the Yamuna waters. So at the end, we know that yes, it can remove both metals and lindane in the reactor level studies in the lab. So with this, we went ahead. But when we took it to the IRI fields, what happened is we could understand some of the limitation. That in the actual field, the Beauveria bassiana was being washed away and this reactor was being enriched by the bacteria. So the fungal strains are unable to compete with the bacteria. So they will not be able to survive in the actual field environment. Then the wastewater characterization indicated that heavy metal concentration is there, but it is lower. So in that lower area is surviving better than this. So learning from this experience, we changed our way, and we decided to do the performance evaluation of native consortium for multi-metal removal. And we selected 2 types of reactor designs and operations, which is one is aerobic, where we use air. Another was anaerobic, which is known for metal precipitation. So we started with lab scale bioreactor configurations. We brought the water directly from the Lohamandi drain. So this is what I want to tell everyone, that if we have to eventually go to the field, it's better that we start with the samples from the field. So we spiked the Lohamandi drain, we kept it as such. We spiked in one set with heavy metals because sometimes the industries are releasing the metals and there is a metal shock that comes. So with this, we did study for about 6 months and the organism in the reactor got enriched. And again, this is a gist of around 6 months of reactor operations. So you can see the aerobic reactor performed very well, and around 4 to 5 days, the metal concentrations came down to very low. The anaerobic reactor, which is on the lower graph, did even better, and you can see a fast decline in the metal concentration as the water entered the reactor, and then it was sustained for a long period of time. So now it was the time for us to decide whether we want to take an aerobic technology forward or we want to install an anaerobic reactor. So, but then it's not always the good performance. We are looking for several other things. So if you smell the anaerobically treated water, it smells because, some of such elements are formed like H2S, which can provide the smell. And we feared that it will not be accepted by the farmers. So we decided to work with the aerobic technology, although it was not as good as anaerobic one. So this was our field location, and as I told you, the Lohamandi drain passing through and IRI campus and the experimental fields close to it. And this work was led by Dr. Neelam Patel. So we selected ladyfinger, potato, wattle gourd, and spinach, and they provided the data on impact of irrigation using untreated water under drip irrigation and flood irrigation. So this was the kind of field structure that they prepared. These are some of the harvest. So this data was important for us, and it was showing that this buildup of different metals in soil after irrigation for about 1 year. And the red color codes here are the ones which are above the permissible limits. Permissible limits are given in the lowermost row. So you can see that nickel, lead, and zinc accumulated, whether it was flood irrigation or whether it was drip irrigation, in most of the fields. So this is what is the gist. So metals were accumulating. This is after the second year that, and we found that there is a concentration buildup. The concentrations are increasing. So this is then the next third year of, I think, activity, where also we saw the buildup. So looking at this consistent performance, then we decided to put our bioreactor, and our overall conclusion was that the drip irrigated vegetables have less accumulation of metals as compared to the ones which are done in the flood irrigation. So now we began with our bioreactor installation, and some of the part of the field area was selected for the bioreactor installation. And we started again with the aerobic reactor for the reason I just told you. And we absolutely worked in the field condition without any control. So we enriched the microbial consortium, began with a 5-liter constantly stirred reactor, and then we scaled it up to 50 liter, and in the third stage to 500 liter per day capacity, so that the treated water can be used for the field experiments. So these are some of the pictures again. This was the shed that we created for the bioreactor installation. And this is the gist of the data that comes from the reactor operation. And you can see that we dosed around 5 mg per liter of heavy metal for each of these metals in the reactor, which was running at the field site. And you can see that this is the time of operation. We operated it around 420 days. And quickly, the heavy metal concentration came down to somewhere below 1 mg per liter. So which means that the organisms were very well adapted. And if we look at this table, they were doing the removals from 96 to 98%. So this was a very favorable observation. And we moved ahead. We looked at some other parameters. How is the COD removal happening? And the other graph here is showing you how is the growth of the organism happening, right? So we see that the bacteria is growing very well, right? And then we took it to the 50 liter and 500 liter. These are some of the results from the scale-up. And here you see the removals of around 57 to 58%. We were constantly aware of the criticism that was going to come, is that where the metal is going, right? Because it is not degradable. So we were monitoring the sludge, and the sludge concentrations of these heavy metals during our experimental work for about 1 year were well below the EPA standard, that is the Environmental Protection Agency of US. So we could say that the sludge which is being generated in the bioreactor is non-toxic, and it will not pose any secondary problem in the environment. So now this is the data that shows you the metal accumulation or presence of metal in treated water. And you can see that none of these is actually red, which means that the metal accumulations are not there, which are hazardous. So below limits. Then this is again irrigation with treated water for the second year. Here also we see in the flood and drip irrigation model, the metal concentrations are below limit. So eventually I can conclude that, working to a 500 liter per day scale, and in the totally outside environment without any control, we could achieve 40 to 90% removal of heavy metals, and all heavy metals in the treated water were below the FAO permissible limits. And this is how the reactor looked like. We did not design any fancy bioreactor. It was all the low-cost material for the reactor body as well as the settler. So the cost of the system was not also too large. And eventually, we can say that where the fields were irrigated with untreated water, nickel, lead, and zinc accumulated in the vegetables. And where we irrigated with the treated water, all the multimetals were below the permissible limits. So this project got over in 2016, but I'm happy that the reactor are still there and they are a part of another series of funding from National Fund where they are trying to club this reactor outlet with a wetland system and do some further investigations on this. So I'm very happy to, just showcase our students who are actually the drivers, both of them who did their PhD and the two other students whose PhD was related. And this work was also awarded by ICAR and of course my partners. So I really feel that working together can make this solution to the complex problems very easy. So before I end, I will just touch upon a few slides that the current work in our lab is going on, and which is algae-mediated wastewater treatment and biofuel production. And here I am very fortunate to get a funding from MNRE, DST, and various international collaborations. And this is just a glimpse from our Indo-Brazilian collaboration that recently concluded. And it's a very heavy slide, friends. But here we use algae for treatment of wastewater. The wastewater comes from different sectors, rural sector, and agricultural waste, and dairy sector, slaughterhouse. And we design different in-house reactor systems for algal growth and wastewater treatment. And with the Brazilian thing, the Indian responsibility was again to check for heavy metal. And in this case, the algae also accumulates the heavy metal. And then we take the biomass to a conversion technique, which is called hydrothermal liquefaction. I'm sure Professor Pant will talk about it. He's collaborated my co-PI in this project. And the wonderful thing it does is that it actually partitions the biomass into an aqueous fraction, which is safe, which has no metal, which can be used for isolation of pharmaceutically relevant compounds. One of my PhD students who is shared with Professor Pant has found some very exciting in the section. Then we get a biocrude, which is quite important. Around 50% of the algal biomass is converted to this biocrude, which can be refined for energy and fuel or platform chemicals. And the most interesting part is that the solid fraction, which we also call as biochar, it gets all the heavy metals. So around 70 to 80% of the heavy metals get partitioned in the char. And now it is very easy to safely use this char for some application like construction or for the brick formation, so that you don't have the problem of heavy metals coming back again into the environment very soon. On the other hand, we also demonstrated how the algae from after wastewater treatment can be used for biogas generation at a pilot scale. So the year-round production of biogas can be obtained. So, and lastly, we are working now on Barapula drain, which is a very big drain in Delhi. And this is with the Dutch collaboration, and we work on the photobioreactor part, and we are trying to convert the algal biomass, which is harvested, into microalgae fertilizer. So this is how the bioreactor looks like. This is a vertical photobioreactor. It treats the wastewater, removes the heavy metals, nitrogen, phosphorus, and other pollutants, and the biomass which comes out. Still, we have to work on how we can produce the biofertilizer out of it. And the treated water is of the irrigational quality. So we have worked for around a year with this photobioreactor on site. And this is how our photobioreactor looks like. And the best thing, or the USP, we say, the unique selling points of this technology is that it has low hydraulic retention time. It treats water in 2 days, while other algal reactors take around 6 to 8 days. The footprint is very low. As you can see, it's a vertical structure. and it gives us both contaminant and pathogen removal. So we are very excited about this work. This is our Dutch collaborators working on it, and this has attracted a lot of visitors because it is on site at the Barapulla drain site. And in the future, soon we are going to scale it up to around 5 meter cube per day plant and going to harvest valuable products out of it. So I think with this, I will end. This is our journey for removal of contaminants and treatment of the water for the irrigation quality. I will be very happy to receive your questions, your inquiries on the same. Thank you.
One or two questions. Two questions.
That was a nice presentation. You have immediately explained different techniques like this clubbing of microorganisms. I have query. Whenever you are switching to club the microorganisms like bacteria, algae, and bacteria in a particular platform, what will be the effective behavior of these microorganisms? Because it is crucial that we are putting a microbe with another microbe, maybe they will suppress their growth, maybe they enhance. Cumulatively, they will come. How to analyze this behavior of the organism?
Right. So there are two approaches for this. This is a very interesting question. One is the typical laboratory approach where we take, say, 4 organisms and we know that we are going to mix them. So we study their interaction well before in solid media plates or in the aqueous form also. And we are typically using some media for that, right? And we express our results as positive interaction, negative interaction, impact on their growth, what is the inhibition radius and all. So that is a very typical approach to do that. And we do that also. But actually it doesn't work in the field because in the field the conditions are totally different. So in fact, with our experience, what we find is that it's better to allow the natural communities to enrich and then look at them, because whatever artificially we are trying to do, eventually after some time the nature will take over. So right now we are at this second approach that we allow the communities to interact with the wastewater. We are not controlling the microbial input, nor are we controlling the environmental conditions. So the reactor stabilizes after, say, 1 or 2 months of operation. And then as a scientist, because the funding agencies will always ask, what is the organism? If you do not know, how do you prove your results? We know that it has no meaning, but still then we do the metagenomics and then say that these are the communities over a given period of time. The third interesting approach that we are planning in the future, I do not know how practically implementable it is going to be, is the use of 3D printing that people are doing in the developing countries, where we can print the communities of different architecture and design. So, but I cannot comment on that actually pertaining to how they will work in the natural environment. So it's the time that, time test is the best thing to look at whether the communities are going to fail or pass. Yeah.
Deep learning can be beneficial for the future aspect. Like you are putting different genes from different organisms in a particular organism, it will benefit for the society.
Yeah. You are talking about the genetic engineering. Okay. So basically, like at IIT Delhi, my center, specifically Center for Rural Development and Technology, and our mindset is against genetic engineering actually, the faculty group. So they actually want us to pursue nature-based technologies. So we have not yet attempted any genetic engineering. My colleagues in biochemical engineering and biological sciences do that, but somehow our environment does not allow that to do practically.
Not accepting this type of genetic.
Yeah, right.
Myself, this talk well, and particularly at present scenario, if we need biodegradation
in present situation and in this context, especially in NCR region. Let me talk about the microtablets and microcapsules. What is the significance of microtablets and microcapsules in pollutant assessment?
Assessment. Okay, it is not for the assessment.
What are these?
Yeah, these are the interventions for remediation, for treating. You know, like this is a product which has these cultures at one place, and based on the, so first you do the assessment. For assessment, either you go through the traditional laboratory based approach or you use biosensors, whatever it is. Now you know that these are the main pollutants. Accordingly, you can select the product out of these microcapsules, that this product is good for a mixture of chromium, copper and lead. So you dose that, and then it multiplies and removes, accumulates that contamination. So it's a live product. It's a live product basically. Yeah.
Talk about the value of bacteria.
Yeah.
This is the old culture. When you have isolated level.
Actually, yeah, we worked on actually my student who were from this university itself. They worked on a totally different topic on control of houseflies in the village environment. So for that, we started working on Beauveria bassiana, and they isolated it from somewhere, some soils, and we did one thesis on making the products for housefly control. Huh?
Will you get the pure culture?
Okay, yeah. Actually, this student finished his PhD, so I will inquire if it is available, surely. Yeah. Any other questions?
Any other? If not, thank you, ma'am. You have very rightly highlighted the condition of pollutants in urban and peri-urban areas. And it was pertinent that almost everything what we are is pollutants, heavy metals and contaminants. We have also highlighted that only 32% of the water is treated and 68% of the wastewater that is still being used for the irrigation and other purposes in the fields. And, we have also highlighted this consortium approach and the cocktail approach of the microorganisms to control these pollutants as a biomedicinal is. And you have developed capsules and tablets for that purpose, and you have successfully demonstrated that in bioreactors they worked, and the level of pollutants that have come down to the permissible limits. I also congratulate you for getting this ICA award for this specific innovation you have done, and we hope that these innovations, maybe bacterial, algal, or Bassiana, they will scale up and we will be able to get less polluted or free of pollution vegetables and fruits in the future. Thank you very much, ma'am, for enlightening us with your work, and we are highly thankful to you for joining us today. Traveling right from Delhi, I was told that you are likely to return just now after this talk. Thank you very much.
Thank you, and I am looking forward to more collaborations with your institute. Thank you.
Thanks, ma'am, for the nice presentation. It was looked that it ignited a spark among all the researchers here. And we are very hopeful because when ma'am told that the situation is alarming and many vegetables are not safe for consumption. But the positive thing is that the successful implementations of aerobic and anaerobic bioreactors and the formation of consortium offers solutions to all these problems. So this is the main aim of this workshop, to discuss among ourselves and get solutions for these problems for conservation, for sustainable environment. And now I would like I request Dr. Rana to offer a memento to ma'am for her enlightening speech. I request Dr. Nilesh to give certificate for this. I would like to ask the co-chairman for the next presentation, that is by Dr. Manoj Tripathi, Principal Scientist from ICR Central Institute of Agricultural Engineering, Bhopal. Dr. Manoj Tripathi is has done his post-graduation in biochemistry from Dr. RNLA University and obtained his PhD in biochemistry from G.B. Pant University of Agriculture and Technology, Pantnagar. He is currently working as a principal scientist at APPD ICAR CAIE, Bhopal. Dr. Tripathi's major areas of professional interest include medicinal compounds, functional and nutraceutical foods, fermented and probiotic food. He has guided more than 50 PG students till now and published more than 85 research papers in journals of national and international repute, 93 abstracts, 54 book chapters, 55 popular articles, and edited 13 books and many technical bulletins. Sir has also developed many processes and technologies and has worked on national projects as principal investigator and co-principal investigator. He has been recipient of several prestigious awards and honors and has qualified the prestigious fellowship. He has also been recipient of the Established Scientist Award in 2014, Best Scientist Award, Young Scientist Award, and Best Presentation Award. In 2017, Dr. Manoj Tripathi received Lifetime Achievement Award for innovative research and for outstanding achievement in the field of food processing and publication. In 2012, he received Center for Excellence for certificate of recognition from Central Silk Board, Dehradun. He also received Best Scientific Contributor. Award as executive member of Scientific Panel FSSAI. Executive member of BIS. He is member of many committees as executive member and editor-in-chief of Krishi Abhiyantriki Darpan. He has served as chairman, convener, organizing secretary, and more than 123 international and national symposiums. And he has also established many research labs in food technology and biotechnology. Now I will like Dr. Sirohi to chair this session and for the presentation by Dr. Tripathi.
Okay, welcome to this brainstorming session. And the audience are eager to listen to you on another aspect of food waste. What are the causes, what are the control measures for this food waste? How we can save food wastage. I welcome the organizers and request you to kindly interview.
Thank you, sir. And I am Advul. Sir, my voice is clear?
Can you hear me?
Yeah, my PPT is, sir, here. I shared my PPT. Sir,
Yes, sir, it is visible, sir. Visible and it is audible very much, sir.
Okay, sir. So can I start? And good evening, good morning, and everyone, and respected all the dignitaries, Honorable Vice Chancellor sir, Professor Anil Sirohi sir, Professor Ravind Kumar sir, Professor Pankaj Kumar sir, and Professor Rekha Dixit and Dr. Malik madam. And thanks for providing these opportunities and also for a nice introduction. Actually, see my topic and brainstorming topic. Basically, I prepare my topic to the coming biotechnology students and try to interact all the faculty members and they make a huge utilization of this by food wastage. If you see the considering that the million
Yes,
people,
yes, you are audible.
millions of people around the world go hungry. And
Yes, yes, it is visible.
it's shocking that the many of us throw away food on the daily basis, which is required the. In fact, 1/3 of the all produced globally goes to the West each year, and amounting of 1.3 billion tons of wasted food, and with the value exceeding more than $1 trillion dollars. So, if the see the according to the some recently published data by the FAO and of the United Nations, the global hunger could be alleviated if just 25% of the food wasted each year was saved in a state. So you can imagine how much we are wasting our foods and our economic and which can be easily converted to the value-added product. So this topic is basically cause, effect, utilization, and management of food wastage.
Hello, Dr. Manoj. Dr. Manoj.
Sir, hello.
Dr. Manoj, it is.
Sir, voice problem is there, but I listen your word, sir. Hello. Hello. I am audible.
Yes, sir, you are audible, sir.
Yeah. So can I continue? So
Yes, sir.
If you see this scenario, why these problems come? Because of the some good things and some bad things. And if you see, India is the production segment. We are either in first, second, and third. We are very good climatic regions, largest irrigated land, largest net irrigated area, cultivable compared to the other countries. We also largest livestock populations, largest milk producers, second largest producer of sugarcane, second largest producer of cereal. Instead of having the largest one, first, second, third position, but we are not properly arranging our whole production. We are not properly utilizing this production. So either we are producing huge amount, but we are also producing a huge amount of food wastage. If you see, this all create a significant opportunities to the food sector. But due to the huge amount of food wastage which goes to the environment and due to their biochemicals and nutritional composition, they are just inviting to the microorganisms and finally contaminate the environment. So this huge amount of production also required a proper process technology for utilization of the waste generated in different sectors. So if you see, these are the causes of food wastage. Because we have not a proper consumption styles, we are excess producing without managing the excess this commodity. We have lack of communications and cooperation between the farmers, which is the suitable, which will be more stable at that commodity, at that farm level, and also the premature harvesting and retail side. These are some regions which causes the huge amount and production of huge losses of this food wastage. If you see, these are the food wastage by the category. We are losing our different foods in different sector. The fruit, 12% of the wastage from the food grain, 7%, added fats and oils, 8%, tree nuts, peanuts, eggs, and meat and vegetable. If you see, everywhere the wastage is high. In somewhere wastage is there, but there is some technology for the conversion into their value-added product. But in some cases, like vegetables, the wastage is 19% because we are not properly utilizing our process, and technology is not much more available and suited for the waste management and byproduct utilization of these vegetables. So this is the important reason of the food waste creation. If you see this is the social, this food waste generated different level. Here we see this this wastage start from the pre-harvest, pro-harvest level, processing level, transport level, retail level, and finally consumption level. Means in any level, if there is a wastage, definitely this is the loss of food available for the consumption. Whatever we losses, that will be the loss for the population for their nutrition. Finally, all these losses is basically causes more pollutions, more environmental impact, more economic impact. In fact, if you see when they goes to the environment in the soil, it causes several types of problems like deforestation, land occupation, air pollution, soil degradation, climate changes, water pollutions, biodiversity. Means they are basically the reasons for not only economy, not only the nutrition, but they are the social and environmental cost of food that are the associated with these losses, and finally they added to the caste of the society and the degrade the economic of the society. So their utilization is very important. If you see, this is the global scenario, 28% of the land area is food waste. You can imagine how much wastage is there. Globally, it has been estimated that this wasted food couples almost 1.4 billion. These numbers may be higher, roughly 28% of the world's total land area. If the food that is grown dead but not eaten will become its own country, means in one day it may be the own country of only by accumulating this wastage. So it is very important and to just utilization and avoid the generation of this food wastage. So if you see this slides, this is the per person food wasted in per kg of year. This is huge losses. And this is the not case of our country. This is also case of the most of the developed country like Australia, United States, Turkey. Everyone is just lodging the food wastage and dumping this food wastage and a good amount of nutrition into the environment. That will be the we are just approaching for the nutritional as well as the environmental problem. So you can see how much food wastage are just dumping in the environment. If you see on percent of the food which goes to the globally, overall 13.8% of our food in globally goes to the simply dustbin. And these are the some commodities like root, tubers and oil bearing crops, 25%, fruit and vegetables in the form of this commodity, 21%, meat and animal product in the form of 11%, and cereal and pulses. I mean, all the commodity, the wastage is high. In some cases, the wastage is more due to some process and technology availability. So if you see, this is the amount of food wastage per country, means each and every country in different steps. In some cases, the production losses is high. In some cases, post-harvest handling losses is high. In some cases, at the stage of processing and packaging, the losses is high. In some cases, the distribution retail is high. And finally, in some cases, the consumers losses is high. Means each and every country just wasting the food wastage in different formats. If you see in the scenario of India, the food that goes to dustbin in our countries are left unused. That could be the only meal of the day for about 194.6 million people of the people. You can imagine how much this food wastage we are losing. India wastes 44,000 crore. That value is maybe increases. Worth of the food recovery per year. Roughly 1/3 of the food produced in the world for the human consumption. Every year, approximately 1.3 billion tons get wasted. So while on the other side, means 1 in 8 people facing starvation, and that starvation problem can be solved by the minimization of this food wastage. So if you see, this is the actual scenario, and there are different steps where we are using our food wastage, starting from the production, agriculture are harvested, goes to the harvested commodity, and here there is some minimal processing and food processing is there for their value additions or the shelf life stability or packaging, and finally goes to the marketable products, and finally here again some processing and make some processed product. If you see at this level, when agriculture produce goes to the harvested commodity, we lost this food. Our agriculture produce in the form of twigs, leaves, straw, parts of the plant, waste commodity, and is suitable for the market, food market. After sorting here, if we goes to the harvested commodity to market products, there are huge losses in the form of fruit stone, fruit pulp, fruit skin, cereal bran, cereal skin, cereal husk, vegetable waste. These are the some examples. When you go to the marketable product, to the advanced or value added products for different marketing purpose and convert to the food, processed food, these losses is there in the form of fruit promises, seeds, spent grain powder, vegetable waste, and several post processing wastage are there. So huge amount of wastage are generated at different levels. If you see, this slide show a very important figures. What are our different industrial wastage farms? If you see, there are two farms. agriculture residues and industrial residues. If you see the agriculture residue, that can be again further either in the field residue or the process residue. So field residue and process residue are the major wastage of the foods. And in case of industrial wastage, a huge amount of organic residue and related effluents, food comes from the foods processing and other development are produced everywhere. And this food processing industry like juice preparation, chips, meats, confectionery and food industry, they are producing a huge percentage of the food wastage. And that are basically due to the unavailability of the proper process and technology, we are not able to utilize in the either food application or energy application. So these are the very important problems. If you see in fruit industrial wastage, industrial, they have, if they see the nutritional composition of this food wastage, it is very good, high in the protein. Some in cases very good source of oligosaccharides. In some cases they are very good source of different fibers and different molecule, which can be, if utilized, which can be very good source for the production of microbial or others value-added products. So if this agro-industrial waste and type is not properly managed, it basically they're responsible for the change in the BOD and COD and also causing several pollutants. So these are the some examples. So if you see, these are the waste of the most food generated. Residential, we are producing 44%. In the restaurant, we are producing 33% of food wastage. In grocery store, we are producing 11%. In institutional, 10% and industrial 2%, because here they are basically processing and they are just to try to utilize in some value-added product. So this is the scenario. If you see, these are the percent of food wastage in different commodity like cereals, roots, oil, seed, fruits. Means not only one steps of the is responsible for food wastage, but in the consumption steps, in distribution stage, in processing stage, in post-harvest stage, in primary production stage, most of the commodities are producing huge amount. In some cases, the primary production stage causes huge waste production. In some cases, post-harvest stage causes huge production. Means each and every these steps of the from your ground to the industry are causing huge amount of percent of food wastage. And these, if you see, these are the wastage at different level, like in the field and harvest, 20% average losses. With the several reasons due to the inadequate nutritional regime and poor weathering, some infections, some in case of tomato, if you see, it's most of the spotted of the soil. So these are the reasons at the farm levels. In the farm storage, this is again one of the reason why huge amount of wastage is generated, because of the some rot and molds we are not uncontrollable at the farm level. No cold storage is there. Rodents problems, unbalanced nutrition and breasting, means these are the some reasons to causing the food wastage at processing level. This is again one of the important level where due to the some sugar content, due to some other biochemical component, specific color, size, shape, the industry person or the market person not taking the farmers material. So these are the reason to the loss during the processing and market requirement. Transport is again one of the reason of the losses of several things, and at markets this is again one of the reason for losses of huge amount of food wastage. And due to the several poor stock management, unbalanced nutrition, no refrigeration system in the market, so huge amount of our food due to the microbial deteriorated and losses there. So if you see the food industry and byproducts, we are working on this aspect and we estimated, and some of the common food industry like white wine production, just 20 to 30% wastage are generated. In red wine production, 20 to 30% wastage are generated. In case of fruit and vegetable juice production, simply a huge amount of 30 to 50% wastage are generated. In case of fruit vegetable processing and preservation, 5 to 30% depend upon the commodity, this wastage and byproduct generated. Vegetable oil production, sugar production from sugar, means if you see the some common food industry, they are just generating a huge amount of byproduct or wastage that are not value, high concentration of organic compound in there, high water activity and poor oxidative stability, and optimum enzymatic activity and proper waste management. If you see the proper waste management and utilization, they plays a very vital role in the growth of our food industry. And most of the our small farmers, large farmers, entrepreneur, and industry person, there will be surely helpful for the enhancing their economy. So if you see, these are the some possible human hazards exposure that comes from the byproduct, which is already discussed, Malik madam. And these are the some possible hazards like metals and metallites. And in first lectures are also discussed. In these are the some byproducts comes possible hazards comes through environments or byproducts. These are waste deterioration in our foods like lead, cadmium, arsenic, nickel, mercury. Mycotoxins are also very problems which when the fruits or vegetable or commodity slightly got the moisture content, humidity, these types of mycotoxins creating fungus is immediately grown. So these are again problems like aflatoxin, oclotoxin. So these are the some common possible contaminants may comes from these byproducts, some antimicrobials and some pesticides. So these different types of compound which causing and the food contaminants which is seriously addressed currently because they directly goes to the food chain, and that food chain definitely just causes the hazards to the environment and finally our human health. So we just estimated and see there some common community like apple. We are working on this community, apple, banana, citrus. If you see a huge percent of wastage is there. Means in case of banana, 35%, in citrus, 50%, and in dragon fruits, 30 to 45%, in peas, in the farm of their cell, 40%, in pineapple, 33%, potato, 15%, tomato, 20%. And these all byproduct and wastage are a very good source of protein, lipids, carbohydrate, and fiber. Means we are just minimizing, we are just dumping our nutrition to the environment and also inviting several pathogenic microbes because of this nutritional composition. So if you see, we have just also work on several byproducts and see the total micronutrient, micro elements are very high in most of the byproducts. If you see, these are the byproducts: orange peels, watermelons, honeydews, and banana peels. And but these are the some common byproducts which is on daily basis going to the environment. And you can see there a huge amount of micro elements are there, and that micro elements, if you are not utilizing, they are just using in the waste, they are just dumping to the environment. So if you see there, the concentration of total trace element and toxic element in this byproduct is also high. In case of micro elements in milli per gram, if you see, every byproducts have a good amount of the micronutrient and nutrient which can be utilized for the nutritional purpose and for the development of value-added compound. But due to some shortage, due to some informations in all the sector, fruit, vegetable, oil seed, and beverage, we are just using as a waste. If you see, these are the some total toxic element, which is mainly in some cases this toxic content is very high, but almost all the byproducts is having some toxic elements, some toxic microbes, and some toxic mycotoxin. If they are not properly utilized, they completely become the part of environment. So here the proper management and utilization for the health, environment, and economic purpose is very much required. We have also just justify of the do the some biochemical composition of some common fruit and vegetable byproduct. If you see from these byproducts, mango peel kernel, mango seed kernel, mango peel, pineapple peel, different, all have a very good amount of protein, good amount of fat, good amount of carbohydrate. In these carbohydrates, some are very good source of oligosaccharide, and they can be converted in the prebiotics, or in the also become the one of the substrate for the microbes and human. So you can imagine how much nutrition we are just dumping in environment. And this is one of the byproducts. So soybean byproducts we are working, and these two byproducts are very common, and they are not utilizing due to the unavailability of proper processing technology, but they have a huge amount of a biological molecule, nutritional molecule, which can become the part of the value-added compound like this soya tofu, where this is the water discharges of the tofu or paneer preparation. There is huge amount of steachiophenol. This two molecule is now one of the burning substrate for the prebiotics or microbial or probiotic growth. So if you can utilize this byproduct, definitely they will be very helpful for the economic growth. So here we are working, and this is our approach, because if you see different types of agri-food wastage are there. If you see in fruit and vegetable wastage byproduct, the wastage are in the form of peels, pulp, and poaceous stalk, leaves, and these are very good source of natural biotic compounds. And also these natural biotic compound, by using several technological, it can be used for the development of several applications. Like we are just working on the drying technology, solid state fermentation technology, and some green extraction technology we're also using for the utilization, like ultrasound, microwave state, pressure liquid extraction, green solvent based. So finally, this technology will be helpful to conversion of this agri food product into several applications for the food. We are mainly concerned to the food application. So fruit and vegetable agri food wastage can be converted to the food supplement, and it can be the fortification of enhancing several nutritional value and oxidative stability. Also, they become the one of the noble functional ingredient for the functional food industry. And if you see the some underutilized seed and legumes, they are again source of unconventional oil-rich biotic tocopherol and specific fatty acids, and they can be again converted into some good oils source in food and cosmetic industry. Basically, the cosmetic industry and pharmaceutical industry are very much interested to the creation of this fruit and vegetable byproduct. If you see the dairy byproduct, we are just discussing on some common. The oligosaccharide with the prebiotic function are very important. Most of this dairy byproduct, way another, are very good source of oligosaccharide. And by using some simple development process technology like micro and nano preparation technology, fermentation technology, and some compositional analysis, we can convert to the prebiotics and anti-inflammatory supplement, which is very demanding in this COVID period for enhancing the growth of probiotics. So if you see the fish and meat industry waste byproduct, they are very good source for the protein hydrolysis, which have a huge amount of biotic peptides. And by using these common biotechnological steps, if you see, all are the very simple biotechnical steps, but their products are very potential for the human health, for the entrepreneurs, for industry, for all the sectors. So this is our approach. If you see why this is important, why the address of these food products and byproduct utilization is very important. If you see here, the agriculture production, in most of the production goes to the fresh foods. In some cases, it goes to the food industry. In case of food industry, they are just using this food component as a supplement, as a substrate for the production of jam, fruit, juice, nectar, and dehydrated. But during the production of these products, a huge amount of food wastage and byproducts generated. And these food waste and byproducts are basically dumped to the environment that causes several environmental pollution. So pollution boards are very sensitive. And if you utilize these pollution boards before the environmental pollution, there's some added them some operating cost, and finally your products become costly. So their proper utilization is very important. If you see this food waste and byproduct, their composition, they have some anti-nutritional compound. Due to this anti-nutritional compound, they are not directly able to the food purpose. So by using some food processing technology, we can just decrease or minimize the anti-nutritional compound factors in the some below range for the food purpose. They can directly use for the animal food, and finally goes to the also hungry persons. In other cases, if you see the composition, whatever we discussed earlier, they have a good source of protein, lipids, carbohydrate, minerals, antioxidant, fiber. If you use the proper processing technology, and they can be converted to food additive, and finally goes to the food products. And these food products are in a value-added products, and it become the part of our economy. So if you used this process applications food supply, plant derived food wastage is also there, and animal derived food wastage and the youth derived food wastage is based source. If you see several phytochemical source, several phenolic source for the food uses, and by using this incineration methods, we are also approaching to develop the hydrogels. And here we are using so many of the agro produce or agriculture wasted by pre-treatment and hydrolysis method. Just we are hydrolyzed, and by using solid state fermentation and by help of several microbial organisms, we can convert to the several useful product, biotechnological product like bioethanol, enzyme, biohydrogen, and bioplastic. Here, animal derived product, we are the persons are also working on the extracting and processing technology, and they are able to develop several collagens, chitosan, protein hydrolyzed, biotic peptide, cosmetic. If you see, these all are the basically come from the wastage. Means our wastage is not wastage if you utilize. They have a good amount of biologically active, chemically active compound for the enhancing the economics. So if you see the wastewater, we are also working on the wastewater, fruit and beverage, which we discussed, soybean products started. Here we are major concerned with the soybean products, means that the major byproduct and major soybean industry, they are producing a huge amount of waste water, and these wastewaters are having the either the product wastewater or washing wastewater. And finally, our purpose is to just convert different biological compound like protein, saccharides, carotenoids, dietary fiber, and pectin. So if you see, this is our waste hierarchy basically, and our waste hierarchy is include the avoid basically the production and other sources which causing excess is the most preferred approach for the waste hierarchy to manage the waste. Next is the reuse. If the products is excess product is there or byproduct generated, we try to reuse the some byproducts or the some things which is uses during the production or development. Third is the recycle. Definitely this approach is very important to the just recycles, and biotechnology is also playing very role to the conversion of to the animal feeds and some fertilizers and some other things. Reprocess, again this byproducts can be reprocessed to the several pharmaceuticals and nutraceutical sources, and they also convert to the cosmetic products. Energy recovery, definitely this is the energy application of these byproducts. And finally dispose. In some people's this dispose hierarchy is just adopted first, but this is the opposite. This is the last, and the first approach is to avoid. So if you see these various byproducts which we are working, they have the several potential. They can be, we can just utilize for the source of recovery of several iron, recovery of proteins, recovery of oligosaccharide, recovery of isoflavone, which is one of the very strong phytoestrogen, production of several cryoprotectant, concentration of whey, production of emulsifier by using some physical method. But when we go to the some advanced method and microbiological enzymatic methods, these byproducts are very good substrate for the peptide production of the probiotics, hydrogen production, novel ingredient from dairy foods, preparation of culture for the pathogen testing, citric acid production, some probiotic production, tofu coagulant. We are also developing coagulant, production of some useful fungal mycelial proteins. Means a huge option is there for their development. And these are the some biotransformation which we are working in our lab. We are developing this byproduct to the microbiological culture. This is very much related to the biotechnology student. We are just starting the PG student. So these are the the technology is not very advanced, but the process and the product is very novel. So we are developing some microbiological culture media using the byproduct and wastage, algal production, prebiotics culture production, fermentation media, novel ingredient for different dairy products. And we are also using for bacteriocin production, functional food beverage developing by using this byproduct, some coagulant which is used in the paneer preparation, and they faces several contamination and of the chemical residue contamination, prebiotic production. So these are the some sectors where we are working for developing this our byproduct and wastes to the evaluated product. So this is the, if you see, this is the our work where we are just try to develop soybean to our paneer. This is the process where we raw soybean can be converted to the soya milk, and soya milk can after coagulation to the tofu. This is the soya paneer. So in this process, huge amount of byproducts is generated. If you see, if you take 1 kg of soybean, you can make 7 liters of this soya milk and 1.5 kg of tofu. But here 1.5 kg of okara and 7 liters of a highly nutritious tofu is generated. So disposal of these two highly nutritious into environment is very problem. If we are discarded into environmental, huge amount of contamination and pathogens are created. If you just burn in the water blooming is caused. So utilization of such types of nutrient and food byproduct is very important. So we are utilizing this byproduct, and we are discussing some of the byproducts, some of the valuable product developed from our food wastage. We basically utilize this tofu. This is the water discharge after the soya paneer preparation, and we are just developing the culture media for the probiotic production. Initially we developed the liquid, but presently we have also developed the solid culture media using this tofu by help of several formulations. So here if you see, we just utilize this tofu and simply added some supplement for the culture media, like our tofu with the yeast extract, nitrogen sources, or the malt extract, or tryptone, or potato extract, and found that alone tofu this after small balancing of the pH, so a good growth of the probiotic culture, and they are eligible for the culture media. But when we added several supplements which provide some nitrogen source, they become one of the good sources. And this tofu, if you see, alone tofu is good nutrient, very good source of preventing and texturizing additive. We are also trying to develop some cosmetic and pharmaceutical. So if this tofu is used byproducts liquid nature, which is easily deteriorated, can be converted a good culture media for the probiotic production. And I'm happy to here declare that in our lab, most about the culture for the fermentations, for the culture for the microbial and fungal or pesticide production, we are using our own this tofu made, and we also claim apply the patent for this intervention. And this actually this tofu but nowadays in food industry, the most of the product is the fermented product. In fermented products, the fermentation media is very important, and in fermentation media, if you see their composition, some non-vegetarian component is also there. So we are trying to develop complete vegetarian component, which is the plant derived byproduct. And finally, we are using as a fermentation media for the probiotic culture, and these probiotic culture are using for the development of some innovative and novel products. That is, here we are using the fermented soya milk product, because of the facing some problems of this soya milk after the fermentations. These problems are just deteriorated, minimized. So this byproducts in the fermentation media is one of the good alternate which we are using. And we are also developing a probiotic cheese product. First time we will develop the soybean probiotic cheese product. Here the most of the component and most of the probiotic comes from our developed fermentation media, which is the tofu and fruit vegetable based. So if you see this byproduct, these products have some problem of the shelf life and some of the pH stability. So by using this byproduct fermentation or some supplement, this problem is minimized. So when we add some okara powder, this is the byproduct, they simply maintain the viability, their texture, their aroma property for the longer time. Finally, they just enhance their quality. So we have also published a paper on this aspect. So fermented soya milk powder, we also developing and the probiotic soya milk powder, where this fermentation technology, where these products, byproducts which develop in our laboratory is basically utilizing. If you see these soya milk powders, they contains all the good ingredient of soya milk. They are the high content of protein up to 49%. They are basically this byproduct. They are good source of dietary and naturally occurring amino acids, a very good source of micronutrient, mineral and vitamin. Means the product which is produced after fermentation by the byproduct based fermentation media has a huge amounts of the valuable compounds. And presently, we are also working on the using some rice hull hydrolysates. And here our approach is to just using the byproduct for the culture media. And in that cases, we are basically hydrolyzing all the modes by using some enzymatic methods, and only by using this a simple acid treatment using the SCL. And we found that the different combinations of this acid hydrolyzed byproducts, and also we using this way as a solvent media during the hydrolysis, and they are very good source for the culture media, and they have also the comparable growth with the commercial or MRS growth media. So this tofu way using hull hydrolysis is become one of, it may be the one of the important substrate or the media in the future, which can be replaced some of the probiotic media, natural artificial probiotic media for the probiotic culture production. So tofu basically this why this is the problem, because we are mainly concentrating, and the removal of some toxins or chemical residue comes through food. If you see this paneer, everyone is liking the paneer, and the paneer is basically prepared by the coagulation methods or precipitation method by using coagulant, mostly citric acids, magnesium chloride, calcium chlorides, and other coagulants are using. But people who are working on this aspect in the market, they are not very much aware about the food grade toxicity. So most of the cases they are using the food, not the non-food grade coagulant, and finally the residue of this coagulant comes inside our body through this food. So we are just developing a naturally chemical free coagulant, and by using this tofu as a coagulant. Simple is our approach to simply this tofu is fermented by use some of the probiotic bacteria, and after fermentation their pH is reduces, and so after the 20 to 18 hours the pH become near to the 4.5, and using some of the food vegetable byproduct to just solidifying, and we found a very good result when using this tofu way food grade tofu, a natural byproduct based culture media for the tofu preparation. If you see, this is the two paneer is there. One paneer is prepared from the magnesium chloride as a chemical coagulant, and other is from our this byproduct based coagulant, and we found a very suitable yield, suitable texture, organoleptic as well as nutritional composition in the magnesium chloride. Only difference is that a slightly decrease of their yield, but all other flavors, organoleptic, sensory, nutrition are just comparable to the reported culture media. So in this way, we are just replacing the chemical load from the foods. And we are also developing and using this fermented tofu way by some known culture in our laboratory for the production of antioxidant, and we found a very good antioxidant activity in their liquids after 24 hours. So this liquids after fermentation, we are just using directly for the some of the beverage as well as some of the supplement to the our food for the antioxidant source. And we have also developed some byproduct based fruit beverage, which now we are claiming as a probiotics. This is the basically the tofu way, the water discharge of the paneer. This is the fermented papaya based product, and this is the millet based beverage. In both cases, the product is basically prepared from the byproduct, and these byproducts are found to be very good organoleptic, sensory, as well as the good shelf life with the higher nutritional compositions. We have also tried to address the byproduct based fermentation media. If you know the millets, these millets are the very important, but the shelf life of millet flours is very low because of the presence of several polyphenolic compound, anti-nutritional compound, and if you see their covers of the millets have a huge nutritional and nutraceutical value. So here we try to develop a fermentation media, and this fermentation media basically used for the fermentation of the whole this fermented flour of the millet like sorghum flour, and found a very good reduction of the tannin, 36%, phytic acid reduction. These two are the major problem in this millets. So by developing this fermentation media, by developing this byproducts based conversion media, these problems is minimized, and I'm happy to ask that actually the flour shelf life is the 15 days, but by after this fermentation, by food grade, it become 49 days, which recently we published in the food chemistry. So this is the again one of the best use of this byproduct, food-based byproduct for the value additions. We have also developed the sauerkraut. This is a common products in the South India, South countries. But here they are not probiotics. They are just basically uses some naturally microbes. But here we develops some probiotics, means our tofu best culture of the probiotic is used for the sauerkraut development and found a very good stability and good probiotic property as well as a high antioxidant and phenolic component after using of this tofu way culture media. So this is again one of the important utilization of this byproduct for the fruit and vegetable based fermented media, culture. We have also published this paper. And nowadays, our group is very much concerned to the development of prebiotic, because in soybean and most of the vegetable, fruit and commodity, their byproducts are very much rich in the oligosaccharide. You know, these oligosaccharides are the very good source, a very good food supplement for the probiotic production. And nowadays, whenever we require immunity, our gut health and other property, the probiotics is very important. So here, the byproduct which is enriched in the oligosaccharide, we are just converting enzyme based and hydrolysis based conversions, and finally we try to develop as a source of prebiotics, as a source of food ingredient and gelling agent for the further production of probiotic and fermentation media. So we are also concerned, our group is also convert. Here we are byproducts. If you see, these are the byproducts we are using: and banana peels, soybean itself, tomato in the fields byproduct, and several our potato byproducts, our cabbage byproducts, our tofu okara byproducts, our hal byproduct for the production of this prebiotic. So this is again one of the important. In future, if you are interested to develop the food supplement in the for the food industry, these are the very good source after conversion. So a tofu way, our one group is also working, and we are also working on the development of the culture media for the microalgae production. Here our purpose is to not for the bio oil. Our purpose is to just enhancing and their protein source and carotenoid source. If you see this microalgae and the product derived from the microalgae is very costly because of this the culture media. So here we are trying to develop a food grade byproducts by using several processing technology to this culture media, and this work is going on, and we also working on different aspect for the during the hydrolysis of this byproduct for the final production media, and we also published very good papers in the Cell Press recently, and our this technology is just considered in the technology of the month. So this work is the going on, but we have found a very good result. And our result is just comparable to the artificial media which are using for the this microalgae production. And so if you see, if you got the culture media option, definitely there are several assembly, several industry just using this microalgae production, and that will be very good alternate in the future by using this byproducts conversion. And we are also developing some natural flavor compounds using some of the byproduct. So this work is also going on. And in our one of the group with the Manit is just developing the polyhydroxyalconate for the packaging material. And here we are just using some lignocellulose, starch bearing and plant or animal fat. And finally, they are using to the converting to the polyhydroxyalconate. And all the component here we are using that come from the food wastage. So conversion of this food wastage to a very good packaging format in the form of polyhydroxyalconate PHA is one of the best option for the utilization. So finally come to the conclusions, and basically if you see the development of process technology, our product is there, our alternate is there, but there is a lacking of some technology and process and the awareness. So if you develop some process technology for small operations, byproduct management, eco-friendly, easy and low-cost production procedure, definitely the utilization of these types of food wastage and byproduct will be very valuable. And if you come to the byproduct utilization in food sector, they have a huge potential. And our country, because of the higher production in different commodity, it can be the food hubs of the food, although after the post-COVID in the next 2 to 3 years. So finally, when you make this food wastage without lodging, about losing and managing. Definitely, this will definitely brings our slogans for the Baujan Sukhai and Baujan Hitai. And I just want to convey and request to all people to see the waste is resource and try to invest it effectively, because this is not simply waste, this is the waste, and we can enhance and we can just make our contribution in our country by just using effectively as a invest. And thank you. And this is actually my groups, our research teams from the different institutions, which we are working together. Some of the minutes, some AIMS and ICERs. And I also thanks to my all team members. And finally, thanks to the all the respected dignitaries and all the chairpersons and all the faculty, as well as all the students and other audience for their patiently and their listening to this talk. Thank you, sir. Thank you.
Thank you, Dr. Tripathi, for a nice presentation.
Sir, you are on mute. Sir, please.
Thank you, Dr. Tripathi, for your elaborative presentation on food waste, their utilization. May I ask audience to put up a few questions, if any, in their minds? Okay, if there are no questions, Dr. Tripathi has highlighted amount of food wastage, and he highlighted that 14% of the global food is wasted. This is a very huge quantity and can feed billions and billions of people who are in need of the people. So this is a great concern today. Dr. Tripathi has again highlighted one important issues, that India is at number 2 in food wastage. This is again a concern for our country, because we have people living in poverty. We can save that food that can be used for those people. Dr. Tripathi has highlighted that the food waste occurs at different stages, right in the field where it is produced, at harvesting stage, at marketplace, and then during the processing of the food. He has also highlighted that disposal of the food globally, that is not environmental friendly. That has toxicity in it, and that again comes into our food, which harms us. So food wastage is of utmost importance. He has also highlighted that agri-food waste, how it can be developed technically into applications in non-functional foods, supplements, pharmaceuticals, and other uses. He has also highlighted various processes by which waste and byproducts can be biotransformed into various usable products. He has highlighted many, like tofu, formation of soy milk, and so on. Many products he has highlighted. He has also highlighted that about 77% of food waste that comes from our houses and restaurants, and that is a major concern. Dr. Tripathi, what highlighted, entertains only 23% of the food wastage which can be processed. 77% still remains unutilized, and that is a waste and contaminates our soil, our water, and our environment. So for that waste, we have to make awareness among the people that we have not to waste the food and we have to save it. And Dr. Tripathi has rightly said, waste is a resource. We should invest it. So let us pledge here. We shall invest it and thank Dr. Manoj Tripathi for a nice presentation. Thank you, Dr. Tripathi. Thank you for joining us.
Thank you, Dr. Patrick, for your nice presentation. And in this technical session, the third speaker from Basmati Export Development Foundation, Dr. Dikshit did his MSc in botany from Lucknow University and a PhD on cyanobacteria from NBRI and Lucknow University both. After that, after PhD, he did his postdoctorate from IIT Roorkee, and he was a part of the integrated genome sequencing project during his postdoctoral work. And in that project, he published the papers in journals of international repute as Nature. In Nature, he got his publication and also worked on tomato genome. He got the honor of working as a senior scientist in South Korea. He was a senior scientist in South Korea for more than 5 years, and where he got more than 80 publications on working of the gene bank. And presently, he is working as a chief scientist in Basmati Export Development Foundation, and he has developed quality checking labs, fingerprinting labs, and testing for aflatoxins. And BDF is a part of APIDA, which is a very prestigious institute. And now I would like to hand over the mic to Dr. Anil Suroi for sharing the presentation of Dr. Dikshit.
Friends, Dr. Dikshit is working in our nearby institute, BEDF, and he is pioneered in the Basmati rice. Basmati rice is an important community of northern India because we have GI for this, geographical indications for this. Basmati rice is contaminated and not only our health but our economy is also hampered. And in past couple of years, certain fungicides have affected our exports but our economy is also hampered. And many consignments resulted from different countries. I request Mr. Dikshit sir to kindly on the Basmati rice and food safety issues.
First slide. Thank you and stage for talking on Basmati rice especially. So as you see, my topic is Basmati rice and food safety issues related to basmati rice. So before going to the main topic, I would like to introduce rice and particularly basmati rice. What is basmati rice? So starting from rice, actually rice is the staple food of more than 60% of the world population. It belongs to the genus most of you already know, Oryza, which has 2 cultivated and 22 wild species. Oryza sativa and Oryza glabrima is the 2 cultivated species. Among these, Oryza sativa is grown all over the world, while Oryza glabrima has been cultivated in West Africa only for the last around 3,500 years. You see more than 45,000 varieties of cultivated rice with grain color differing from white, yellow, red, brown, and black, including several aromatic varieties with small, medium, and long grains. The International Rice Gene Bank, maintained by International Rice Research Institute in Philippines, holds more than 1,17,000 types of rice, which include modern and traditional varieties and wide variety of rice. So basmati rice is a small group, say about 34 varieties of rice, which are aromatic and traditionally grown in certain geographic region of Himalayan foothills. Initially, there were only 6. Basmati is a group of with breeding efforts and high-yielding basmati varieties are developed by different institutions. Like there are several universities and institutions are involved in developing high-yielding basmati varieties. So now there are 34 basmati rice varieties. These are different from other aromatic. There are more than 100 aromatic rice varieties in the market or in the country, in the world. But these are different from other aromatic. Not all aromatic rice varieties are basmati rice. Basmati rice has some special unique characteristics which make them different from other aromatic rice varieties. This is mainly due to extra long slender grains that elongate at least twice of their original. You see it elongates up to 22 times from their original size, and they produce a typical aroma and distinct flavor, soft texture. As I told, to date 34 rice varieties which are notified as basmati rice under Seed Act 1966. You see, after cooking, they elongate and looks beautiful. These are minimum specification basmati rice. If any, you can call them like rice length. It should not even 6.6 millimeters.
This is due to amylosalt. It is very important. They have intermediate type of intermediate amylocentrate, that is 20 to 25%. Other varieties which are sticky, they have less amylocentrate, say below 20%.
Unique characteristics of basmati rice, and anyway, in 2016, actually, basmati rice has got the tag of GI. You know, GI is a geographical indication, and it is like a patent. It means that no other country can use basmati word for their rice variety. So there are 7 states which are tagged as GI area or GI states. It means cultivation of any basmati rice variety which are cultivated in these 7 states or districts of these 7 states. They are only covered under basmati rice. If they are grown other than these varieties, they will not be considered as basmati. You see the export volume of basmati. We have data from 2006 to 2021. You see that in 2006-7, it was export was only 1 million tons, 1 million ton. Then today we have 4.63 million ton, and revenue generated in 2006, it was 2,792 crores, and nowadays it is last year revenue was generated around 29,850 crore rupees. So this is a huge amount which our government has got by selling basmati rice, and you are surprised to know that there are only 180 countries in the world, and basmati rice is exported around 150 countries. So you see its popularity, and now come to the food safety issue associated with basmati rice. Prior to 2006, rice was considered a trouble-free commodity. With regard to food safety issue, before that, there were no food safety issues. The importing countries, they have not made any regulations for export and other things. After 2006, the major issues concerned dead insects and foreign objects. They were found in basmati consignment. Even that time, labor which were working in the shipyard by making containers ready for shipment. Even sleeper and their sleepers and cigarette butts, BD, they were found in the consignment. So these consignments were all rejected at that time. The second major issue is mycotoxins such as aflatoxins, then heavy metals, pesticides, residue. These are the major concerns, and I will go one by one. Aflatoxin issues. You know, aflatoxins are naturally occurring toxins produced by moths of the genus Aspergillus. There are few species of Aspergillus which grow during storage of the grain if they put under warm and humid conditions. High doses of these mycotoxins can cause liver failure and eventually death. So they are highly toxic. There are aflatoxins. There are several types of aflatoxins. In food, generally they are B1, B2, G1, G2 are found. In milk, M1 and M2 aflatoxins are very much prevalent. The major aflatoxin occurring in rice is B1. Actually, it is highly dangerous. B2 aflatoxin is not very toxic as compared to B1. B1 is the most toxic aflatoxin. More than 100 notifications on aflatoxin in rice appeared in the rapid alert system for food and feed of the European Union. European Union countries and USA, these governments are very much conscious about the food they are importing from other countries, and they put a very stringent regulations, other things. Yeah, if there are only two countries which are producing basmati rice, Pakistan and India. Pakistan was the part of India, so naturally when it was separated, so basmati rice also goes with him. So they also export basmati to other countries, but we are better than Pakistan basmati, as Pakistan exceeded the maximum limits of EU most frequently, followed by rice and rice products from India. So Indian basmati is better than Pakistan. The rising number of notifications concerning rice in RASFF of EU reflects the challenges that the rice industry and the rice trade have been through since 2006. Now come to the heavy metals. In the morning session, you have learned so many things about heavy metals, their toxic effects, and how they come into the food chain. So these are mainly present in natural condition and agricultural environment through various activities such as agriculture, mining, and industrial, as well as gases, which are distributed from the vehicles exhaust. The main source of heavy metal pollution in soil are irrigation, especially with sewage, solid waste, disposal, for example, sludge and compost refuse, the use of pesticides and fertilizers. You know, many pesticides and fertilizers which are we use, they contain heavy metals also, like arsenic, lead, cadmium. So excessive use of pesticides and fertilizers also contaminate heavy metals also in the soil. Sorry. So excessive accumulation of these metals in human body can lead to some diseases such as prostate cancer, high blood pressure, testicular tissues and red blood cells damage, kidney stain, coagulation of some proteins. So these are highly toxic if they accumulate in long term course. Some heavy metals such as arsenic, cadmium, lead, mercury, and chromium are considered as environmental pollutants and are recognized as toxic elements. Absorption as subsequent accumulation of these elements in excess along the food chain could have serious consequences on human health. Heavy metal contamination of food is one of the most important assessment parameters of the food quality assurance. Therefore, it is important to monitor the levels of such heavy metals in rice, particularly basmati rice, at periodic interval in order to prevent the health risk. And we have recently such a study was conducted by BDF in association with SBP University with 23 basmati samples collected from different districts of Western UP in order to understand the distribution pattern of some harmful heavy metals in paddy, brown, and white rice samples. What we did, we collected paddy samples from the farmer's field, and first we saw the presence of heavy metals in paddy. Then we make by hulling we made brown rice from paddy, and again saw the tested the for heavy metals presence, and then finally brown rice was milled, and we got white rice, and we detect heavy metals in the white rice of the same sample. So you see, these are some results I would like to share with you. This is cadmium. It is very harmful. and present in the environment through nickel cadmium coating of battery plates, and it is present in the vehicle's exhaust. So you see, we in paddy actually the concentration was highest. If we ask about the average concentration, the concentration are generally described in mg per kilogram, generally called ppm, parts per million. White rice has the lowest concentration of cadmium, and all the samples they were found in the safer side, as in white rice, which is generally not consumed, and only husk is also not consumed. White rice is only consumed, so they are on safer side as well as cadmium is concerned. Then arsenic. It is also found in all the 23 samples, but in paddy the level was highest, and it was on above the MRL value. MRL value means maximum residue level. If they are these contaminants are present more than MRL set by a country, it is rejected. But safely, husk is not consumed by human, so only white rice. So this is also at safer side. Then lead. Similarly, lead is also in white rice. It is in safer side. Only in husk it was present in very high concentration, and to some extent in brown layer also. But this is called brown layer. After removing the brown layer, you will get white rice or milled rice. You see, in case of chromium, it is a very interesting result. You see, in case of chromium, most of the chromium accumulated in husk, while in white rice and brown rice, it concentration was very low as compared to husk, paddy rice. So what are the conclusions from these studies? Concentration of arsenic, cadmium, lead and chromium in white rice were below their MRL values and are safe for human consumption. Their concentration was significantly higher in paddy and brown rice of the same sample. Accumulation patterns showed that on an average, rice husk accumulated highest level of chromium, 96%. 96% chromium it is accumulated in husk, followed by lead, arsenic and cadmium. Significantly high level of chromium and lead in rice husk indicates these contaminants might come through aerial deposition. It means these contaminants are present in air, and from there it deposited on the grain. You see, husk is hard protecting layer of rice grain. This is the outer layer, outermost layer, and is made of silica and lignin. It is indigestible to humans. That's why we do not eat. Though only white and brown rice are consumed by human, the husk layer is usually consumed by livestock. Thus, these harmful elements may also accumulate in human body through food chain and pose serious issues in human health. The rice husk seems to be a good absorbent for heavy metals and can be used as a bioabsorbent. Now, a lot of research are going on this direction using rice husk. It's a for remediation of heavy metals from the soil. Now, the third issue is pesticide. This is the burning issue, and export of basmati rice has faced severe problems in the past few years in different markets such as US, EU, and Iran. owing to detection of pesticide residues exceeding the prescribed maximum residue limits. Indian farmers, they are using pesticide in excessive amounts, and their residues are still remain and they come in the grain. So when it is exported and tested there, the foreign countries, importing countries, so they reject because they are not able to pass the set MRL values of those countries. According to All India Rice Exporter Association, roughly 30% of the total exports to the US were rejected due to high pesticide content. Similarly, in case of European Union countries, there are 28 countries in European Union. So 19% of the total export was rejected last year due to high pesticide content. So this is a serious issue. Following rejection of such rice, a number of rice exporters have suffered huge loss. So it is an economic loss also, health loss also. There are so many things. Declining of niche, but also demoralizing farmers to adopt crop diversification. So what are pesticide? By definition, pesticide is a chemical that covers a broad variety of compounds, including insecticides, fungicides, herbicides, rodenticides, molluscicides, nematicides, and plant growth regulator and others. An ideal pesticide should act selectively against certain pest organisms without causing any negative effect to humans, non-target plants, animals, and the environments. But it is not so. It is difficult to achieve absolute selectivity, and most pesticides are also toxic to humans and other non-target organisms. Because of their widespread use, ease of accessibility, the impact of pesticide on the environment has become profound and significant. This impact is no longer confined to agricultural fields, but can also include homes, business, academic institutions, and some other areas which are otherwise believed to be protected. Food products contaminated with toxic pesticides are associated with severe effects on the human health. The link between pesticides and cancer has increased exposure to pesticides may be linked to a higher risk of Parkinson's disease, Alzheimer's disease, and several types of cancers. According to one study, in over 30,000 female spouses of pesticide applicators, increased exposure to organophosphate was linked to a significantly higher risk for hormone-related cancer in female. Several other studies on human, animal, and test tube means in laboratory, had similar findings, reporting that exposure to organophosphate pesticides like malathion, turbophos, and chlorpyrifos may be associated with a higher risk of developing breast cancer over the time. How are these pesticides in food regulated? There are multiple kinds of studies are used to understand what level of pesticides are harmful. Here I am talking about the MRL value, how it has been decided, what should be the MRL, minimum, maximum residue level. So these are depends upon the several studies. Actually, some examples include measuring levels in people who are accidentally exposed to too much pesticide, animal testing, and studying the long-term health of people who use pesticides in their job. This information is combined to create limits For example, the lowest dose of a pesticide causing even the most subtle or sometimes no observed adverse effect. World Health Organization, Agriculture and Fashion the information to create a threshold for exposure that is considered safe. Threshold means MRL value. To do this, they add an extra cushion of safety by setting threshold 100 to 1000 times lower than the well or Noel value. This is the data we have collected 2019-20. We have collected around 97 samples from 3 states, including Punjab, and 97 samples were collected. The status of pesticide in each bar diagram. So you see, these are 97 samples. Each bar indicate a separate sample, individual sample. And on the top of each bar, there is some number is given. These numbers are showing the number of pesticide residue per sample. You see, there are 16, maximum number of 16 residues. They were found in sample number 41. And several sample residues, they are present in several samples. So this is the situation. Now we separate these data into state-wise. From Haryana, we have collected 36 samples, and you see almost all the samples, they have 6 to 15 pesticide residue in per sample. In Punjab, here also 6 to 14. On all the samples, more than 6 pesticide residues were found. In UP, situation is better. Maybe we have collected a very less number of samples as compared to Punjab and Haryana. But in Punjab, especially in Haryana, situation is very poor. And the farmers from Haryana and Punjab, they use excessively all these pesticides. Even many pesticides, they are banned, but still they are using. So in this study, we have tested around 98 pesticides on total 97 samples. Out of 98 pesticides, only 31 pesticide residues were detected in all the samples. So these are 31 pesticide residues. Mostly among these, most of them, they are insecticides. Few are fungicides, but mainly these are insecticides. You see, there are only 2 or 3 insects. They are troublesome in case of rice, but here it seems that more than 16 pesticide residues are detected in one sample, 15 in one sample. So farmers, they for killing only 2-3 insects, they put all kind of insecticides in a bulk. That's why pesticide residues are detected in these samples. If you see a number of samples with more than 9 residues, in Haryana, 89% samples, they have more than 9 residues in a sample. Punjab, 65%, Uttar Pradesh, only 35%. So in Uttar Pradesh, situation is still better, but in Haryana and Punjab, you see these are number of samples detected with residue level more than MRL values. So if you see acephate, it was found in 6 samples only, and out of 6 samples, 5 samples, they were residue level was more than their MRL value. MRL value was in case of acephate, it was 0.01 ppm, so very low level. But you see the problematic pesticides are buprofagin. This is also insecticides. Out of 80 samples, 81 samples, 51 samples were in 59 samples, residue level was more than MRL values. So if they export to other countries, particularly European countries, they were all going to reject it. So similarly, chloropyrifos, it is in fact anti-termite, and it is used in the field. Out of 97, all the samples they were detected with more than their MRL value. Similarly, other imidacloprid, luckily it was detected in 95 samples, and only one sample it was found above MRL value. So even it was found in almost 99% samples, but in only one sample it was not safer side. So these results shows that how much pesticides are used by our farmers in excessive amounts, and which goes actually in fact to human and consumed by us also. So in fact, when it was rejected, any consignment was rejected to European country or by USA. So exporters, they sent those consignment to other countries like Saudi Arab or Iran and other countries where there were no regulation. So, but now following the European countries and USA, even Saudi Arab and country like Jordan, they have made their own parameters for importing basmati rice from India. So now it is very difficult to export basmati rice if it has excess amount of pesticide residue. So what were the reason for excessive use? This is mainly, actually it is only the farmer who can control the presence of residue by following good agriculture practice. And it is a lack of good agriculture practice among farmers. It is the one and most important reason for contamination of rice with chemical residue, because once farmer put these chemicals in the field, no one can remove it from the field. And so there is a need to emphasize that farmer has to use pesticides properly if he does not want his produce to be rejected by exporter. There are so many side effects of pesticide. The quality is reduced, and price, you cannot get a poor quality rice good price. So for this, our organization, Basmati Export Development Foundation, we have decided to aware the farmers for the good agriculture practices. So to educate them. So every year during Kharif season, about 15 to 16 workshops covering all the 7 states, GI states, we organize one-day workshop, and we help the experts from KVK of this university in Punjab, Ludhiana Agriculture University, whatever agriculture universities are there in each state, we take help from the expertise from those KVK centers there, and organize one-day workshop for farmers which are growing basmati rice. And just to share information about improved package of practices, for quality production of basmati rice. Nowadays, this year we have started, we have aimed to organize 75 such workshops covering 7 GI states under the भारत का अमृत महोत्सव जो स्वतंत्रता के 75th वर्षगांठ के उपलक्ष्य में मनाया जा रहा है सरकार द्वारा, तो हम लोगों ने डिसाइड किया है 75 such workshops we are going to organize, and we are organizing. In fact, 40 workshops we have already organized. These are some workshops for aflatoxin, management of aflatoxin in rice. We have started these activities in 2008. These are photos from some workshop, one-day workshop. We have, I just mentioned earlier, for rice variety identification. This is the अभी अमृत महोत्सव के अंतर्गत जो वर्कशॉप हमने की मुरादाबाद में. And we also doing campaigning for quality aspects of basmati for use of a fungicide tricyclazole. There was a 2 years ago, it was a major issue because its MRL value was very high at that time, and suddenly European countries, they have decided to lower its MRL value of this fungicide. So most of the export from basmati rice consignment, they were rejected one by one. So it was a chaos on the farmers, and there is a loss of high amount of revenue to the country. So by campaigning, we use this organized Kisan Mela and some pamphlets like this, and to just educate and tell them not to use excessive amount, and they should use this fungicide at the right time and in the right amount, without they should not do without consulting the scientists from AVK or other institutions or organizations. Even farm to farm, actually, we went to farmers at their home level and distribute these materials just to educate them. Like this, we have in using van also. So these are the first approach, just to educate farmers who are using these excessive amount of pesticides. And other approach may be scientific approach. What it is an alternative approach to pesticide applications to breed basmati rice for disease resistance. Once you develop a basmati rice which is disease resistant, so automatically use of pesticide, you don't need to give extra pesticide, insecticides or fungicides. You don't need to put. This involves the transfer of a specific disease resistant gene from both traditional van races, cultivars, and wild relative of rice into existing high yielding basmati varieties. Scientists from IARI New Delhi, PUSA, have transferred the PI9 gene into its popular PUSA Basmati 1 variety. From where they got this gene? The gene source from Oryza minuta. It is a wild relative of Oryza sativa and provides a very high resistance called leaf blast and moderate resistance against neck leaf blast fungus. The resultant variety, which is called PUSA Basmati 1637. So now, after introducing these genes in PUSA Basmati 1, now they call this resistant variety PUSA Basmati 1637, which has all the characteristics of PUSA Basmati 1 and high yielding trait and resistance against fungus. So this is similarly two new varieties, Pusa Basmati 1718 and Pusa Basmati 1728, are released from IRI, New Delhi. Both of them incorporating XA21 and XA13 genes that confer resistance to the bacterial blight pathogen. The first varieties were introduced in Pusa Basmati 1121 and Pusa Basmati 1401. These varieties contain both genes obtained from Oryza longis temnata. This is another wild variety of rice, and BJ1, a traditional land races respectively. And these two varieties are performing very good in the farm and tested and now gaining popularity among the farmers and exporters also. So this is all about basmati and basmati rice related food safety issues. I just time limit. Thank you so much. Any question if you want to ask?
Any questions from students side? Thank you very much. Thank you, Dr. Dixit. Nice presentation. Dr. Dixit has highlighted basmati rice, which is a commodity of importance to India, around revenue of 30,000 crores. He has also highlighted that pollutants, they also affect this important commodity. He highlighted particularly aflatoxin, particularly B1 variant, which is of importance during confined human conditions and storage. And currently have got over 100 notifications from the Union System 2016. He has also highlighted that various heavy metals like arsenic, lead, they affect rice. However, they are below MRL values, so that is of not concern. But husk is larger quantities, and that is of importance for us because that again comes into the food chain. Similarly, pesticides, he has highlighted data from the basmati rice growing states, and again, but thanks to Dixit and his team from APID, their massive campaign has contained the pesticide use in basmati rice. And to control, keep this commodity alive in the country, it is highlighted that farmers have judiciously use fertilizers and pesticides so that this commodity remains alive in the market. Alternative strategies for eco-friendly pesticides or varieties are the alternatives which he has suggested. So thank you, Dr. Dixit, for sharing your views on basmati. And now is lunchtime. I have already summarized previous speakers' lectures, so I am not taking much of your time. And my thanks to all the participants here this morning, particularly students. I hope I have got many insights in this important field. I request you all to join the second session, which is again two important speakers, Dr. K.K. Pant, IIT Delhi, and get insights into the topics.
Thanks, Dr. Dixit, for a insightful presentation, and also rapporteurs and chairperson of the first session for the smooth organization of the technical session. And now I would like to request Dr. Organizing Chairman, Dr. Pankaj Kumar, to present a memento to invited speaker, Dr. Anupam Dixit. At the end of this session, again, I request Dr. Pankaj to present the moment to chairman of the session, Professor Anil Sirohi. I will request chairman of the session and organizing chairman to present. The rapporteur of the session. Okay, now we'll meet for the second session at 2:30.
Please join us for lunch and kindly assemble. Try to be at here at 2:41. Let it be.
See you next month. हाँ सर, आप मोड खराब हो जाए ये बड़ा पॉसिबिलिटी है नहीं, हम हो तो खराब हो जाए तो देखिए अरे नहीं टीचर वो डाउनलोड कर दे मैं लॉग आउट कर दूंगा उसी से चल रहा है जब इम्पोर्टेंस होती है तो गायब भी हो जाता है ये छोड़ दो मुझे मैं तो सबसे पहले आऊंगा निकल के देखना अरे भैया मैं तो दिल्ली अपने रूम
लेकिन ये बात तो
So welcome, Dr. Pant. Can you hear me?
Good afternoon. Can you hear me?
Yes, sir.
Okay,
So we'll start within 5 minutes.
okay,
Just I was checking.
sure, sure. My screen is visible. PPT can be seen. Can you see my PPT? Dr. Ranjan? Can you see my PPT? Can you hear me?
Yes, sir. I can hear you.
Yeah,
You are audible.
yeah. My screen is visible.
Yes, your screen is also visible.
Okay, fine.
So, Dr. Pant is honorary faculty, University of Queensland, Australia,
Adjunct Professor, University of Canada, Saskatchewan, Canada. Joint Faculty CRDT IIT Delhi. His research interest includes heterogeneous green catalysis, syngas and CO2 conversion to methanol and DME, and chemicals, green technologies for sustainable energy and environment, biomass pyrolysis, gasification, hydrogen, and renewable fuels from coal, biomass, feedstocks, coal, crude oil, chemicals, and residue generation. He has worked on various projects, including consultancies valued more than 1.5 billion Indian rupees from India and world's topmost premier companies, organizations such as GAIL, HPCL, DRDO, Tata Chemicals, Noord, and so on. He is a product of IIT Kanpur for his PhD and Masters, and his BTech in Chemical Engineering is from HPTI Kanpur. He is presently Dean Faculty, Professor Chemical Engineering. He has held many posts like Professor HAG, Department of Chemical Engineering, Head Department of Chemical Engineering, Joint Faculty CRDT. In addition, he has invited visiting professor at Tuskegee, USA, University in Canada, Brainstorming, University, UK, recipient of DART International Faculty Visit Grant. He has been Warden, Vice Chairman of GATE, Vice Chairman JAM, Chairman GATE, and so on. So his biodata includes a long list. He has been awarded many prestigious awards like LEAP, Chopra Applied Research Award, Ardelia Award for Excellence, CSIR ICT Award, and so on. So list includes a long list. If I go on speaking, it will take a lot of time to speak about him. So he has got many fellowships like Fellow Royal Society of Chemistry, London, Fellow National Academy of Science, India, Fellow Indian National Academy of Engineering, and so on. He is member expert committee of many advisory committees of the in country and abroad. So I welcome Dr. Pant. I will not take much time between the participants and the speakers. So I request Dr. Pant to start this talk.
Thank you. Yeah. So very good afternoon to all of you. And am I audible?
Yes.
Yeah. So thank you, Dr. Ranjan, for speaking about my biodata in such a elaborative way. I am really grateful. So before starting my talk, I would like to thank Professor Rekha, the Dean of the college, SUPUAT, and all other organizations, right, for inviting me for this talk, which is one of the important aspects when you look at the food chain, right, and the contamination of the food quality, right, where you talk about the mission, when you talk about, right, Clean India and right, food for all. So that these are a couple of important aspects. And in that way, when you talk about the food quality, then the water becomes very important, right? And because most of these things, when you talk about the plant, right, growth of plant, and then how this, right, the role of water. So all of everybody knows that we have a huge amount of water available, but when you talk about the drinking water, there is always a scarcity of the drinking water. So we always strive to find out more and more sources of clean or potable water. Especially if you look at the rural part of the country, the challenges are much more, we have the lot of threats of arsenic, we have threat of fluoride, and not only these, some kind, it's a kind of what you see from the rocks, from underground water, there are issues. But sometimes it is because of the anthropogenic or man-made type also. Lot of industries are coming. You have a lot of pulp and paper, tanneries, So all these kind of, and if you see this data on this right hand side where your Indian map is shown, you see that 54% of India faces a high to extremely high water stress, and all these what you see, the red zone, these are the where the scarcity of the clean water, so it means even now, when a large number of the population in India is looking for the clean and drinking water, and so it is a responsibility, when you talk about the sustainable development goal. So clean water, clean food or good food, hygienic conditions. This is one of the important aspects where we have to give it to the society. So Clean India Mission, when you talk Swachh Bharat Mission, Atmanirbhar Bharat Mission, all these concepts which have been given by Government of India, it includes that how to make the society sustainable, and that is the where every individual has to work. When you talk about the plantation, trees, save water, avoid the pollution, so a lot of plastic goes into the environment, water, a lot of other type of contaminants, it goes to the water, and that creates the threat to the society. So we need to understand all these things, You see that the fate, when you talk about heavy metals. So today I am covering mainly all these kind of arsenic, lead, cadmium, mercury, chromium, copper, zinc, nickel, and there are many more, when you talk about, because every industry is giving some kind of the industrial effluent, and although the concept of zero waste discharge, zero water discharge, that has already been given to every industry, and they are trying hard also, to what extent now the treated water which comes and mixed to the the lake or river, so those the qualities are being maintained, but need a kind of societal awareness. That's very important, Sometimes because of lack of awareness, these things are being done. You see that the heavy metals from pollution, natural sources. When you talk about so this goes we have a lot of phyto remediation concept. All of you know that we have a lot of wetland areas, and these where you have the chrome contaminated water, or some kind of tannery effluents, and where you have a plant, something like that, and nearby you have a river, and you see that and you are consuming this kind of whether it is a kind of plant based, or a kind of this kind of fish or all these are. So all this is a kind of cyclic process. You see here the heavy metals, absorb, dissolve, and then again it goes into this. So and if these are aquatic, so then this goes to our body, So that's what. So it's a kind of transfer of the heavy metal from fish to humans, as a result of bioaccumulation and biomagnification of aquatic food chain. So this is where you talk about the entire cycle, and then heavy metal contaminates the life, and that's where we need to look at the metals, Just I have given some glimpse that what are the different kind of sources, As I said, it's a kind of anthropogenic activity. It may be because of natural sources. Natural source, lot of rocks are there, and during the rain, because of the acid rain, so these seepage comes into the groundwater, so that is a kind of natural sources. When you talk about natural rock, volcanic eruption, wind blown, dust particles, aerosols. So this is one of the kind of where we don't have any control, but these from the rocks, from the mines, coal mines, all these things, it comes into the ground, and other thing is that a kind of other where the industries are there and they are polluting, when you see the platinum, or this car industry, where the chrome plating thing is done, leather industries and several other heavy metal industries, they also have this kind of thing. And see, so this, when you talk about this, so industry, where mining, combustion of fossil fuels, metal processing, Then agricultural side also you have pesticides, inorganic fertilizers, sewage sludge, fungicide, and domestic level also, right? Lot of the battery, right? The lot of time we talk about electronic waste now. Inorganic or organic waste, right? That also had some kind of right metal contaminant. Detergents. So this is another issue which is nowadays coming. Personal care products, so which I will come back later. But these are the kind of right the contaminants which are affecting. If you see the data in terms of arsenic, cadmium, chromium, copper, lead, manganese, mercury, nickel, zinc, every right these kind of right metals, right, which are coming from different sources, right, which I was talking about the polluting industry, paint, pesticides, batteries, all these are contaminating the right the groundwater and this water, right, which goes into the food and other plant waste, right. So everything that is when you talk about the some types of plantations, right, or wetland areas where the plants are there. So those plants also take out those water, right, because it's a capillary action, and because of that, the water, right, sucks, and along with the water, these kind of contaminants are. You see that permissible limit, right, of heavy metal in the drinking water. These guidelines are already available, right. So basically, when you talk about the drinking water standards, right, which are coming more and more stringent, European norms are much more stringent, right, and WHO guidelines are already defined. So like arsenic chloride, all of you know, right? It's a threat, right, in most of the part like Rajasthan, West Bengal, now in the UP also, right, now, right, and several other areas where you see the groundwater is contaminated because of fluoride and arsenic, right, and the concentration range, right, which is 0.05, it is written, but it should be much low, right? 0.01 is the right the guideline where the future guideline may come in that way. So we need to understand that how the groundwater or what is the fate of this, right, when you talk about all these kind of pollution, right. So CPCB, they have some guideline, but that is for the industry purpose or what should be the discharge. But this is drinking water limit is very important as per the WHO or US EPA norms, and this should be much more stricter, right, compared to whatever the data shown here. So mercury is another threat, right? Arsenic, mercury, zinc, so these heavy metals, because lot of these things are now coming from the CFL lamps, lot of CFL lamps, right, after the use, we just throw it without any understanding, right, and broken, and a lot of mercury, right, then your mercury thermometers, which were earlier being used, right, there are a lot of right areas of this, right, when you talk about the south side where the Hicks was the company who used to make this kind of thermometer. Now the problem has come, right, the mercury has gone to the right soil, and from soil it has gone to right that is the agricultural, and finally to the water also. So those issues are coming, and solutions should come accordingly. So this is what the picture, right, again and again I am keeping, right, that accumulation of heavy metals in the food chain when you talk about, right, how do, right, they go into the right main body. So that's the concentration. There is a kind of, right, what you call the mechanism, right, a capillary action, right, when you talk about the high concentration versus low concentration up to certain concentration levels when it goes into the human body, and then they, right, affect the human body or organs, right. So there are several things when you need to understand, right, the oxidation reduction cycle, because it's a question of electrochemistry, right, depending upon the pH, right, and water pH, acid rain, right, and then these, right, becomes more and more active. So that's what written here, biomagnification of this entire concept when you talk about, right. So metal precipitation, then there is a kind of methylation also, right, because the human body or more of bioorganic, right, there are the degradation of these, right. So heavy metals generally they don't degrade into the, this is the another important aspect, right, when you talk about they can come and they remain in the metallic form in these are, but oxide forms, chloride forms sometimes, right, they go into the different oxidation states also. So suppose arsenic is there, and then there is a right pH of 3, right, it will have different form, pH of 5, it will have right, a pH of say acid medium and alkali medium, so arsenic 3, arsenic 5. So those kind of oxidation reduction states may be there, right, and some of the say chromium you see trivalent, hexavalent. So some form of these metals may be difficult to remove also, right. So this entire cycle, if you see, right, metal oxidation reduction which happens, right, animals and human feed, right, when you look at a kind of cyst right here, the salmon feed, right, and then these are again the different kind of, right, what you see the concentrated metals which go into the what you call phytoplankton, right? They utilize them, and that is zooplankton feed on phytoplanktons, right? Concentrated metal. So this cycle, when you talk about the effluent discharge toxic metal, this goes through a series which comes from the industry, which comes from some other right of resources, and then this goes into the different cycles, right? Sea animals or the land animals, and ultimately it affects the human being also. So contaminated plants, right, with that is again a mechanism here, that how does it goes to the right plant roots, right? These all metals, and we have also done right a kind of the research on this algae cultivation, right, and from the wetland area, and which was right the kind of the concept of phytoremediation. So certain kind of algae biomass, they capture these kind of heavy metals and they grow at a faster rate, right? And but when you talk about whether you are using these algae biomass for the food versus right, you can take it for the fuel purpose, so food versus fuel. So when you talk about for the food purpose, then you need to understand, are these really right good for the right taking it as a food, because this will have all these kind of right heavy metal, which because this has been created in the area what you call wetland area, right, or a concept of phytoremediation. So when it happens, right, the body, human body organs are affected, and that's what it is mentioned here, right, different kind of right, it's the brain, right, affects the brain, it affects the liver, it affects the kidney, digestive system, reproductive system, skeletal system, mitochondria, right, which is oxidative, what I was talking about, phosphorylation or ATP synthesis. So all these things, right, when you talk about the heavy metals, so up to a right below a certain permissible limit, they are okay in the body, but when the concentration increase to beyond a certain range, then this affect the different body organs, right? And several cases of these type of cases have been reported in those areas where the water is contaminated of these kind of right heavy metals. So we need to understand right the entire what you call the concept of this, that how there is a population of these heavy metals into the right plant based or into an animal life, and then how does it come to the body system. So mainly all these type of contaminant comes from the water, right? And these were right the water, I am saying drinking water, potable water, or this water may be right in the plants also, like sometimes lot of this vegetation is done in the area which is a wetland area, but we don't think that what is the effect say in the sewage area, right? A lot of the vegetation, although the growth is faster, but there are a lot of side effects also, right? So organic versus right, when you see the fertilizers and all these things, they also have those kind of impact on this kind of food product. So we need to understand, because this caused a serious health effects on the human. So see what we have discussed here, you see that the Indian scenario in terms of average values of heavy metal content in the surface water body in different countries. So I'm not going in rest of the part, right? You see that China versus India. So we are right 1500 microgram per gram of the iron. We talk iron, right? It is a sometimes we use the iron right as an adsorbent also, right? And we talk in terms of iron nanoparticles. People talk right lot of nanoparticles, right? But the fate of these nanoparticles, right, in the drinking water is also to be studied, because sometimes the nano is always better because it has a high surface area, right, per unit volume. And then you say that when you have a nanoparticle, right, the efficiency is better. And of course, the electronic configurations are also different. But at the same time, after treatment, what is the fate of these nanoparticles? Where do they go, right? That is, I'm just talking one is from the contaminant side. Sometimes we use the right metal or metal oxide for the treatment also, right, for different purpose. So now that is also important, that they may leach out into the drinking water itself, and we need to find out how much has been leached out. So nano is always not good, because sometimes the nano is good for treatment purpose, because your reaction kinetics or your right overall mechanism is better, but at the same time you have to make sure that these nanoparticles are intact with the adsorbent and they have not been leached out. So that's very important. You see that the concentration which I'm talking, right, the permissible limit is less than 1 microgram of this per gram when you talk about drinking water, right, or even 0.01 time. But if you see that in the surface water, the concentration as high as 1500 right microgram per gram of iron have been reported. And similarly, the chromium, right, when we talk about the concentration level less than 0.01, and the concentration levels are as high as, let's say, 400 to 500 microgram per gram. So these are the couple of scenarios, right, where one needs to understand that how the concentration of these metals, right? So iron, manganese, cobalt, when we are talking these are selected metals, but beside that there are several other metals contaminants, right, which affect the food, or which affect the water contaminant, water, and then in total, if you look at the food system, food chain, right? So a lot of R&Ds have been done in order to understand, right, that what is the concentration of these heavy metals. One can easily extract these metals from this kind of food, right, or vegetable products, and then one can study and then analyze that what is the contamination level of these kind of food products, and basically especially in the wetland areas when we talk about the effect of these contaminants. Then industry, right, every time we talk about the zero waste discharge concept, right, lot of norms have come, that the pollution control board, right, already CPCB, right, or DPCC, they are doing a lot of work in that, right, textile industry, which, right, a huge, right, producer of this kind of, right, discharge, and there are not only textile, tannery, and several other industries. Although every industry is trying hard, right, to reduce the concentration, or after, right, there are different kind of treatment methods by which they reduce the concentration in the form of BOD, say, right, less than 30, and other type of concentration, and they are following the guideline. But this is where the treatment is required, and you see that, right, the wastewater constituents which cause harmful and hazardous effects, right, to all kind of, right, and this industries are also need, right, to be trained, right, and sometimes especially the small scale industry, right, because they just look at in terms of profits. Many industries, right, in the small scale industry, right, or sometimes like the metal extraction unit, they do in the batch at home only, right. These are very small scale. They cannot afford the treatment process like this, because the treatment will require a kind of, right, investment. And so sometimes small scale industry, they just try to avoid this. Although the norms are stricter, so they have to follow, but sometimes they just, right, ETP is not working, or not efficient. Then the effluent treatment plant, right, if it is not efficient, then it will go to the right, river. And so one needs to understand, right, and that's what I am saying. Now another threat which is coming nowadays is those kind of all kind of the personal care products, right, and these are the new kind of contaminants nowadays which are coming, right. Say lot of cosmetics are being used, right, for, right, that is the face cream, right, a lot of body lotions, right. But nobody is thinking that after the shower and it is going to the, right, where does it go and how does it affect the entire, again, the problem of this. And then similarly, so all these what you see, all kind of these, right, contaminants are there. So not only in the industrial, but at domestic level also these problems are coming. And this is where I was talking, the source and pathways of these pollutants, right. So we always blame the industry, but now a lot of these things are coming from domestic households also, right. So you see that this change, the domestic versus industry. So you have the wastewater treatment plants, and sometimes we at the, right, what you see, the block levels, this kind of wastewater treatment, where the kitchen water, all this, right, gray water that can be treated and again used for the irrigation purpose. So that can be done, right, and that's why now every hoteliers, right, they have been given guideline that whatever the waste, the concept of zero waste, if it is not zero waste, at least 70% or 80% of the water should be recycled in the system, right. So it can be a kind of, right, which can be used for the, right, a kind of for the kitchen garden, or some kind of, right, the secondary use, which are not for the drinking purpose but for secondary. So that is where the purpose is very, that the concept is that, right. So if the discharge is illicit, it goes to the river. That's what that's a gray water, right, or blackish water, which is not only from that. So these both two streams are undesired or unwanted, right. So all these should be routed through a kind of the wastewater treatment plant, or some kind of effluent treatment plant. If not at a small level, but at least at a kind of central effluent treatment plant. So those which industries which are small, they have to route all the kind of effluent to one unit, which may be a centralized unit, right? Or if they, so it depends on the kind of capacity, and then only it should be discharged, right? And separately sludge should be utilized, river should be, right, this should be discharged to river, or the water works, right, that where it can be drinking water and again separated. So that is where the kind of investment is required, a kind of reverse osmosis units, right, or some kind of ultrafiltration unit or some other adsorption this process. So that is. So this plus this groundwater, you see that irrigation water, soils, all this runoff. So this can be entirely kept in a closed loop. This is what the concept should be done, right? Although how much of this has been done or is being done or will be done, that's a very important point. That but this is ideal thing that everything should be done. But you see that, right, most of the time, right, only 80% of this is done, or sometimes only 50%. So that is making the contaminant either at nanogram per liter or microgram per liter, and all these are present in the environment. And that's why the Environment Protection Act, that is US EPA, they also have given the guidelines, and all the contaminants which are emerging, right, concern including pharmaceutical and personal care product, which I have talked, PPCP, right, that is very important. So pharmaceutical industry, right, lot of say phenol, organic compound, these are going into the groundwater, right, and that is going. So these are a lot of volatile organic compounds are coming into it. And this PCP is now a big challenge, in fact, because the consumption of cosmetics, consumption of body lotions, right, this is being, right, this has increased a lot tremendously, right, if you look at the in last 10 decades, right, and that's where last 10 years. So when you talk about this, right, so this is where you need to, right, take care in terms of contaminants of emerging concern, CEC we call it, right, which include the pharmaceutical and personal care product, and these are increasingly, right, these have been detected in the surface water now. Earlier days it was not being considered, but this is a new area of research that how to treat. So sometimes what we are creating the problem, right, ourselves, and then we are looking the solution for it. So that is how to use these kind of cosmetics, right, or is it really need, right, those kind of body lotion, because this is, and then if it is going right from your bathroom to the, right, the water body, then this is again another issue or threat. You see that, right, the kind of chemical structures of all these kind of, right, PCPs, right, or different kind of what is written, and that has made, right, this, you see the scenario, large number of articles are being written on this right now, and in top in high quality journals, right. So because this has become a new challenge, one can prepare a review article and give the fate of these kind of, right, PCPs, right, on the human body or on human health, right, or food quality. So a lot of things are left to be done, right, that is. And you see that I'm not going in names, but parabens is a kind of, right, PCP, right, where you have a methyl paraben, ethyl paraben, right, all these kind of, right, contaminants which are now coming up in the, right, because of the cosmetics and the, right, body lotion. So these, what you see, the different kind of structure, these are, right, different kind of parabens on this and different names, right, chemical names. So mainly phenolic compounds, these are there, and they are now creating the serious health hazards, right, in the body. So that's what went on. So this is the guideline which have been already been defined by WHO or European bodies, right? 8 to, right, say microgram per liter. So that's what the in the percent drinking water, right, for the all these kind of the, right, the products which are nowadays coming up in terms of the human body, or, right. So that's it. For drinking water, one needs to be very cautious in terms of the concentration or for the safe guidelines for their presence in the drinking water. So this, right, the compound which I am talking about, paraben, right, ethyl paraben, methyl paraben, right, or propyl paraben, then trilocan. So all these, right, are very, right, when to a certain levels, right, they are okay. But you see now the maximum and minimum concentration which have been reported in surface water in India itself. So the one of the R&D could be that, say R&Ds could be that, just take the waters from different bodies and measure, determine the concentration. So that is where the ranges have been given, and these are now a threat, right, for the future. So that one needs to find out. Even in Australia, if you see the concentration of TCS, they have this 14 to 75, and in India this range has gone 954, right? So which is a, right, the reported concentration in different parts, right? And so that is where the PCP presence in, right, different part of the world, you see, right. So which is no reported case, something like this, versus the 36 to 42, right? That is a kind of the concentrations of PCB in different parts of the world. So this is the threat where the all these, right, dark gray, dark blue colors have been shown, and we needs to understand and take the care for the future. So industrial wastewater treatment, I'm not going in detail of this, but there are certain remedies for the, because I told you that zero wastewater concept, or zero water discharge concept is already there. So there can be those who work on those areas of wastewater treatment, physical, chemical, and biological. All methods are physical, of course. This is just the where you need there's no chemical reaction basically, and you can take it again and again. But the limitations are there, low concentration versus high concentration. Lot of unit operations are involved, lot of challenge of maintenance are involved. Then chemical, which may be faster, like oxidation, precipitation, coagulation. All these methods are also being used, for different purpose. So which method is to be selected, and that third one is the biological method, which is slow, but you need some kind of biological treatment, some bacteria, sometimes enzymes, or some kind of plant-based materials also can be used. So the methods are available, but the efficacy of these methods, in terms of treatment, is also to be understood. Economy of the process, then how fast the kinetics is working for this, and how many times, that is recyclability, or multi times if it can be used. So that is also required. So reverse osmosis, nanofiltration. Am I audible? So, am I audible?
Yes, you are audible.
Okay.
Yes,
There was some issue. So reverse osmosis, nanofiltration, ultrafiltration, all of you know, because that depends on the pore size of the membrane, So this is when you talk about at domestic level, most of you must have used this reverse osmosis, But sometimes you feel that, when the TDS is 0 in the water, it is good for you, So there should be maintained certain level of TDS is required. Say 40, if you are saying 40 ppm is the TDS, that's okay, 40 to 1. But 0 is not good. Sometimes we remove all the kind of minerals and then add the mineralize it again in the water. That is again a kind of not a good thing, So you need to understand that what should be, how the treatment should be done. And then you are using a lot of wastewater when you treat it, the percentage of water during the reverse osmosis, how much is the pressure drop, all these things are important, and similarly here, when you talk about the carbon filter, distillation, coagulation, so these are kind of, generally methods which are used for the treatment, I'm not going in depth of this, but you need to. Another one which you see, the adsorption, Everybody talks about because it's a easy process, It can work under a certain condition, but you need to understand the mechanism. It's not like that you take any biomass, convert into biochar, and then work with that. It will remove all kind of metals, So you need to understand that what is the mechanism. You see the surfaces, The adsorbent surfaces have different kind of adsorbent surfaces are there, It's not that you have a carbon, it will do the miracle for all kind of metals. One needs to understand what is happening with different kind of adsorbent, which is just shown here, Because it has to be done under certain concentration levels, So this is what you see left in the right hand side bottom. You have the adsorbate which adsorbs on the surface, then it, So this is the water contaminant, And it goes into the pores, Here are some kind of sites which are available, which I have shown here on the left hand side. And then this is a kind of adsorption, then surface reaction and desorption, and then the multi-layer, single layer, multi-layer adsorption. So many theories. And from there, how will you remove them? Because all these are adsorbed here. Your water is treated now, but after a certain period of time, what happens to that, So that is another important. And this is relatively easy to handle the process. Most of these adsorbent-based materials are available also, but a lot of things are still to be done on this. But challenges are there. Carbon dot people talk, those are different kind of methods by which it can be. You see the publications which are coming now, on these areas, different areas. So adsorption is dominating, in terms of the number of publications. So that is another areas of R&D, where one can look into, or develop different kind of adsorbent. But it's a very challenging, and novelty of the work is very important. So when you talk about in adsorbents also for treatment of water, several adsorbents have already been tested, for removal. Nothing. So what is the novelty of the work? That the people talk activated carbon, People talk commercial activated carbon from the biomass-based materials, Then non-conventional low-cost adsorbent, where you have a waste from agricultural industry, and that can also be used as an adsorbent. Some natural materials like clay, zeolites, silica, all these can also be. Then bioabsorbent also being tested, like biomass, biopolymer, peat, right? Then you have a series of other methods like that. But I'm saying that the contaminated water, right, which is generally to be made to be potable, right, for the kind of right food grain, food application, then you need to have to find out the method, its economy, its scalability, and then only you can use it, right? Otherwise, everybody is talking about nanomaterials, right? So nanomaterial adsorbents, which is nothing but similar a part of when you talk about adsorbents, then you have nanoparticles, nanotube, nanowires, nanorods. So I'll cover some of these, right? Then composite, nanocomposite adsorbents, right? And several other kinds. So a lot of things are being done on the adsorption-based method, and that is helpful also, but especially at the low concentration range, right? For high concentration range, this is not feasible process. Suppose you have a 200 milligram per liter, right, of some heavy metal, and if you are using adsorption-based process, that will not be economical, because just with a small, because it has certain capacity, right? Adsorption capacity is very important. It's a surface area, right? It's pore size. All these are important when you look at the treatment methods. So I'm not going in detail, but these are the kind of equation when you do the adsorption, you need to understand, right, how the adsorption process can be effective, and then you can do the kinetics for the scale up. So these are a couple of experiments which, right, well-known isotherms which are generally used for the adsorption. But what I am trying to give you here, that how this contaminated water can be treated, right, and how can you improve the adsorption capacity? Because I told you, you may convert a biomass by some heating at 550 degrees centigrade for 5 hours, you will have a biochar, right? Biochar is one of the adsorbents, right? Good adsorbent. You can use it as a soil fertilizer also when you have biomass waste, or you can use it as an adsorption also, right? But when you have this kind of, you need to understand what is this here, what I'm saying, how to strengthen the adsorption capacity, right? How efficiency of adsorbent, right, can be improved. And then you need to understand all these things, a detailed chemistry, functionalization, because sometimes functionalization works, right? That is, then functionalization will be related to what kind of metal. So you need to understand that what kind of metal. So this is what we call metal synergy, right? So synergy of the organic complex, right? That is, this will make a kind of complex. Then is it just on surface or is it in depth? You need to understand. Then there may be a kind of functionalization of the surface. So sometimes you may have formaldehyde based treatment, sometimes you may have other sulfuric acid kind treatment. So you need to understand that. Then the crystal, sometimes the crystals may be, right, particular metal will absorb on particular surface which is crystallized surface. So you need to develop the crystal form, right? Some kind of planes need to be developed, and that can make right morphology, right? The pore size distribution, all are important. So this is what just shown, that you can have a carbon graphite surface, reduced graphite, graphene oxides or graphene, right? And then over that you can personalize with different, right? As I said, formaldehyde, some kind of epoxy groups, right? And these can help, right? Because it is nothing but a kind of 0 point charge on the surface, hydrophobicity, hydrophilicity. So all those kind of interactions, like pi-pi interaction, that is for the polyaromatic hydrocarbons, right? So you need to understand that what kind of contaminant is there in the water, and to remove that water, what kind of mechanism will come in picture, and accordingly the adsorption treatment is to be given. So these are called functionalization of graphene material for inert adsorption. So now sometimes many people talk about the nanoparticles, right? Silver nanoparticles, which are very good, right? Antibacterial, antifungal, but silver is little bit costly. One can replace the silver from the similar group. So you need to understand whether copper is good, copper in which form it will be good. So people are working with copper based also, right? So instead of silver, you know that at domestic level, generally people put the water in the copper, right? Bottle or copper, right? And next day they take, because copper is a good kind of, right? It has very good antibacterial, antifungal, right? Those kind of properties similar to the silver based nanoparticle. You see that where the source of these, right, the kind of plant-based materials can be used for this purpose. And this is a biological extract. So this kind of work we are doing with one of the Australian partner in our lab also. And then metal and source, right? And then you have biological reduction, you can have a nanoparticle, right? And this is where the entire mechanism in terms of pH, temperature, reaction time, reaction concentration, this does not require a very high investment also, right? It's simple. In the beaker and the lab scale, one can do this kind of work, right? And then you see that how these silver nanoparticles, right? When you talk about capped stable silver nanoparticles, you can very easily characterize this kind of, right? So reduction and then your nucleation. So where the, right, you see the silver particles, and then this is how the capped and stable silver nanoparticles, because leaching is undesired. So that's what you need to understand, that how these nanoparticles can be made stable on the surface, and then you need to understand the effect of each process. So this kind of green synthesis of silver nanoparticles, when we are talking, it should not, right? We are talking green, so no toxic byproduct, renewable, right? Non-complex, cost-effective, energy consumption should be low. So all these are challenge, in fact, when you talk about how many unit operations are done, how many unit processes are being done. So those things are important when you talk about these kind of particles, right? So we do this kind of, right, what you call the right peel of this, and that can biosorbent, right? This is a kind of biological method. So this is where you talk about, right, a bottom-up approach. You start from this kind of material, and then you trap the silver nanoparticles on these kind of, right, biological surfaces, right? And then these, we are looking at treatment of PPCP, that is what I call like personal care or pharmaceutical and personal care products, right? Degradation. Even this treatment can be used for some kind of low concentration of the organic, right, effluents, right, coming from this. So this is a kind of, right, disinfection of microbes, and you can have the clean water. So any dirty water, if you have these kind of surfaces, you can make it a clean water, right, which is definitely potable. So this is a kind of scheme which we are, right, we have, and some literature based on literature. So this is a typical methodology for biological synthesis of silver nanoparticles. And this is what you see here, right, that the application for the water treatment, the silver nanoparticles, right, these are widely being used, right. So lot of already technologies are available, but low cost synthesis of the silver nanoparticle is very important, and stable also, right, it should not go. So you need to have the characterization, right, all these characterization methods are done. So contaminated water can be converted to silver nanoparticles. So you need to synthesize, characterize, then look at the antimicrobial activity, then photocatalytic degradation activity for these kind of, and water treatment. So that is where you can impregnate these silver nanoparticles over the graphene oxide. So that those are the approaches, right, one can work on that. So this is, I'm not going in detail, but this is a kind of photocatalytic degradation of, right, PCP, which is personal care products, right, by silver nanoparticles. It's just a kind of mechanism which needs to be understood, right, that how this, right, a band gap is important here when you talk about the, right, because of otherwise what will happen, they recombine. So this is very important, that how to tune the band gap for the photocatalytic reaction. And you see the concentration, right, this one, which is the reduced graphene oxide, the blue line here, that has gone, right. So in terms of the, right, UV light, when you see the treatment, so this definite concentration of reduced graphene oxide and silver, that works better for the reduction of structure. But these things are still, right, at the infant stage, and lot of work is to be done when far as the, right, these kind of, right, bisphenol A, right, all these kind of organic pollutants or PCP are concerned. So a lot of challenges are there, you know, in these kind of treatments. So this is where we are just looking, right, different kind of approaches based on the literature, right. Then you can scale up the contaminated water, right. There are the live E. coli cells, all these, right, pump it. Then you have a silver, right, activated carbon, and this design, so it can be upward or downward, and you can have the disinfected water, right. So activated carbon, silver treated nanoparticles. So silver nanoparticles can be replaced by copper. So that is again an R&D, that how effective will be copper nanoparticle, and to what concentration levels, right. So that's what. So compared to silver, right, the copper is cheaper, and but the mechanism and electronic configuration, right, of copper, because both are from the same group, but this copper can also be used, right. So that's where one needs to do the R&D in terms of treatment. So this is the, right, when we talk about the green biomimetic silver nanoparticles, right, and that can be used for mercury treatment also. So a lot of R&Ds are being done in this area now, because mercury is also one of the heavy contaminant, and this side, right, there is a need to treat, right, because coal-based plants also have a lot of mercury, and this has gone right into the river, and that has contaminated the water, and it has gone to the food product. So sometimes these kind of natural, right, bio-based product can be used. So extract of this bio-based product, right, which is, this is a Vigna mungo seeds, right, and then you just convert it into the extracted seed, and then you just treat it with the silver nanoparticles here, right, and then you can have the silver nanoparticles, right, by SAM or TAM, you can do that. And this is how these are the stabilized silver nanoparticles and tested for mercury at different concentration. And then you can have the right treated water and look at the stability of all entire process. So that is important for the. So silver nanoparticles are widely being tested for different kind of treatment. So when we talk about this kind of treatments, the carbon-based materials which are effective nowadays, but what kind of carbon-based material? Like carbon nanotube, right? This is the graphenes, right? Or some kind of multi-walled carbon nanotube, simple fluorine, because all these things you can develop from bio-based products, biomass, and that's the kind of what I'm talking, green adsorbent. So the properties are important. So properties can be modified also by some physical treatment, by some chemical treatment, right? So that is where you talk about these important, right? The interlayer distance. So you define the graphene versus multi-walled carbon nanotube. So these are different, right? Thermal conductivity is very important. So this is graphene, which is highly thermal conductive material versus multi-walled carbon nanotube. And then other things like pore volume, of course, this is important, but surface area is important, right? When you talk about the mesopore surface area or pore volume and surface area. So this is 322, and here it depends on the range, right? So that is where the graphene oxides, right? These are, because for adsorption purpose, high surface area per unit volume is very important, right? So graphene sheets or layered graphene sheets or reduced graphene oxides, so these can be used as an adsorbent based material for this kind of treatment of the wastewater. So this is right how heavy metals, right? Because the mechanism is important. That's what I said. Right, this is the carbon-based material, so that is a kind of physical adsorption right here. And then there may be a phosphate or carbonate which are present in the water. They may also affect. There are some minerals, salt. So effect of co-ions is also to be understood, because otherwise all these heavy. Suppose your water is hard water, so all these will get adsorbed on the surface, and then your absorption capacity will go down. So whether the hard water or soft water, what concentration, all these are important when you talk about the removal of heavy metals by using the adsorption. So functional groups are sometimes very important. As I said, sometimes the formaldehyde treatment, some resin treatment, all these, they add the functional group on the surface. And that also controls the pH, ZPC, what we call 0-point charge on the surface. And based on that, your adsorption capacity can be there. So one of the major challenges now when you see that adsorption and the wastewater treatment is that how to enhance the adsorption capacity, right? And based on the addition of the functional group. So these are a couple of, you see that different adsorbent. Rice has corn straw, right? And there are different kind of, biological materials also, and the carbon nanotube, which can be obtained from the, other bio-based business. So rice has carbon versus straw, right? Corn straw carbon. So their properties are, depending upon the treatment, depending upon the synthesis, you see the adsorption capacity, right? 175 versus 47, just for the same chromium, right? So what I mean to say, that it depends on the structure which has come at the end in terms of carbon formation. So, and this structure which I'm saying, they are resemble to this kind of structure. So that is required in terms of understanding of that, right? So that's what I wanted to show. So all these kind of, when you talk about the personal care products, right, these kind of adsorption based process can be effective, right? And this is what we are doing now, right? We are synthesizing these kind of carbon nanotube in the graphene interlayer, right? And then trying to understand that whether they sometimes say when you talk about the aerosols of graphene, right? They are highly porous material. So their density is of the order of say 0.02 to 0.03 gram per centimeter cube. Such a lightweight material, and they are being used in fact for the treatment of oil spillage, right? Or dye removal from textiles. So that same thing, these kind of materials, right? They have been reported that 15 times higher capacity of desalination. That is what the given 40 versus 633 for these kind of carbon nanotube in the graphene interlayer. So, but how to synthesize these kind of material, right? How much is the cost of treated water? And those things are again need to be understood. But these kind of materials are really good for the treatment by adsorption process, right? So synthesis is very important. You see that aerogel, when we are saying these have a definite structure, right? And they need a freeze dry under. So your drying is very important under high pressure, right, and low temperature. So how this drying has been done, and that gives this kind of structure of gel structure, what I'm saying, colloids, and then silica gel type structure you can draw from here. Another important thing when you talk about the carbon nanotube versus graphene is that how these are oriented on the surface, right? lamellar, something like vertically, something like this, right? So that is again, you see different kind of graphene oxide, right? The kind of carbon nanotube there and their interaction. So when you do this kind of treatment, so chemistry is very important. Understanding of this, that how does it work, right? You see that first is carbon nanotube, which is adsorbed horizontally to the graphene surface. Second is the carbon nanotube, which is adsorbed perpendicular to the graphene surface. And third, it is wrapped in. So different carbon nanotube will have a different kind of physicochemical properties when you talk in terms of the, and accordingly, right, you see that, right, the depending upon their architecture, say 1, 2, and 3, or 1 and 3 have been shown, because this has not been effective generally for water treatment, and that has shown that how much their adsorption capacity in terms of their own weight. So that is also right important in terms of the treatment of this. So that kind of van der Waals interaction, pi stacking. So all these, right, the chemical structure is to be understood for development of these, right, different graphene structure. So one can make these kind of structures, right? That is what written here. The aerogels of graphene, carbon nanotube, agro waste from wastewater treatment. So this is a kind of the method which we have synthesized, right? A graphene oxide. You can see the pictures here, right? And these are the kind of the characterized different kind of biomass materials, right? And ash of agro waste, right? Basically biomass ash that can be used, right? So you see that this can be used for treatment of the refinery wastewater, phenols and its derivatives, right? Treatment, the graphene. So it's a kind of hybrid rice husk if they convert it into hybrid aerogels, right? Which can be overlaid on the graphene or carbon nanotube, and then this can be used for treated water. So carbon nanotube is right one of the very versatile material which can be used, right, for the treatment of the water. And this carbon nanotube you can make from the rice husk, right, and make a porous aerogel structure. So that's the entire process which is written 1, 2, 3, 4 here, right? Preparation of graphene oxide, and this can be done just by the pyrolysis process and little bit treatment, or even sulfuric acid can be done. Instead of pyrolysis, you put biomass rice husk into the sulfuric acid for a certain period of time, you will have this kind of structure. Then preparation of multi-wall, you need to do some kind of treatments, right? Then conversion of rice husk into ash, you can use that ash also, and then novel hybrid aerogel. So different kind of, say, fly ash based silica can also be used for the aerogel structure. So that is where the lot of R&D are being done in these areas nowadays. So what is that? What I am talking from? How these kind of structures have been developed, right? Synthesis of, right, graphene oxide using graphite structure, then conversion of rice husk, right, into ash content. That is what I shown in the earlier slide, and synthesis of polydopamine modified carbon. So these are kind of methods, right, which are generally used. I am not going in depth of this, but these methods are generally used. So one can do the R&D in terms of the novelty of the process and improve the structure. So you see the structure of the hybrid aerogel, right? How these steps are done. So these kind of structures may come, right, in terms of graphene oxide or carboxyl rich graphene oxide, which have the property now in terms of the adsorption. So those things are important in terms of the chemical treatment. So that is a kind of synthesis of hybrid aerogel multi-wall, right? When you talk carbon nanotube, right? And then you have some kind of dopamines and all these amine-based compound, and you can have this kind of structure, right? And these dopamines are, right, slight treatment, you can make them in the different, right? Because you need to understand the metal, right, type of metal, type of organic contaminant, their pore size, right, kinetic diameter of the molecule, and then accordingly you can control these structures and pore size. Because larger molecular weight compound will not go through the pores, right? Then it will not work, right? So those things are to be understood. So this is what I am not going in detail of this, but the modification of activated carbon, right, for adsorption is important. So you see that for different kind of rice husks, these are treated with nitric acid, sulfuric acid also can be used here. Then malachite green again, right, is used, right, where you have a carbon source, right, granular sludge based material. Again, some nitric acid can be used, and these are the adsorption capacities which have been reported, right, for lead, for chromium, all these things. So these are the, right, when you talk about the concept in terms of the heavy metals, right, which are present in the wastewater or water and being discharged into the, right, drinking water sources, then that, right, there are the, right, harmful effects. So we need to do the treatment before they go into the, right, river or discharge into the river. So, right, so, and that makes the entire cycle, because whether it is a plant, right, the heavy metals are present in the plant materials or food-based materials, or drinking water. The fate is right. Ultimately, it is going to the human body or animal's body, right? So that is to be avoided. So this is what the different kind of metals, right? Cobalt, cadmium, lead, zinc, copper, and all these kind of multi-walled carbon nanotube, right? With some different functional groups, they have the kind of bonding with these kind of, right? That is where written in metal bonding, then oxygen, right? Hydrogen, carbon. So all these, right, bondings are functional groups, right? If one can understand, and that can be, so this is a kind of review paper which we have written in the science of total environment. So the details are available there in that paper. But this can definitely increase the absorption capacity when you give the treatment of this. So we modified the surfaces like hydrophilic and hydrophobic combination, right, which has the improved absorption capacity. The idea is that you are comparing the electrostatic interactions with these metals, right, and then that again increase the the activity of this metal. So these are a couple of results I can share with you, right, on the slides. So different kind of iron oxide based, right, these graphenes, right, and trapped or silver nanoparticles. So different activities are being done in the research group, right, for the water treatment. So that is where we are looking in terms of improved absorption capacity. These are some work, right, which is on arsenic and third and arsenic 5, and using the iron oxide based, right, impregnated activated alumina. So that is a kind of lab skill study. You study the, right, and then look at that how these kind of, right, alumina versus iron oxide, right, nanoparticles and impregnated on alumina, and then fate of these nanoparticles also studied, right, for the treatment. So these are the kind of, right, breakthrough curves, absorption breakthrough curve. So these the results which have been shown, right, very effective in terms of the removal of arsenic from the groundwater. So right, so more than 99% arsenic could be removed by these kind of absorption based process. So we are synthesizing, right, arsenic chloride removal. So this is what the, right, a kind of biomass based material. So we synthesize this kind of adsorbent and then we use it for a biochar, and this biochar was used and a lot of response surface methodology was done, kinetic study was done. So this kind of activated biochar also had been found effective for arsenic and fluoride removal. In fact, this was the simultaneous removal of arsenic and fluoride from the groundwater, right? And that has also, because this water was taken from the Rajasthan area in the near Udaipur, and was used. So it had the concentration of 5 ppm, and that reduced to a level of 0.1 ppm from these kind of studies. Aerogel also, which I said that this kind of carbon-based material agrochar, right? So you see that how light is this aerogel. This is synthesized from the right freeze drying method, right? Sonication and freeze drying in our lab itself. And you see that we kept it on the flower, and this is very compressible material. If you press it, it will squeeze, and if you release your pressure, it will come to its original, and very stable up to 700 degrees centigrade. So this was used for textile dye and all these kind of, right? You see that this is the treated water, and it has captured all these kind of, right, dye and all these contaminants which is present in the water. So that is a kind of an ultralight flexible aerogel, which is, right, on this is derived from the carbon nanotube, which is derived from the methane decomposition, and then we use this silica and CMC mixture and synthesize this, and that has been found very effective for the, right, textile effluent water and other kind of dye removal. So those kind of what I'm saying, and even oil spillage also. If it is the oil spillage, this is the oil which is right, lube oils, and that can be removed from this, and then it is recovered also. Oil can be recovered. So we tested different type of graphene, mesoporous silica aerogels, right, for absorption of phenol also, and different phenol compound. You see our results of phenol, catechol, resorphenol, all these which are what I said, the personal care products, right, and we are getting very, right, good impressive results with these kind of products. So that is where the graphene oxide, right, when you talk about these, right, aerogels, and then you need to kind of, right, CTAPs, all these kind of, right, the structure, 3D structure, and these structures are effective for treatment of this phenolic compound, right, and so that is where the, right, lot of things are to be done, right, and this can be effective for future. So this is how the synthesis is done. So this is the typical method for synthesis. So I'm not going, but these kind of graphene oxide aerogels are effective for treatment. Right. So again, this is the absorption of graphene oxide using on CMC aerogels. right? And a kind of kinetic study was done, and all these things are right reported in literature also. And this is our lab work which has been done. Then this is again reduced graphene oxide. So we are working on different kind of metals, heavy metals, right? So that is also which can be synthesized, and then these heavy metals can be removed, right? Whether it is antimony, third, 5 by using this kind of graphene oxide. So this work has just not been done in all. This is by one of the reference which we are taken. So what I'm saying that this kind of graphene oxide can be used for multi metals present in the wastewater. At the end, when you talk about these, right, the fate or contaminants of the heavy water, right, or the water, the contaminated water on the food material, there are a lot of challenges when we talk about the nanomaterials, because most of the time we talk about titanium, nano, silica, right, nano, zinc. All these are effective for water treatment because they have very good absorption capacity, and with this absorption capacity, which can be used for the treatment of the water, right? So, but at the end, we need to understand that does it leach out also? So you need to look at the ANSI standards. The standards are available where you need to understand the leachability index also. So after that treatment, whether the treated water has leached out, right, nanoparticles of these, then again the water quality is not good. So this is again an important study where we need to understand the environmental implication of the nanomaterials in the water treatment. Because most of the time we remove one kind of waste has been treated, but what is the fate of the, right, the kind of the material which has been used, kind of adsorbent which has been used for treatment, so that is important. So that is what you see, that resources and energy used, the kind of reagents which are used, drinking water applications. When you talk about non-intentional release, right, you are not interested in it, but it is going into that. So those release, right, landfills, wastewater sludge release or leaching. So that is also important. That sometimes the adsorption also may not be effective if it the adsorbent itself is going into the treatment. So that is also you see the different sources which you see, and it has been observed that certain kind of these kind of materials, right, are not, you see the concentration is increasing for these kind of metals, right? Whereas this kind of carbon nanotube and graphene based materials are relatively stable, right? So that is also important, that how these studies which have been done, how these are, right, exposing, and their use is right, contaminating the environment, whether it is in a, because nanoparticles are, you know, highly unstable, right, and they will remain in the water, they will go into the air, and then it will create another kind of problem. So one problem is solved and the problem is created. That is not a concept of sustainable goal, development goal, right? So entire thing should be in a closed loop, and that is where we talk about fate of nanomaterials in the environment. So this is very important, right? So this is where you talk about, right, how the, right, the aquatic life is being affected. You see that, right, the mechanism, understanding the mechanism of this is very important here, right? The aerosols, all of you know, all of acid rain, right, you know, and because of those also, right, because the pH is low, so all these, right, the deposition which is happening, right, where you talk wind erosion, right, these are the natural, right, when you talk about advection, right, so that is the, because of the movement of the air, right, these nanoparticles, right, when you talk, they go into the environment, right, so we talk nanoparticles, but how safe are these nanoparticles is very important, right? This is where we talk about the, right, the wastewater treatment plants and all these things, right? The soil, right, these are infiltrating the soil jobs and deep soils, right, they remain there, they come to the water, they flow, right, suspended sedimentation. So all these mechanisms are to be understood, right? So leaching of nanomaterials which cause, right, their release into the environment. Exposure pathways of life cycle of nanomaterial in the environment needed to be modeled. And these nanomaterial can produce toxic effects, right, on the planktons and microbes in the water. So those things are very important, that how the treatment, right, is done, but what is the ultimate end. So entire life cycle assessment of these also, right, nanoparticles also. This is what's shown right here in the slide. Well, it looks very complicated, but not very complicated if you try to understand the physical transformation, biological transformation, interaction with these kind of, right, macromolecules. So a lot of things are to be understood when you talk about, right, this biological degradation, the environmental transformation of nanomaterials. So that these nanomaterials move from one form to another form, right, because they are highly unstable molecules, right, and they have all kind of transformation, right, biological and physical, chemical, right, and thermal also, right, and so that is where you talk about, right, how these nanoparticles contaminate the right, groundwater or right, ultimately the food items. So sometimes lot of food items have right been found contaminated with these kind of right nanoparticles because these have not been checked or not been observed. You see that right how these right are just accumulated here, heterogeneous right aggregation, what you say, increase the size of the particle because these are unstable particles, right? So they try to agglomerate right when the surface and reach to a certain surface area. So just as a summary, just to conclude, right, what I'm saying here that textile industry, pharma, waste, all these industries, right, they are right contaminating the groundwater, right, and there is a need to right, and this personal care products which I'm talking now, right, is a threat, right, and the treatments can be done by aerogel, graphene, low cost absorbent, and that the nanoparticles. But at the end, right, the clean water should be sustainable, environment friendly, right. So that is where the right lot of things are to be done. The toxicity of the adsorbent, cost analysis, modification for increasing the effectivity, disposal of adsorbent, energy generation from waste adsorbent, efficient use. So all these things are still not resolved, right? And that's why the lot of things are being done. Lot of workshops and all these things are to be done. Brainstorming sessions are to be done. That what are the different treatment methods to be taken, right, for the treatment for these kind of wastewater. So just I thank you everyone for your patience hearing, and I acknowledge my research scholars Marut Jain and Saddaf, right, who work on this kind of wastewater treatment presently, and earlier student Tony who worked on arsenic and fluoride, and Vikrant Sarin who worked on chromium removal from the industrial effluent. So thank you everyone for your patience hearing. If you have any question, please feel free to ask. Any question?
Elaborative talk on this second session, fate of heavy metal contaminants in water and food and treatment approaches. So before we end up his talk, I would request any participants who have the questions. So Professor Pant shall answer that. Anybody who wishes to speak?
Yeah, if you have any question, please feel free to ask. You can connect me through my mail IDs also, right?
Mike. Malaraj, Mike. You ask.
There is one question. My question is related to the paraben and methyl paraben and high and
Your voice is little bit feeble. Hello.
Now you can ask.
Sir, one question is related to the personal health care products. They contain methyl paraben, paraben, and triclosan. So my question is that how is it impacting the human health?
Yeah, actually, these all are carcinogenic chemicals, right? We have submitted a review paper very recently on this. This is a new problem, in fact. Earlier, this problem was not right entertained, and recently it has been observed that all these kind of right personal body care, right? Although the concentration is very low, but even the low concentration is affecting the human health, and lot of carcinogenic effects have been observed. And therefore, in US already, right, these things have now been restricted to use where a lot of these kind of chemicals are being used in this. So natural type of right, the herbal-based products are being recommended instead of going for this chemical-based product for this. And treatment-wise, when you look at right, very limited treatment methods have been observed so far. What we are doing in our lab, we are developing these, as I said, right, from peels of some right, say like this orange peels. And then we are converting into some product like graphene based, and then absorbing silver nanoparticles on that, and then we are using it for treatment. So that kind of work we are doing presently for treatment. But very limited work has been done on this PCP and even for the PPCP in total.
So any other questions from Dr. Chauhan you want to ask? So if there are no questions, I thank Professor Pant for speaking on very important topic of fate of heavy metals in water and food chain. He spoke about the scarcity of water. About 50% of Indian population faces this scarcity. Then heavy metals from various sources, natural sources, river and cyclic rains, acid rains, and how it comes from pesticides, detergents, sewage, etc. He spoke about the guidelines of WHO, US EPA, ISI, ICMR, CP, and different metals like chromium, copper, cadmium, arsenic, mercury, etc. And he spoke about accumulation of heavy metals in food chain, how it reaches to the plants and to the different body parts. It reaches to the cells, organelles, mitochondria, and so on. He also spoke about heavy metals in industrial wastewater. So small scale industries do not treat the wastewater, so it creates a lot of problems. And pharmaceuticals and PCPs, personal care products, and it reaches to households, then different classes of personal care products, and it contains natural hormones, phenolics. It PCP contains different type of materials which are harmful, and then different methods of wastewater treatment and its efficacy. Then absorption method, it includes activated carbon, low cost adsorbents, nanomaterials, and he also spoke about different forms of adsorbents, then green synthesis of silver nanoparticles, how it is synthesized, characterized, and utilized, and its efficiency. And then carbon-based materials for effective wastewater treatment, graphene sheets, and carbon nanoparticles for removal of heavy metals. Then various strategies for methods for removal of these heavy metals. Then modification of methods like modification of activated carbon, and then modification of carbon nanotubes. So again, he spoke about aerogels for oil adsorption, GMS adsorption phenols, challenges associated with the nanomaterials, fate of nanomaterials in the environment, and toxic effects on planktons. So different transformation of nanomaterials, physical, chemical, and biological methods. So with this, I thank you, Professor Pant, for very elaborative talk on the topic.
Thank you very much.
We have another speaker with us, Dr. Rekha, Professor Rekha S. Singhal from Department of Food Engineering and Technology from ICT Mumbai. She shall be speaking on ecotoxicity of nanoparticles and its influence on food chain. So before we proceed with her talk, I would request Dr. Rekha Dikshit to introduce the speaker.
Thank you, Dr. Khan, for such an informing and enlightening lecture. And indeed, our students are benefiting from the exposure to the advanced research in the field of remediation of pollutants, whether natural or man-made. Thanks, sir. And it is my privilege to introduce to yourself a very distinguished professor from ICT Mumbai, Dr. Rekha Singhal, who will be delivering the last lecture in this workshop today. And from the morning, I was seeing that the workshop is very vital, very vibrant. Students are very enthusiastic because of the very interesting lectures which are very relevant to the problems faced by the real world today. So Professor Rekha Singham is very distinguished, and starting from her journey from PG education, and did her PG from the University of Mumbai in 1986, and from the same university, she completed her doctoral degree in food technology. And after that, she began her career as a lecturer in food technology. And from lecturer, she was promoted to the reader and then professor in food technology. She had to hold the prestigious position of after heading the Department of Food Engineering and Technology for 5 years at Institute of Chemical Technology, teaching and research experience at UG, PG, and doctorate level. Ma'am has successfully supervised 100 MTech and 39 doctoral candidates. Area of research of Professor Rekha Singham includes carbohydrate chemistry, technology, that include modified starches, microencapsulation of sensitive food components using conventional and newer raw materials, polysaccharide interactions, biofuels from agro-industrial waste, fermentation, production, and downstream processing. Well, Professor Rekha Singham has the prestigious positions as investigator and co-investigator of major projects by AICTE, DBT, UGC, and several industrial houses. She has also been editor of Journal of Food Science and Technology, Carbohydrate Polymers, International Food Research Journal, and have invited lectures in various institutes. Professor Rekha Singham has collaborated with scientists from BARC Mumbai, Forest Research Institute Dehradun, NBRI Lucknow, and IALTO University Finland, Illinois Institute of Technology Chicago on projects of the common interest. She has referred to many internationally reviewed journals. Also being a member of Selection Committee for Promotions, BARC Mumbai, RL Trivandrum, faculty position, various universities across India, adds feather to the caps. And a member of scientific panel of FSSI on food additives and water. Examiner, she also had the distinguished record of examiner of PhD thesis in University of Mysore, Kerala University, Bhavnagar University, Thapar University, IIT Kharagpur, Jadhav University. It is a very long list and a very illustrious and enlightening, motivating biodata, and we should also aspire to gain the knowledge because learning is a never ending procedure. Students in her guidance have received awards for master's, doctoral degrees in ICT. And fellowships, her fellowships include Maharashtra Academy of Sciences for significant contributions to engineering sciences and technology. And she has been recipient of various awards. To name a few of them, Malviya Memorial Award, Biotechnical Society of India for year 2011, and MM Sharma Award by Marathi Vigyan Parishad for the year 2015. Author of one book, 35 book chapter, 2 patents, 35 review papers, and more than 380 research papers. About 400 research papers in peer-reviewed national and international journals speak volumes about the experience and passion for research in Professor Rekha Singham. Now I would like to invite ma'am for the much awaited talk. Thanks.
Yeah, thank you, ma'am. Thank you for the words of introduction. I'm having some problem here with me, so I would be grateful if you could share the presentation. I'll speak. Yeah, because actually my computer has crashed. I'm working from my mobile. That is why this request is.
We are doing so. We are doing so.
Yes, sir. Yes, sir. Okay. Yeah. Thank you for your words of introduction. And because I know this is the last lecture and I have an advantage because many of the aspects that I would have liked to cover have already been covered. So I will try to cover on something different than probably what has been covered. So the topic which was given to me, or rather which I selected, is ecotoxicity of nanoparticles and its influence on the food chain. Please let us go to the next slide. Yeah, let us please go to the next slide. Okay, so this is just to give a brief idea about the sources of nanoparticles. And we know that, we have majorly 3 categories of nanoparticles that are released into the environment from varied industrial sources, including food. But let us talk first about the sources. So we have natural nanoparticles, which come from dust storms and which could be terrestrial or even extraterrestrial. Then we have seen a lot of forest fires in recent times, volcanoes, even organisms, and even ocean and water evaporation. So these are all nanoparticles which are natural in origin. Then we have a lot of nanoparticles which come due to human activity, but they create environmental pollution. And these are anthropogenic nanoparticles. Some examples over here are engineered nanoparticles. Then Professor Pant has spoken about personal care. So we have cosmetics and other consumer products, even indoor pollution, cigarette smoke, as well as diesel and engine. These will all come under the category of anthropogenic nanoparticles. In addition, we have metals and other dusts ranging from beryllium to lead, copper, cadmium, silver, gold, and even polymeric fumes. So these will all come under metals and other dusts. Now, why I have taken this is because all of them find their way into the environment and from the environment to the food and then into the human body. So let us please go to the next slide. Yeah, next one. Hello. Okay. Now these are the pathways and transformations of nanoparticles in the environment, which Professor Pant has covered, but I will just briefly, describe it once again. So as we see that we have a lot of nanoparticles coming from waste treatment plants, and these nanoparticles, they can have 2 routes. One is on the land and other is through the water bodies. Now, when it comes into the water bodies, it will undergo a lot of changes. For example, these nanoparticles would either homoaggregate, that means aggregation of nanoparticles of similar types, or it can undergo heteroaggregation, which is aggregation with another nanoparticle of a different kind, or it could undergo various kinds of transformation and subsequently degradation, or it can undergo dissolution, and it can also get incorporated inside a live organism. So this is what happens when nanoparticles find their way into water bodies. However, when they come onto the landfills or land, there can be 2 things that could happen. One is that it could go below the soil surface, and again there will be similar kind of changes, namely degradation, dissolution, homoaggregation, and attachment, or that is heteroaggregation. Apart from that, there are also photodegradation that can happen, as we can see, and sedimentation could also happen due to the flocculation of nanoparticles. Now, these homoaggregation and heteroaggregation are dictated by the soil conditions, for example, pH, ionic strength, and the presence of cations. Now, let us see the next slide. Yeah, next one, please. Yeah. Now, when these nanoparticles come into the soil, in the soil, they come in contact with organic matter. Now, this organic matter could either be plant roots, soil fungi, or bacteria, or it could be algal biomass. And these nanoparticles, they can combine with organic matter, and you can see that they will increase the bioavailability of these nanoparticles towards the organism. On the other hand, you will see that these nanoparticles can also combine with toxicants. Here it is toxicant A and toxicant B, and you will see that these two toxicants get surrounded by the nanoparticles, and they increase the bioavailability to the organism, which we can see in the center with dark arrows. You will also see that these nanoparticles can bind to the salt ions, and in this case, there is actually a reduction in the bioavailability. So effectively, what it means is that once the nanoparticles are released into the environment, they interact with the organic matter, and then their interaction influences the bioavailability of these nanoparticles either positively or negatively, depending upon with which constituents it is associated with. Let us see the next slide. Next one, please. I think next one. Hello. Yeah. If you see over here, we have three examples of three nanoparticles, metal nanoparticles, and their action on a freshwater alga, which is Pseudocarcinella subcapitata. And this is a synthetic soft water which has been evaluated. The toxicity has been evaluated as IC50. That means the concentration required to kill or inactivate 50% of the biomass. And lower the value, the more is the toxicity. So we can see that the ionic form as well as the nano form of silver, and also you can see cerium oxide. These are much lower as compared to the microform. With respect to zinc oxide, we see that the IC50 is similar for the zinc oxide, but then zinc oxide does get converted into zinc ions, and so the toxicity is due to the soluble zinc ions. In the case of cerium, it is also there is a direct interaction with algal cell wall, and it is also supposed to be assisted by light. And same is the case with silver, where there is oxidative dissolution of silver as toxic silver ions. And there are questions whether it is also assisted by light. So we know that, the form in which the metal is present could have differences in their toxicity. And the magnitude is quite huge, as one can see from this figure. Now, let us go to the next point. Next slide, please. Yeah, hello. I wish I could have shared it. Hello. Now, let us look at the application of nanotechnology in food industry. Now, if you look at food packaging, the consequences of adding nanoparticles for multiple reasons, this lands up in creating problems for the environment. But when we use nanoparticles in food processing, again for so-called beneficial activities, it lands up entering inside the human body. Okay, so both of them, and from the food packaging through the environment, again it will enter the food body. That I will show later. Now, in the packaging, food packaging, why nanoparticles are used? It is whether irrespective of whether the packaging is biodegradable or non-biodegradable. But then the physical performance, mechanical properties, these are actually enhanced. And similarly, you can have nanoparticles as antimicrobial agents, which I heard Professor Pan talking about. And we also have smart packaging, which are in the form of nanobiosensors, and they can tell you whether the food inside the package is worth consumption or it has spoiled and has to be thrown away. So these are the applications of nanoparticles in food. But when it comes to food processing, there are again direct addition of nanoparticles. For example, in anti-caking agents, if this anti-caking agent is in a nano form, there is prevention of lump formation. When you use it as a gelating agent, then there is an improvement in food texture. And then we have nano additives and nutraceuticals, where it is believed that they have better bioavailability. And then we have nano encapsulation and nano carriers, which are essentially to protect the aroma, flavor, and other ingredients in food. And that is where the market price of the food product lies. Let us see the next slide. I will focus in the next slide on the nutraceuticals, or rather the use of nanoparticles for fortification. The government of India has now actually permitted fortification of lot of food products, and there are many players who are wanting to actually use nanoscience and nanotechnology in this area. But these engineered nanoparticles, particularly for food fortification, you will see that they present new morphologies. Their chemistries are quite different, and therefore their physico-chemical, mechanical functions are vastly different. These are used either in the form of nano-sized powders, as I said, to increase the absorption because of increased surface area, or for better absorption, improved stability, and better delivery, as is the purpose for nano encapsulation. And also we have nano chelates, which can deliver the nutrients more efficiently without affecting the color or taste. Given that these are positives, there are many negative points as well, and these are known to be potentially toxic due to very high aspect ratio. That means it cannot be cleared from the lungs via the mucociliary escalator. So there is a possibility of accumulation in the lungs. Biopersistence, that means it will remain in the body for a potentially longer time. And it has reactive surfaces. So there is every possibility of it reacting with the constituents in the tissues. And also composition and stability. They are capable of delivering super physiological concentration of materials. For example, we need iron fortification, but excess of iron can be much worse than the manifestations of excess of iron can be much worse than the deficiency of iron. So when we use nanoparticles for the purpose of food fortification, we have to take this factor that it could deliver a much higher level than that is expected. Now, next slide, please. Next slide actually will show you when I told you that nanoparticles present new morphologies. So what are these new morphologies? We can see, first of all, there is a change in the dimension. You can see the original circular shape can have a change in the morphology as a rod shape. It could have charge, which can be negative, positive, or neutral. So there can be variations. Or these nanoparticles can aggregate, or sometimes they could be altered on the surface, which I just mentioned, or sometimes inside the nanoparticle, there could be something that is entrapped. So therefore, you will see that there is a difference in the morphology of these nanoparticles once they are added into the food product. What are the consequences? Let us have a look. Okay, next slide, please. Now, in the next slide, again, I'm taking you back to packaging. The reason is the chances of nanoparticles coming into our food and body is much, much higher than from the packaging rather than from the direct addition. The reason is because we use foods, almost all foods are packed, and this package after the consumption is actually discarded. And from once it is discarded, then it finds its path, which we will see. Now, again, if you see improved packaging, here what are incorporated are polymers with nanoclays and carbon nanotubes for better mechanical properties and tensile strength. Then we have active packaging. Now, active packaging actually is a function of packaging other than other than actually just containing the food. So we can have certain components which are absorbing the agents that are responsible for food spoilage. Like for example, oxygen, carbon dioxide, moisture, ethylene, or odor. So if there are any off odors or if there is excess production of ethylene, you can have active agents which will absorb this. On the other hand, you could have emitters which will release agents that can actually extend the shelf life. For example, antimicrobial agents like silver or flavors. And then of course, I mentioned about smart packaging, which are useful for the consumers to understand what is the real-time status of the food, and also it tells the consumer about the food quality and safety. So this is just a slide which briefs about the nanoparticles in food packaging. Now let us go to this next slide. The next slide is a slightly busy slide, but it encapsulates quite a bit. Yeah, please go to the next slide. Hello. Hello. Next slide, please. Yeah. So here I have already explained that engineered nanoparticles are manufactured, they are incorporated into packaging for multiple purposes. And now as a consumer, you and me go into the supermarket, purchase this product. What happens? We keep the product, we store it at home, and after we have consumed the food, there is an end of shelf life. So during the process of storage, there is migration of engineered nanoparticles from the packaging into the food, and this food is directly going to go inside our body after ingestion. And then what happens? These nanoparticles are there is a dissolution, either it will be eliminated or it will be absorbed. There will be an effect on the gastrointestinal fluids, it will be absorbed, and after absorption it will be translocated across the gut epithelium and it will enter the systemic circulation. And after it comes into systemic circulation, the potential interactions are the target organs. So this is when the packaging, food packaging containing nanoparticles and with food packaged is stored at home after the consumer purchases it. Then after the consumer has consumed the food, the packaging material is simply thrown away. It is the end of life or disposal. Now what happens to this package? It will either enter the technosphere or it will enter the ecosphere. So in technosphere, what will happen? It will go through sewage treatment plant or incineration or landfill. And again from the landfill, it will go to the soil. Then from the soil into the water and sediment. So it's a huge cycle. And you see the problem with an issue which started with manufacturing. It started with the manufacturing of nanoparticles, finding its way into the food packaging, purchasing by the consumer, holding it for the shelf life of the of the food product, and having two possible outcomes. One is migration of the nanoparticles into the food, which can actually interact with the target organs as explained, or it can actually go into the technosphere or ecosphere. Now let us see the next slide. The next slide is on, next slide please. This is on assessment of nanoparticles from food packaging. How serious it is or not serious. Okay, so if you look at this slide, there are some concerns on leaching of nanoparticles in food. As I've already explained that on ingestion, it could damage the internal organs and food is something which we consume on a regular basis. And if we like a particular brand, we continue to purchase that brand. And then what happens? Continuously, there would be an exposure of nanoparticles. Apart from that, these nanoparticles could also interact with the food constituents, and they could alter the food quality and safety. Now, this release of nanoparticles is actually dependent on the properties of the polymer. It's not that every packaging material will release it. The properties of the polymer are equally important. And for example, we have crystallinity, density, and degree of cross-linking and branching. So if there is branching, then the chances would be low. So we need to have an understanding of how the polymer properties are influencing the migration of these nanoparticles. The other important part as far as food is concerned, that if there are nanoparticles on the inner surface of the packaging, it can come in direct contact with the food product due to mechanical stress. So there would be packets one on top of other, and due to mechanical stress, the particles which are adhering loosely on the inner side, they can actually directly come in contact with the food. And not only that, the food product, it is not benign. It could be acidic, it could be alcoholic, it could be fatty in nature. So the food product itself can stimulate the release of surface nanoparticles depending upon the affinity. And for example, migration of nanoclay now from the package into food, this can be detected by the presence of aluminium or silica. And in fact, there has been work which has shown that MMT clay is the primary component of volcanic ash and bentonite. And we know that long-term exposure can cause lung disease. And if this MMT is modified with surfactants like diammonium dialkyl ammonium chloride, which is used to the extent of nearly 10% in ethylene-based polymers, they come in direct contact with the food. So it is not a hypothetical situation, a very real situation if it is included in the packaging. The next slide, please. The next slide will tell you, by the time it comes, I will talk so that continuity is maintained. If you look at the discharge of nanoclay, there are blow molded PET containers, which we see our carbonated beverages are packed. Please go to the next slide. So you will see that, there are reports where nanoclays have actually migrated from the blow molded PET bottles into carbonated beverages. These are reported. And unfortunately, there are no regulations anywhere on precise migration of nanoclay or silicon particles. People are simply thinking about the positive aspect, but nobody has yet thought about whether this should be regulated. And today, if you see the amount of engineered nanoparticles which are released in the atmosphere is approximately 8,100 metric tons, which is not by any measure a small number. And these nanoparticles, they are transported, diffused, agglomerated, transformed. And as Professor Pant said, they behave like an aerosol in the atmosphere. They also undergo chemical transformations like oxidation, sulfidation, photo-induced chemical reactions. There is not much work which has been done in this area, and they need thorough investigation. Then we have the intake of engineered nanoparticles by the organisms through normal feeding, through the gills of the fish, or sorption of engineered nanoparticles onto aquatic organisms. So you will find that when there is a coating of these particles, there is a coating of nanoparticles on or aquatic organisms. This is actually ubiquitous in nature, and you will find its toxicity might cause extensive adverse impressions on environmental health. Now, these nanoparticles actually generate a lot of oxidative stress by releasing ROS and RNS. That is reactive oxygen species and reactive nitrogen species. And they can have a massive effect on public health as well as biosphere. And let us not forget the climate. Yeah, I think you have come to the earlier. I need to see the earlier slide, please. Okay. So migration. No, you are going ahead. You missed one slide. Yeah, you have to go 2 slides ahead. Yeah, before this, please. So this particular slide, the one before this. Yeah, a survey had been done of 2,500 companies. All of them were using nanoparticles in food packaging, but they did not have any policy on its regulation. So the regulators are quiet, but the manufacturers who are responsible or answerable to the consumers, they are also quiet. And there has been migration of silver nanoparticles from PVC packaging to chicken meat. This also has been actually documented. And because there are no regulatory limits, so what is its effect on health remains uncertain. Then in today's times, all of us are busy and we heat up lot of our food in microwave oven. And microwave oven heating has shown even higher migration into the food. And this is again, there are 2 reasons. One is the microwave heating actually helps in detachment of the nanoparticles from the polymers, or microwave heating can actually modify the polymer itself, which will facilitate the release of nanoparticles. So this is how it is getting connecting to processing. Now, if you look at the next slide, so these nanoparticles, are they leading to nano pollution? And because they are also used for water purification, as very nicely explained by Professor Pant, and then an increased usage leads to increased level of nanoparticles in waste, and therefore an increase in nano pollution. Now, once these nanoparticles accumulate in the water, they can affect all the ecosystems on the earth via the water and atmospheric cycles. And these are actually connecting all of us on planet Earth. So it is a very dicey kind of a situation. Right now, we are only seeing the trailer. Now, that brings me to the next slide, how it is getting connected. Yeah, please come to the next slide. In the next slide, I am trying to explain to you, this is an experiment which has been done, how algae, zooplankton, fish food chain. You have come ahead, please let me complete the earlier one. Now, there has been contamination of gold nanoparticles in the experimental model, algal zooplankton food chain. As you can, yeah, please the earlier one. Yeah, in this slide, you can see that this has actually been demonstrated that there is a contamination of gold nanoparticles in the experimental model algal zooplankton food chain. This has been demonstrated. And these gold nanoparticles, they enter the body of a zooplankton grazer. So this is the Daphnia magna, which is the grazer, and the zooplankton is denoted in the figure up. So they will enter the zooplankton, and this zooplankton will then enter Daphnia. Now, how do they do it? With the contaminated plankton food and directly from the water. There is also plant uptake and trophic transfer of gold nanoparticles, and this has been linked with the structure of cell membrane and size. Now, this is just a diagram. I will not go into details. Yeah, please, it is there in the slide, but just briefly, I'll try to explain. Now, when these nanoparticles come into the soil, they can be taken up through irrigation. They can be picked up by the roots. And once it is picked up by the roots, it can interfere with the materials, with the organisms that are there, soil organisms. So both organic matter, salt ions, rhizobacter, or if it is used as a foliar spray, it will come onto the leaf. Once it comes onto the leaf, it moves through the plant. There are through various layers, it moves into the plant, and then from after this movement, it actually penetrates into the plant cell wall. And please, we all of us know that many of the plants actually are consumed as salad vegetables or even green leafy vegetables. We consume them. So this is how even through plant sources, we could actually encounter nanoparticles in our food. The next slide. Now, the next slide will tell you whether it is really, again, it is wishful thinking or imaginary thinking, or it has really happened. Now, in this slide, you can see that there is a soybean. In this study, this was done. Soybean plant is there and it was exposed hydroponically to silver sulfide nanoparticles. And the concentrations are given, and stable isotope enriched silver nanoparticles, that was used. And then it was monitored how much is actually going in the roots, how much is actually going into the leaves, and if these leaves are consumed by the snails, whether it is going into the snails. So to cut the long story short, it was found that about 29% of the silver in the treated leaves was accumulated from the root. So from the root, it came into the leaves, whereas all the silver in the soybean roots and untreated leaves were sourced from silver sulfide nanoparticles. So first, all the silver sulfide nanoparticles were taken up by the roots initially, and then after that it was taken up by the leaves. So this clearly proved that silver sulfide nanoparticles are phytoavailable, they translocate, and this was clear because they had used isotopes, 109 isotope of silver. And again, it was seen that, these leaves, when they were taken by the snails, it was found in the snails also. So this particular study tells us very clearly the importance of root uptake of silver sulfide nanoparticles in plant uptake, and then possibility of trophic transfer to herbivores. It also gives an idea about the understanding of biogeochemical fate of silver containing nanoparticles in the terrestrial environment. Now, this is a case study with silver, but there could be many more nanoparticles which could be evaluated, many more plants which could be evaluated, and this becomes an entire subject area by itself. The next slide, please. So I indicated that, we are getting exposed to the nanoparticles through the food, either from packaging or from direct ingestion. So what happens when we ingest it? So as you can see the normal path of ingestion: mouth, esophagus, stomach, small intestine, large intestine. And you will also see in the blood the concentration versus time after ingestion. So if you look at what happens, you will see that these nanoparticles interfere with the normal functioning of gastrointestinal tract, and there are references that it also inhibits gut microbiota. And these foods, if there are any pesticides which are there, which is bound to be there, they enhance the bioavailability of pesticide. They alter the location of the release of bioactives, and they also accumulate within specific tissues. So there definitely the physiology is not normal. Similarly, if you look at the profile after the ingestion, time after ingestion, there are ranges. One is the range, you can see the lowermost, which is no observable effect zone. Then if you have beneficial effect zone, and then you have a zone which is the adverse effect health zone. So these nanoparticles, for some time, they are definitely in an area which is indicative of adverse effect. So this is what happens when the nanoparticles come into our food, either by direct addition or after migration through the packaging. Let us go to the next slide. Yeah, the next slide, you can see, yeah, this is one picture, very small picture, and it tells you about the protozoa. See, these are two samples of protozoa, and the large filled food vacuoles, you can see vacuoles, and with a white arrow. So they have more trouble digesting the bacterial meals cultured with quantum dots. So, they are surrounded by quantum dots, and you will see that in the cases where there is no quantum dots, the digestion is happening. But in the protozoa where it was subjected to, or it was exposed to quantum dots, the food was not digested. So imagine even a tiny organism, you can see the difference, and you can imagine what can happen to the higher life forms. The next slide, please. Yeah, the next slide. So effectively, you see the cause effect diagram of nanoparticles on environment and human health. So I've already said that the environmental portion I have discussed, so I will not repeat it. But human exposure is not just through food. We are inhaling it, and it is also coming in our body through dermal route. So human exposure is through inhalation, ingestion, and dermal. And what are the consequences? There are reports of airway inflammation, that means blocking of lungs, allergy, even cancer, cardiovascular diseases, developmental toxicity, immunotoxicity, neurodegenerative disorders, targeted organ toxicity, which I explained, and respiratory diseases. Again, these are not imaginary, but there are reports where this has actually happened. Yeah. The next slide. Okay, this is actually to give you, this is just a figure. Next slide. The next slide will explain to you whether all the nanoparticles are equally toxic or some are less toxic than the other. So here you can see the green, red, amber, and yellow. So the ones which are yellow do not show toxicity. The one which are in amber color, they have intermediate toxicity. The one which are in green are not toxic, and the one which are in red are highly toxic. So you can therefore see silver is highly toxic. You can see over here cadmium, selenium, and the other portions you can see. And one has to really take a call on how to really evaluate the toxicity. So then you have to actually, once you know how to evaluate it, then you can actually take steps to either control the toxicity or design safer counterparts. Then we have ecotoxicity of nanoparticles, some facts which we need to understand. As I've already said, there are no defined guidelines and data for sustainable use of nanomaterials in order to protect aquatic life from probable hazards after nanoparticle exposure. And most of the work which has been done have been performed on titanium dioxide nanoparticles. In fact, I must add that European Food Safety Association has actually banned the use of titanium dioxide nanoparticles because of severe toxicity. So they have actually included in their regulation, and in the European Union now there is the titanium dioxide is no longer permitted. So toxicity experiments are on zinc oxide as well as silver. And because there are so many organisms, you cannot be studying on each and every organism. So some model organisms have been used to study. So it could be bacteria, algae, or crustaceans. And in most cases, people have used Daphnia magna because 90% people have used this organism. And there are very few reports on toxic effects of Moina macrocopa. Now, again, the toxicity has been studied only up to a certain level, but not beyond. So there is no detailed investigation on mechanism of toxicity. And it is believed that the concept of biomarkers, see, when you are exposed to toxicity, there will be certain biomarkers which could actually give you much better indications of toxicity. And these could actually be correlated with toxic effects. So we did do some work on this, and just in a couple of slides, I'll explain. Yeah, go to the next slide, please. Yeah, please go to the next slide. Yeah, so we used Moina macrocopa as a non-targeted aquatic organism. The reasons for this is because it's a lot of work has been done with Daphnia, and you need to widen the range of organisms. Secondly, you will see that Moina macrocopa has a very small size and it can be maintained easily. It has a short life cycle. It is very sensitive to pollutants, and it is widely distributed around the world. And it inhabits the upper layer of the water bodies, so that it is much more susceptible to nano exposure. So these were some of the reasons we used this Moina macrocopa as a model. And we evaluated the toxicity. Please go to the next slide. On silver nanoparticles, gold nanoparticles, and even on zinc oxide nanoparticles. These have all been published in different journals. Yeah, please go to the next slide. Yeah, I won't go into the details, but this is just to tell you that this work has been published. And yeah, if you go to the next slide, I will just give you a glimpse. Yeah, so you can see that this is this Moina macrocopa, and you can see 8 figures. So A, B, A and E are controls, B, F and C, G. These are 2 hours after the exposure to nanoparticles, whether you call it as silver or gold. In this case, it is silver. And you will see that, after 2 hours, the gut has started accumulating the nanoparticles. And after 5 hours, the gut is completely full with nanoparticles. Okay, so you will see that, so these are after different time periods that you can see, and these are at different levels. Like B and F are at different levels, C and G are at different levels. So one is, I think, 20, the other one is 40, and one is after 2 hours and after 5 hours. The sum total is that there is an accumulation of nanoparticles in the gut. And D and G, if you see, these are the dead organisms after exposure to a very high concentration. So, and if you look at the control gut, there is no accumulation. The next one. So the earlier slide, this slide is on silver, and the next slide, the next slide. Yeah, this is with gold. This is just to give you an idea that you can see even with gold nanoparticles, the gut is completely full with gold nanoparticles after a period of time. Next one. Next one. Yeah, this is again, accumulation of this Moina macrocopa after 0.51 and 2 hours after exposure to gold nanoparticles. Yeah, so after different time periods, you can see over a period of time, the gut keeps getting full with nanoparticles. So we had actually evaluated a lot of enzyme profiling we had done, and we could actually find that there was a down regulation of digestive enzymes, and there was also a decrease in the acetylcholinesterase activity. Acetylcholinesterase is a neurological enzyme, and there was a decrease. It also generated oxidative stress, and because of that, there was an increase in the antioxidant enzymes like superoxide dismutase. So these are assays which are possible in the lab. The next slide, please. The next slide. Yeah, after this. The next one. Yeah, after this. I explained this orally. So I will just summarize the entire nanoparticles in the food chain, food, human and environment, how we are connected in the last summary slide. Please let us go to the next one. Please, next one. Yeah, you can see over here that we have nanoparticles which are synthesized by different methods: chemical, biochemical, green, physical. They are used in the agriculture sector for pesticides, insecticide, nano-encapsulated micronutrient, various purposes. From the agriculture sector, it comes into the food. Apart from the ones which are coming from agriculture, we add some more. So either through the packaging or through additives or through nanosensors. Nanosensors will not be a problem because it may not come in contact with food. But food packaging and food additives together, along with that coming from the agriculture sector, will find their way into finished packaged foods. The products of these packaged foods reach the humans. And humans also receive nanoparticles from aquaculture, livestock, horticulture, and poultry. And all of them actually, then it goes up the food chain, if you see, from plants to animals to human. So across the, as you go upwards the food pyramid chain, you will find that the nanoparticles keep on migrating. So this is actually a one diagram which tells you the typical cycle of migration of nanoparticles from their source of synthesis to the agriculture sector, and finally accumulation in the human body through the packaged food. The next slide, please. Yeah, just some concluding remarks. The next slide, concluding remarks. So I would like to just conclude by saying that migration of nanoparticles from smart packaging, their accumulation into the organs, and their impact on the environment, they need deeper studies. There are long-term consequences of of accumulation of such particles over time, both inside the human body as well as in the environment. They have to be evaluated. They need attention. Then there is a need to set up some standardized protocol to determine the impact of nanomaterials in general and in food packaging in particular, using case by case approach. Particularly, now that we want to put the package in a microwave also, so we have to understand these things as well. There is required introduction of additional regulations. So right now there are no regulations. So introduction of additional regulations for proper handling and disposal to outweigh the limitations and gain consumer acceptability. Now, regulations cannot come unless we know the safety. So first the safety evaluation has to be done, after which the regulations can come into place. Now, I would also like to say that we are continuing to use nanoparticles in every single aspect of life, and an increase in the use and concentration of nanoparticles could become a problem which is analogous to microplastics in current times, or probably even worse. See, when plastics were introduced years back, people found it so convenient, and it is now that we are realizing that microplastics has even been found in breast milk. Now it has come to that level. It has been found in human fetus also. So you can imagine what can happen to the nanoparticles. So this nanoparticles is a microplastics waiting to happen in future, and we need to warn. We need to be careful to try and see that if we can prevent this from happening. So with this, some concluding remarks. I thank all of you. I thank the organizers, the authorities, for giving me opportunity to talk to you. To my namesake for coordinating with me, Dr. Rekha Dikshit, and to the audience, who have waited so long to listen to this information. Thank you very much. And if you have any questions, I would be happy to answer them.
So may I ask my question?
Yes, please, sir.
With the
Please go
permission
ahead.
of the chair. Yeah, I'm Dr. Chandrasekhar, working as Professor of Agronomy at UAS Dharwad, madam. Very nice to hear your lecture. I too started working on nanoparticles, particularly as a foliar and seed treatment in cotton. When I initiated my work, there were a lot of restrictions because we don't know what happens if nanoparticles goes through the eyebrows, eyes, lids, or through respiration. So they advised us to do the research in laboratories, and also utmost care is to be taken in order to avoid inhalation of these nanoparticles. But
yes,
nowadays, this nanourea is already recommended from the government to use on commercial scale in agriculture by the IFCO company. And similarly, a lot of research is going on nano zinc and nano iron particles as foliar nutrition in cotton or in any of the crops. Many research is going on. But will you not think without knowing the effects of or the bad effects of these nanoparticles, permitting the nanoparticle micronutrients or fertilizers to use in the commercial scale? What's your opinion?
Yeah, I will, what I think I will tell you. See, what has happened is many companies have invested lot of money into it, and they have chosen to see what they want to see. So they have tried to evaluate only the positives of, the outcome, and having invested a lot of money, they are now not, they are very hesitant to trace back their steps. So it is just that, they are not seeing what is obvious. And many of these investors or big company people, they are not the ones who are going to be working. Can you think of people where it can become an occupational hazard? So this is what
Yeah.
I feel. And I have spoken to many people on this topic, and that is what they say. We have done such a lot of work, we have established centers, we have spent crores of rupees, and now we are getting and they are also actually getting the permissions from the government by uh, portraying all the positive outcomes, which are no doubt positive. But what is seen, but there is a lot which is there which is not shown, even if it is seen. And the manifestations will not be clear now. The manifestations will be clear after some years, when the exposure levels or use levels will multiply tenfold. Did we imagine microplastics in human milk or in human fetus 10 years back or 5 years back?
Yeah. At that time, plastic was a fantastic material, and everybody used to like to use it and used it. Now we are seeing the negative effects of that plastic in the environment and pollution, the level it is causing.
My guess is that, sir, the same thing will happen with nanoparticles. Right now, because the exposure is not as much, but the signs are all there to be seen.
Yeah, particularly, I think the farmers are the ultimate sufferers, because they will not take the utmost care. They will not use any gloves or aprons, even goggles while spraying many of the pesticides. They use with bare hands, and they handle the material. And they are most susceptible, and ultimately it may enter to the food chain also. But on the contrary, if you see the genetically modified crops, because of the environmental protest, they're not permitted to release the BT brinjals that is developed even from our institute, from our university. They're not permitted. It has gone to the court, and still they want to know the effects of BT brinjal in human gut. So anyway, my feeling is that it should be confirmed. If it is not affecting the human body or human physiology, then it's well and good. We can encash the advantages with the nanoparticles or nanofertilizers. Or otherwise, if it is causing, then there should be some restrictions and control measures.
Agreed, sir. Very much. Thank you so much. Yeah. Any other questions? I think people will wake up only when majority of the population starts.
And you rightly said that though nanotechnology has now penetrated in every sphere of life, in medical, for imaging, diagnostics, therapeutics, in agriculture as nanofertilizers, and in environmental for remediation. But we have to look upon its negative aspects when it enters the food, and from the food when it enters of the human body. So nanoparticles, though they considered as wonder materials today, but after some time the negative effects will be visible. So this is the correct time to rein in and made some policies to regulate the use of nanoparticles. And rightly said that nanoparticles can enter the food through two ways, either by food packaging, what we are calling the smart packaging, which includes nanosensors, or some nanoparticles are added directly to the food as antimicrobials. Both are equally harmful. They can cause the effects which will manifest after several years, though not now. So we should be very careful, and we should not make the history to repeat as the microplastics is repeating, and we can find microplastics in human milk, in embryos. So though the nanotechnology is a very emerging technology, a wonder technology, and we should reap its benefits, but with some regulations. This is my feeling. Thanks, ma'am.
Yeah. Thank you so much. I think, when it is a question of life and death, in medical cases, it may be justified. But in all other cases, there could be better technologies or equivalent technologies which could be explored. That's what I think.
We are switching for nanotechnology. But by using nanotechnology as a future goal, we have to focus on the impact of this nanotechnology also. By this, now I am here to be announced session focus with two lectures based on nanotechnology. Now, at the end of this session, I Nilesh Kapoor, as the joint organizing secretary of this workshop, I will invite Dr. Anil Suri sir to provide a gratitude of love to co-chair of this session. First, I will invite Dr. Rekha Dixit ma'am. To host this event, I will invite Suresh Rani. Along with Rekha Dixit ma'am, I request Dr. Anurag Dixit sir to join Suresh sir to grace Dr. Rekha Dixit ma'am. I request Suresh sir and Anupam Dixit sir to grace with the memento to chairman of this session and co-chair of this sessions, Dr. Pankaj Kumar. Now I invite Siddiqui sir to provide a memento to the reporter of this session, Dr. Kaushalesh Ranjan sir. I also invite Dr. Pankaj Chauhan sir. Another reporter of this session, Dr. Manish Kumar Bharti, I invited you sir. Thank you, sir. Thank you very much, sir. Now, for the next session, there is a brainstorming and panel discussion session. I will invite Siroli sir to host the chair for this panel discussion session. Thank you very much, sir.
There is a small session, a panel discussion in which we will be inviting your independent views on a topic. For this, we have with us faculty members from College of Biotechnology who will be acting as panelists. I will request Dr. Pankaj Kumar, Dr. Rekha Dixit, Dr. Nilesh Kapoor, Dr. Pankaj Chauhan, Dr. Akash Tomar, and Dr. Naresh Pratap to be the panelists from Cornell College of Biotechnology, and Dr. Mahesh, Dr. Kashish, Dr. Siddiq, and Dr. Harshit to kindly represent as panelists from College of Veterinary Sciences to highlight the livestock position for the topic. I will also request some of our students and will be happy to have their opinions on the topic. Please feel free, Shainer and feel free to give your opinions. The topic I'm floating is pollutants in food chain, the major contributors to diseases in our country. You have listened since morning, right from inaugural session till last lecture, that pollutants, they are contributing, their environment is contributing pollutants to the food chain significantly, be it heavy metals, be it pesticides, plastics, and whatnot, we have been listening till morning. Simultaneously, if we look at the world, look at the countries in India today, the diseases in human beings as well as animals, they have also increased sharply. In human beings, there are many diseases, cardiovascular diseases, diabetes, blood pressure, and many other diseases. Similarly, livestock, my fellow colleagues from College of Veterinary Sciences are sitting here. So I request all of you to contribute your comments on this topic, independent comments as a panelist, so that we can record some of them in our proceedings. So first of all, I request the panelists from College of Biotechnology for their reaction. Before that, I will request Dr. Dikshit if we want to say something. Dr. Dikshit.
The system was since beginning actually I am saying on the various aspects of contents which are carcinogenic and private with so many diseases. And so every speaker own his or her own view and expertise about their expertise. So it is a global concern nowadays of these kind of contents which are very much carcinogenic to humans, and once they enter in the food chain, they get accumulated in the crops and other things, and from there, from the food chain, they go to humans. So we have listened many remedies to remove them from the environment. So they are mostly based on the high R&D work, so not possible for normal person. They are high tech.
निकल भी निकला है ना तो हमारे लग जाएंगे कस के। मलियत तो है ना अरे तो मलियत को उसमें थोड़ा ही ना है, वो तो ऑर्गेनाइजिंग कमेटी में है। ऑर्गेनाइजिंग कमेटी में तो हम क्या करें, हम तो पीएचडी वाले, हम तो आ गए। क्यों आ गए भोसड़ी के? नहीं
all of you have listened very carefully all the topics, and for students actually I think this is very good exposure to hear all these things, new things regarding pollutants, understand now types of pollutants, how they can be removed, what are the strategies. So with this I finish my works, and good luck for you, all of you, for students.
So you request students.
I am Anish Raskar, and currently we are working in microbiology. So I have solution for this. In the morning block, we were talking about wastewater treatment, and the thing is, the heavy metals are accumulating in groundwater because of excessive use of pesticides and insecticides. They contain heavy metals, and they are leading to bioaccumulation. Then they are going for biomagnification and contributing to big diseases. But I think if we use biofertilizers and biomaculants like rhizobia and other kind of microorganisms, with the help of that we can increase soil productivity and less use of fertilizer and chemical pesticides will be used. So that is what I would like to add. It will lead to major results according to me.
इंट्रोड्यूस योरसेल्फ करें एक बार अपना नाम और बोल दीजिए अविनाश अवनीश
As we know, so nowadays they are completely nanotechnology nowadays nanotechnology emerging with. Nowadays in our sector, in plant sector, animal sector, they are completely facing the problem of nanotechnology. So many nano particles we are using nowadays in different various researches. So nanopollution are a big issue. So it should be there how we can dedicate or we can make the guidelines for this nanopollution. We can save our livestock sector, our research. We are completely getting over the knowledge. Then nanoparticles are completely reducing the food chain in the animal sector. My question is that there should be some guidelines for these nanoparticles.
So even when we use our mobiles, laptops, touch screens, so they also have nanoparticles. So once we throw our mobiles, we do not know how we should throw it and where we should throw it. And they also contain a lot of that should also be. I am just adding to Dr. Mahesh that how that should be. That should also be important.
Not only nanoparticles, they contain lead, cadmium, like in their battery, mobile battery, heavy metals.
That was in my morning's presentation, electronic waste. That has to be disposed of.
E-waste.
We have seen the main lectures on the nanoparticles and its pollutants and so the current science and technology accumulates so. As far as we are getting different types of benefits from science and technology, similarly we are getting different types of problems of pollutants. So similarly, in case of the nanotechnology or the pesticide science or heavy metals, maybe they are equal in some other way, but they are causing different types of problems. Environment, animal health, and everything. Some of the heavy metals they are getting in the water, they are coming in pieces, they are causing some of the diseases like water disease. But ultimately, those heavy metals and pesticides they are coming in our body itself. Similarly, in case of the nanometals or nanomaterials also, so we are using different types of the nano devices like electronic devices, even some of the things we are using in the households, they are made up of the nanoparticles and nano-based technologies. If just we are rubbing our hands on the wall, millions or billions of nanoparticles are reaching in our body. But nobody knows that how these nanoparticles will react in our body. Either they will react with our nucleic acid, DNA, RNA, or proteins, amino acids, and how they will damage our body in coming time, nobody knows. So definitely a strict regulations is required for all these things. Thank you.
It was a moderate discussion we had throughout the day, and in this session, I would like to emphasize on the environmental impact on the particularly on the reactive oxygen species and associated diseases. Since we know these pollutants, they impact heavily on the human and animal body, and they impact on certain enzymes, particularly those who are responsible for the antioxidant activity of the animal and human body, particularly catalase, peroxidase, superoxide, dismutase. So it is heavily impacting on these enzymes, and even it is also impacting on one of the most important enzyme, that is acetylcholine esterase. So you know that the activity of acetylcholine should be replenished, and if it is not replenished, it may lead to certain even neuromuscular diseases, certain degenerative diseases. So it is heavily impacting on this. And as it has been proved that this reactive oxygen species is the major cause for many metabolic disorder, metabolic diseases, particularly diabetes mellitus type 2, eye disorders. It is affecting on the certain skin diseases, cardiovascular system, and many pollutants are nephrotoxic too. And as we know, many components of the our cosmetic products which are having paraben, methyl paraben, and triclosan. I have gone through even using one. And as it has been proved, regarding the use of the nanoparticles, it was very fascinating, and we were studying, and we were just learning about the nanoparticles that have the wonder impact. But today I came to know that it is very basis, and even may lead to the biomagnification in the human and even in the animal, and eventually lead to many toxic effects later on. So there is a need to address this situation very honestly, and to have certain restrictions on the use of this sort of chemicals, either in the healthcare products or for their or in the industrial use. So thank you.
Thank you very much, sir. It was a great, indeed successful event today. We are gaining the knowledge impacts of pollutant in daily life, accumulate in the system, and what would be the changes we can face, and what would be the thing we have to focus on developing for removal of such type of pollutant. As last speaker, Mr. Rekha Singhal talked about this ecotoxic effects of nanoparticles, and my fellow colleagues are also talking about the nanoparticles because we are moving towards this nanotechnology new era. But we cannot escape the things that it will cause the damaging effect in our near future. So, and whatever the level of damaging, it is much more higher than the normal pesticides or herbicides we are using nowadays. So we cannot ignore such type of problem. But in my opinion, there are alternatives how we can escape this type of toxicity level happen in future. Like one aspect I can suggest that we can use these nanoparticles in a conjugation. So that bioconjugations of this nanoparticle will target it toward the organism and it will not move to the next generation, and even it will not secrete inside the soil system and water system, so it will not cause the damage to the further system. Secondly, we can do one more thing that we can implement these nanoparticles directly to the particular plant or animal system where we want to target it, like conjugation with target specific protein or target specific characterizing moieties. We can target these nanoparticles to the particular organ so that it will move to that particular organ. It will enhance the growth of plant, it will enhance the animal growth, but it will not move to the next successive generation because we are not taking a complete plant as a complement. Whatever the animals they are thinking about we are reading about the leaf or the particular vegetable parts of that particular plant. So we have to target that nanoparticles in a particular plant system, like in food system or other system which cannot be consumed by humans or animals. So this will not cause the damage. This is my solution, and thank you once again to the organizers for doing such knowledgeable sessions. Thank you.
Less amount of pesticide for government should organize such programs by the agricultural institutes to the farmers to teach their knowledge about the use of these pesticides and fertilizers. Secondly, the members of the party has said very good that food are mostly the food is wasted from our houses only that must be preserved. Such programs or such advertisements should be go on to the radio or television to the families, which will encourage the family members to don't waste food so that that can be preserved for the population.
Thank you very much, sir. We funding we are here. We have the eminent scientists organized by the chairman and the co-chairman. First, your congratulations, my colleagues, to keep such that the development environment in impact of developments and carcinogens in future. Particularly today morning, Professor Abhishek talked about bioregulation. I think at present scenario we are living such type of standard we are living we are living with we are facing a lot of new states of particularly virus and the bacterial states, and this is very necessary to think and to search about such type of impact of new states particularly. And we talked about the bioregulation and biodegradation, and we were talking about the biodegradation. Indeed, at presently we have to spread there is a need to spread new strains of bacterial and fungal strains, because there is a such type of related varieties. Still, there is endangered in the soil. There is a need to spread that particular strain, that we can develop some new research, particularly about environment and impact of development. Today, such another speaker talked about drainage of mainly the drainage of some such toxic agents. Their disposal of drainage, the disposal of water, particularly in Gangas and other projects, is a very important problem. Because in this first type of a workshop, we are focusing on food chain. So we are talking about the safety of the food chain, particularly. Then we have to see the food chain, particularly food chain of aquatic systems. And one thing another scientist talked about the food safety. That is a very important issue. Food safety, and particularly as Dr. Gurun sir already talked about today's spreading of sustainable pesticides. By the convincing the farmers, there is a recommended dose, but over some farmers are not listening, and that's why we are going through several types of disease like cancer, carcinogens, depression, high blood pressure, and so on. This is the very problem. In this type of workshop, we should focus. We must think about that. And particularly very one speaker today very tremendously explained. And there's in fact there's a need in nanoparticles because at present there's the nanoparticles capsule. We are working on such type of research, but there is again issue that we should think about, as Dr. Vika Bishop mentioned about, that we should think about the side effects of such type of nanocapsules. And presently we are working particularly by ICMR results also. And food-based problem, one scientist have mentioned that there is very unfortunate to declare that 194.6 million from are based in India. So I think food issue and safety is very important. It's just that in the coming workshop we should think, and my another colleagues already explained that I think this workshop will continue and since that the research will go. Thank you very much.
In today's workshop, we introduced to several issues. One started from the types of pollutants. We also have there are several types of pollutants, particularly we have heavy metals, we have pesticides, and we have molecules. So out of all those things, most of these are utilized by human beings for managing several problems. We are using pesticides to manage insects. We are using many of those are heavy metals are also fertilizers or nutrients required by agriculture or humans. And also several organic compounds accumulate, they may be released by human activity, industrial activity, microbial activity, etc. So there are several types of pollutants in environment. One thing is important that they are pollutants only above a certain concentration. So they are not pollutants below a threshold level, but above a certain concentration they are pollutants. Now if we can manage that, the heavy metals, the pesticides, and the organic matter below the prescribed level, then they are not pollutants. So managing judicious use of all compounds and the proper disposal is important. The second thing is once it something goes above a threshold level and a requirement becomes what to say pollutant. It becomes a pollutant. How to remove that problem? So what we saw was that there are several strategies being used. Bioremediation is one of them, and using plants, animals, small insects, bacteria. These all these microorganisms also can be used to remove these pollutants. And once we remove these pollutants, these microorganisms again the point is that all the living organisms how to dispose of it. The organisms tend to be disposed of. Some organisms plant that pick up arsenic. Then the disposal of that plant is very important. It must not enter into the food chain. The second aspect in bioremediation was nanoparticles. What we saw was an important opportunity. It's important technology, and from these nanoparticles, either we can make it from carbon, carbon, activated carbon, carbon graphene, carbon nanotubes. And we are also seeing use of activated aluminium, microporous silica, and many other polycarbohydrates like cellulose and cytosine. So all these molecules could act as nanoparticles, and few of those nanoparticles may accumulate also if they are not very researchfully used. If that nanoparticles accumulate, then again it becomes a problem, which many scientific different institutions are trying to answer. That how these nanoparticles or bioevolution techniques could be used to control these nanoparticles. And we'd like to thank the speakers for giving us such an informative way. Any other person who wishes to speak? So now I request the chairman to.
ये प्लेटफॉर्म ऐसा है कि आपको अपने सबको करना चाहिए.
As we are advancing the technological world, the amount of pollutants are increasing day by day. And one thing I would like to talk about bioaccumulation. As we know, as plastics are deposited in our environment, and plastic causes deposition of toxins over it. So as that plastic enters in our food chain and food web, means it causes bioaccumulation of animal chain. As it moves our higher and higher in food web, it causes bioaccumulation. And one thing more, sir, this food web, as we move higher at the top level, the amount of pollutants will be maximum. Thank you.
Okay, thank you very much. Thanks everybody. So thank you very much for participating in this discussion, and we have got a number of good points which we can record in our proceedings. One thing is clear that these pollutants, they are playing a role in our health. Negative role, adverse role they are playing in our health. And one thing that emerged from the discussion is that strict regulations are required for that. Besides that, strict implication of those rules is more important. So this is one point which I think that has emerged. Another point is how we can control this. I think we have to reduce bioaccumulation so that further it doesn't biomagnify further. So for that matter, we have to think in two lines: either judicious use of pesticides and all those things, or secondly, we have to think for alternatives, eco-friendly alternatives like biofertilizer, as taught by Avinash, biofertilizers, biopesticides, which have less effects on the environment. So thank you very much. We will take your comments in our proceedings. And now I request Organizing Secretary Dr. Rekha Dikshit to please move on to further on the valedictory session to close the.
So we start the valedictory function. So Organizing Secretary, you would like to speak something?
Since I'm waiting since morning, I would like to discuss.
नहीं पहले कर लीजिए बात पहले आप वो इनॉग्रेशन को कंक्लूड
Request the chairman of this valedictory function, and we will have sessions reports first. So first we will have session report from session 1.
पहले इनॉग्रेशन
Dr. Rekha Dikshit, Organizing Secretary, will speak about the session, inaugural session. So thereafter we will have sessions report. Dr. Chairman of the first session. Second session, I will speak one about one talk, and Dr. Rekha Dikshit will speak. And then
We will have chairman.
We are having very fruitful, informative lecture series this morning in this brainstorming session and workshop on pollutants and carcinogens in food chain. In the morning, the session started beautifully with the question from Mark Sastati. So, and then Dr. Kumar, Dean, College of Technology, extended a warm welcome to the chief guest, distinguished speakers, Dr. Roshni, Dr. Hasina, Dr. Anupam Dixit, Dr. Manoj Tripathi, and these are all the deans of colleges and activities of the departments. And he just said he appreciated the effects of organizing appreciated the effects of organizing to create a vibrant platform to share the ideas on the impact of pollutants and the technological advances to offer solutions to this threat. After that, Dr. Rekha Dixit gave a brief report about the theme of the workshop, and the theme included various kinds of pollutants that are classified as carcinogens, and the methods to offer solutions to them, and that included pollutants of mycotoxins, heavy metals, pesticides, and persistent organic pollutants. Then it is addressed by Dr. Anil Surani, Director Research. He included that there is a serious threat to human and animal health due to the persistent pollutants. Actually, the pollutants can be divided into two types to explain. Some are biodegradable and some are non-biodegradable. Non-biodegradable pollutants are persistent in nature, and then they accumulate via the food chain. And at each traffic level, the concentrations get some increasing, and it becomes a cause of serious concern to the top predators in the food chain. So there is need to address this issue urgently, and then in this address, Dr. Our honorable chief guest, Dr. Akhil, he gave different. He gave a light at the end of the dark tunnel. That there are many recent approaches which are there to solve this problem. There are biosensors to monitor the level of the different pollutants in food or agricultural products. He also told about the different technologies which will address those issues. And also with our experts also we saw that there are many efforts by the scientific community to address this problem. For the environmental microbiologists, they are developing the consortium of microbes for bioremediation, and also developing bioreactors, installing bioreactors, ionic bioreactors, or aerobic bioreactors. And so there are many different, after monitoring, there are also regulatory policies in place by international and national committees. So after this inaugural exercise, chief guest Dr. Akhil, and the conduct model problem session community about of science in all and one, and the global security.
Thank you, Dr. Dixit, for a brief report about the inaugural session. So we had two sessions, and the first session was shared by Dr. Anil Surani, and he was shared with Dr. Akmal, though he had some health issues, so he left. So now I request the session chairman, Dr. Surani, to present the brief report about this session.
Thank you, Dr. Pankaj. The technical session 1 was chaired by me and Dr. Harshit Verma, recorded proceedings with me as a reporter. It was a very informative session, and there were three lead lectures by eminent personalities in their respective fields. The first talk was given by Dr. Anushri Malik, who is head of the department at Center for Rural Development and Technology at IIT Delhi. She talked on bioremediation of waste streams for agricultural applications. And second talk was Dr. Manoj Tripathi, who is principal scientist at ICR Central Institute of Agriculture Engineering, Bhopal. He talked on causes, effects, and solutions to the growing problem of food waste. And third talk was on basmati rice and food safety issues by Dr. Anupam Dikshit, who is working as chief scientist at BADF Modipuram, Meerut. The major recommendations which emerged from the session are first is to mitigate the problem of pollutants and carcinogens in food chain. There is an urgent and timely need of green solutions. The second recommendations that emerged from this session was for bioremediation of waste streams for agricultural applications in urban and peri-urban areas. Consortium approach or cocktail approach is a green solution where a combination of microbes, fungus, or bacteria may be used in bioreactors for water treatment to keep the pollutants in irrigation water at permissible limits. The cocktail in the form of tablets is not successful. However, in capsules with separate compartments, it is highly successful in the urban and peri-urban areas of national capital regions. Third recommendation that emerged was use of Bavaria bassiana, a commonly known bioagent that can be used in bioreactors for multi-metal removal from waste streams, and it can be adopted as a strategy to purify the polluted water in waste streams. Then native microbial consortium is effective in up to 97% removal of heavy metals from the water streams. So it will be better if we adopt native strains for the matter of bioremediation. Then food waste and food byproducts are the major concerns, as their disposal is not eco-friendly and hence leaches different toxins into the environment that affect food chain. Agriculture food waste and byproducts with the help of technological development vectors can be utilized into various useful applications like non-functional foods, cosmetics, supplements, and pharmaceutical industries, thus saving our environment by polluting it with the heavy metals and all other type of pollutants. Biotransformation of food waste is another strategy which can be adopted to various useful products like tofu whey, fermented soy milk, prebiotics, probiotics may be developed out of these byproducts with improved storage life. Then basmati rice is an important economic commodity where aflatoxins, heavy metals, and pesticides are posing serious threat. However, these pollutants are within permissible limits. For sustainable cultivation of basmati rice, massive awareness programs needs to be organized to aware farmers about judicious use of pesticides and fertilizers to keep residual effect within permissible limits, or novel eco-friendly pesticides and fertilizers with least residual effects needs to be developed. Alternative strategy could be to develop disease-resistant nutrient and water-responsive varieties of the basmati rice. Then monitoring contamination levels, enactment of control measures, including remediation and consideration of socio-political implications are vital to provide safe food. Awareness on pollutants and carcinogens in food chain should be intensified. Research is required to further assess and to find out other methods to reduce pollutants emission to the environment, hence ultimately the food chain. The protection of the environment and human health from the adverse effects of pollutants and carcinogens in food chain requires significant capacity in monitoring and testing of sources, environmental and production contamination, as well as human exposure. strict supervision of chemical production and use and disposal, including production of wastes, needs implementation. Monitoring of illegal trade of pesticides and other chemicals and other pollutants needs strict monitoring. Identification and application of alternatives and controls in food production system should be other strictly. So these are some of the important recommendations which emerged, and these recommendations will be compiled in detail and will be put on university website sometimes later on. Thank you very much.
Thank you, sir. So this is about the brief report of session 1. In session 2, I was co-chair along with Dr. Rekha Dixit, organizing secretary of this workshop. So in this session, 2 lectures are there. One from Dr. K.K. Head of Department of IIT Delhi, on fate of heavy metal contaminants in water and food and treatment. Another was on ecotoxicity of nanoparticles and its influence on food chain. So I'll be speaking about the first lecture, and the report about the second lecture. I will not take much time. So Dr. Pant emphasized how much scarcity of water, about 50%, 54% scarcity of water in India. Heavy metal, how it comes from natural resources, river and other sources, rains, acid rains, and through pesticides, detergents, sewage, etc. And they have the guidelines of WHO, how much critical values of heavy metals, WHO, US EPA, ISI, etc. Of the heavy metals which have been taken into consideration by these organizations. Then accumulation of heavy metals in food chain, how it reaches to the plant and it reaches to the different body parts of the human, animals, and other organisms. So lot of heavy metals comes from small scale industry, and why it is so? Because no treatment of waste material or wastewater, so it reaches to plants, animals, and products. Apart from this, spoke about personal care products and pharmaceuticals. Also, Dr. Siddiqui also emphasized on that how much we are using that domestic household products. They may be harmful if it is used in higher concentration. So PCPs are very important, and it contains natural hormones, and then different methods of water treatment. We emphasized like activated carbon, low cost absorbent, and nanomaterials. And different forms of absorbents also used. How they are synthesized, silver nanoparticles, characterization and utilization, efficacy, how we can use. And then carbon-based material for effective wastewater treatment, its removal of heavy metals, graphene and carbon nanotubes, utilize and modification of activated carbon and modification of carbon nanotubes. So we also talked about biochar for removal of arsenic and chloride and fluoride. And then about the toxic effect of planktons and the physical development and the chemical transformations of nanomaterials. So with this talk of Dr. Pankaj and emphasized and the recommendation of utilize these wastewater treatment. So now I request the co-chairman of the second session, Dr. Dixit, to give her report on second topic.
In the second session, I was a co-chair with Professor Pankaj Kumar and Dr. Vasu Children and Dr. Vishnu Happy. In these sessions, I was told about the foundations of the software of the TP Fund. I will talk about Professor Rekha Singhal from Department of Food Engineering Technology, ICT Mumbai. She gave talk on the ecotoxicity of nanoparticles and in influence and food chain. And she emphasized that when the nanoparticles enter to the food from the environment, then they migrate to our bodies and they can pose serious threat to our health. So, but how the nanoparticles are present in the environment? So she told about the three main sources from which the nanoparticles will enter the environment. One are the natural resources. Natural resources means very strong or forest fires, volcanoes, or oceans. And the man-made resources are cigarette fuel, smoke, diesel, vehicular emissions, cosmetic to personal care products. These are the source of nanoparticles in the environment. And also engineered nanomaterials, because now nanotechnology is used, the nanotechnology industry is growing very fast. So engineered nanomaterials are also the cause of the persistence of nanoparticles in the environment. And for the metal dust and the polymer fumes are also the another source of nanoparticles. And when these nanoparticles are present in the environment, they are present in the naked form and sometimes they undergo transformation. In waters, the nanoparticles may aggregate. They may homoaggregate. Homoaggregate means nanoparticles of same kind may aggregate, or they may heteroaggregate. Means nanoparticles of different types may aggregate. And other than this, they can also undergo photodegradation. So their form changes. And then when they enter the body, how they will interact with our internal organs, the transformed forms, which needs advanced research. So, and when this is the transformation in water, and when the environmental nanoparticles enter the soil, they when come in contact with organic matter, their bioavailability towards the organisms increases. So it is also a matter of concern. So she told about some of our research experiments in which they have done a research study, an interesting research study, in which the toxicity of nanoparticles in fresh water alga were studied, and they found that some toxic effects of the nanoparticles in the freshwater algae. So in food industry, when these are the examples when the nanoparticles are in environment and by transforming they can enter the human body. But when we deliberately use the nanoparticles in industries, for example, in food industry, the nanoparticles are being promoted that they can make smart packaging materials by using nanosensors, which will show the status of food, whether the food is getting stale or there is some problem, or whether when they are directly added to the food as anticated material or as a nutraceuticals. So all this eventually they end up into the nanoparticles entering our bodies and with some undesirable effects. So with all these things, another study, interesting study was the models, the different models on which the study of the effect of ecotoxicity of nanoparticles is being done. So she told about that the ecotoxicity studies being done on bacteria, algae, and crustaceans. And one important non-toxic organism, Moina macrocarpa, is a very useful model organism for the assessment of ecotoxicity, in which they found that acetylcholine oxidase is kind of decline, and they can use the decline in the activity of enzyme as a biomarker to assess the level and ecotoxicity of the nanoparticles. And after her talk, she gave certain recommendations that the migration of nanoparticles from smart packaging, their and accumulation in the organs and impact on the environment needs deeper studies. The long-term consequences need attention due to accumulation of such particles over time, both in body and in the environment. Next is that standardized protocols are required to determine the impact of nanomaterials in general and in food packaging in particular, using case-by-case approach. There is the dire need of introduction of additional regulations for the proper handling and disposal to outweigh the limitation and gain consumer accessibility. So, lastly, she said that the problem of nanoparticles should not become analogous to microplastic problem which the humanity is facing.
So thank you, Dr. Dixit. So after this session, we had brainstorming sessions. All of us were present, so we need to discuss again about that. Okay, and it will be on the repetitions. So I would request Dr. Anil Sirohi and Dr. Dixit to give certificates to some of the students. Only 10 we have compiled so far, and rest of you will be getting it. So Dr. Rekha Dixit will call the name one by one. आप आपके नाम बताइए, आप सर्टिफिकेट दें।
Come from Anand. Abhika Sharma. Nikita. Abneesh Srivastava. Ayushi Singh. Soumya Jaiswal. Soumya. Rahul. Sakshi Singh. Anshul Anand. Purna Mishra. Rani Singh.
second chance.
Now I request Dr. Pankaj Chauhan to Dr. Pankaj Chauhan, joint organizing. Actually, Dr. Nilesh Kapoor. Director Research and Patron of this workshop. Secretary and
also the organizing chairman.
I thought I would my So now I request the chairman of the function to give the story, please.
I'll not take much time. Thank you all. I think the brainstorming session and workshop for the purpose it was organized was successful in communicating its message, that is food pollution in food chain. So at the end, I would request all of you that message is well conveyed. All of us are at risk. Be ready, work for that to have a safe food and safe generation. And second message from today's workshop was, it was from food-based lecture. Waste is a resource. Invest it efficiently. So have safe food, simultaneously safe food. Thank you very much for joining us today. And my special thanks to dear students who have been here for a whole day. And I will request in future you to be the part of our organizing committees in organizing such kind of events. Thank you very much.
Thank you, sir. So morning mobilization, I give vote of thanks. Now organizing secretary is requested to give vote of thanks. Though she is emphasizing that I should say, but it is the organizing secretary who has.
But because I'm talking since morning, so I said everybody is getting vote.
Madam,
But actually, from the bottom of my heart.
मैडम आपके थैंक्स से पहले मैं ये एक क्वेश्चन पूछना चाहूंगा कि मुझे एक आइडेंटिफाई करके बताइए कौन बोर फील किया। मुझे तो कोई नहीं लगा जो बोर होगा।
But for the bottom, I'm thankful to all and each and everyone who participated to make this workshop successful for all of the participants. And first of all, I'd like to thank Honorable Vice Chancellor, Professor Architecture. He is the main instrumental in getting this IT grant and this new project. And just guiding throughout since the backing down of the project.